Hello, everybody. Welcome to the January AgConnect. And as we kick off 2021, I'll just tell you a little bit about what we're doing. First of all, I'll tell you that my name is Al Kriz with Bayer. We've been sponsoring AgConnect as a networking opportunity for the past year and a half or so. Of course, we're gone virtual. And what we're doing now is taking AgConnect and expanding that out from a kind of a smaller networking session to a larger opportunity within Bayer to be able to connect with the external community, particularly with the focus on ag. So what we're doing today is we have a great lineup, a symposium focused on cell biology. So you're going to hear from some great speakers here coming up. So what I'm going to do now is transfer over to the actual host of the Cellfies Symposium, my colleague Annie, also from Bayer.
All right. Thanks so much, Al. Welcome, everyone. Good morning and good afternoon / good evening for any folks that are joining from our European sites. And so as Al mentioned, my name is Annie Saltaricos. I am the Biotech Transformation Lead here at Bayer Crop Science, and I'm very pleased to present the Cellfies Symposium today. And with that, it takes a village to put this on, and I'm very thankful to the Bayer Life Hub Boston and the Bayer AgConnect community for being able to host this symposium today. And so I want to start with some acknowledgements before we kick it off. Again, really thanking the community for joining us today, and also Bayer Life Hub Boston for having these sessions. I think it's a wonderful opportunity to hear a lot of great talks today, as well as interact across our community of individuals that work in plant biology. I also want to thank all of the symposium organizers, along with myself, Pat Boyle, Edward Cargill, Yurong Chen, Larry Gilbertson, Al, who you've just met, Hua-Chen LaRue, Tom Ream, and then Pei-Shen Yang. I also want to just give some quick thanks to all of the folks that worked really hard on making this available today. As we're all working in our virtual environment, we're learning how to work and how to communicate through these different forms. And so really want to thank Jody, Meredith, Michael, and Michael for helping us today as we were getting set up. I'm going to go through the agenda very quickly, but I also want to do a big thanks to all of our speakers that were able to attend today. We walked through our slides this morning and made sure everything was connected. So again, we're really excited to have such a great format to offer today. And then last but not least, thank you to all the participants. We're really looking forward to your active engagement and in time for these talks today. So very quickly, just wanted to again, welcome to the Bayer AgConnect community. And this is our agenda that we have today. So again, I'm Annie, I'm going through the introduction. I'm going to pass it off to my colleagues to introduce some of our speakers today, but we're going to have Michael Strano from MIT talking about Michael and Lanying are actually going to talk about different advancements and different types of delivery systems for plant biology. So we're going to hear more about nanoparticles and then about agrobacterium and other non-agrobacterium delivery systems. So that's going to be our morning session. We do have some breaks built in. You can see here all the times that are listed on the agenda are Central Standard Time. So adjust accordingly if you're in a different time zone. But we're going to have a break, and then Mark Johnson and we'll have another break for lunch, and then Mike Scanlon are going to talk about some key plant biology areas, specifically developments in pollen biology as well as maize shoot development. So look forward to that. And then in the afternoon, as we go through the sessions, we're going to have Yiping Qi and Dan Voitas talk about our gene editing tools and then plant genome editing challenges and some opportunities there to capture. So stay tuned for that. And again, we're really looking forward to these talks today. I also just wanted to do some logistics and reminders. So this event is being recorded, as you can see on your teams. For Bayer internal folks, we just want to remind everyone this is a non-confidential discussion, no CDAs in place. So just a reminder there. For folks that are maybe not as familiar with using Teams, we just want to show some logistics here that you can post your questions in the Q&A format. It may look a little different on your screen, but what you're going to see is an opportunity there at the bottom in purple to actually ask a question. We're going to take questions during the Q&A. The Bayer host will help to moderate those questions, and that'll happen at the end of each seminar. So again, if you have questions, please encourage you to post them in the Q&A, and we will get to those questions. And then again, it just as a final reminder, there are 3 scheduled breaks, and we're going to do our best to stay on time today. All right. So with that, actually, I'm going to now hand off to my colleague, Larry Gilbertson, and he's going to introduce our first speaker.
Good morning, good afternoon, good evening, everyone. I'm Larry Gilbertson. I'm a scientist in Bayer Crop Science. I lead our team in applied genome modification, one of our gene editing teams in Bayer. And it's my great privilege and honor to introduce our first speaker today, Dr. Michael Strano. Michael is the Carbon P. Dubs Professor in Chemical Engineering Department at MIT, and I had the pleasure of meeting him there in person a few years ago, so I'm delighted that he's here. By way of background, I'll give a little bio here. He got his bachelor's degree from Polytechnic University in Brooklyn, got his PhD from the University of Delaware in chemical engineering, then went on to do a postdoc at Rice University in chemistry and physics departments. From 2003 to 2007, he was an assistant professor at the University of Illinois before moving on to MIT, where he is now. Michael is a recipient of many prestigious awards, including the 2005 Presidential Early Career Award, the 2006 Beckman Young Investigator Award, the 2006 Koblenz Award for Molecular Spectroscopy, the Unilever Award from the American Chemical Society in 2007, and the Young Investigator Award, and the 2008 Alan P. Colburn Award from the American Institute of Chemical Engineers. He was also elected to the National Academy of Engineering in 2007. And today he's going to talk about applications of nanoparticles in plant biology, nanocarriers or nanosensors. And just as a reminder, as Annie just said, if you have questions, please put them in the chat. I'll be monitoring the chat. And then at the end, we'll save some time and I'll moderate a brief Q&A. So with that, I'd like to turn it over to Dr. Strano.
Thank you very much, Larry. I appreciate being here. I'm honored actually to speak at this symposium, the Cellfies Symposium. I am not a plant biologist by training, and every time I attend events like this, I'm humbled, and I get reminders of how little I know. So don't let those awards fool you. In this space, I'm very much still a student, so you should view me that way. I encourage you, if you have questions, please email me after the talk. I put my email address on the slide. Hopefully I'm showing my slides. Can someone verify? Is my screen shared?
Yes, we see your slide.
Excellent, great, good. And if you put questions in the chat, the organizers can send those to me. I want to answer each and every one of your questions. And there may be a lot because I'm a chemical engineer by training. My field is nanotechnology, and I moved into plants. And so what we're seeing is a merger of 2 fields, and I'm very cognizant of how little I know. I want to learn from you, and hopefully we'll learn a language. And I think I have some things that we've learned, but there's far more that I want to learn from everyone at this conference. So my talk today is applications of nanoparticles in plant biology. I come at this from the nanoparticle perspective. I work in nanotechnology. It's hard for me to thank all of my students, but the work in this talk is from a recent graduate, Tedrick Liu, now doing a postdoc in Singapore, Minho Wang, who went to Boston Consulting, my current students, Ming-Kyung Park, Alan Brooks, and Leslie Q. Sun Kwok is actually a faculty member at Seoul National University. Makita Landry, now faculty at UC Berkeley, and Juan Pablo Geraldo is at UC Riverside. So I just want to acknowledge their contributions. And I should point out, if you don't know, Larry mentioned that we met at MIT to a new audience, I'd just like to introduce, I'm in the chemical engineering department at MIT. Of course, MIT is in Cambridge, Massachusetts. We're one of the first PhD degree offering chemical engineering departments. Actually, we're the first in 1920, and about 10% of chemical engineering faculty around the United States are from MIT. So we're a top ranked department, but remember that we're engineers, right? So so we're still novices in this plant biology space. Our building is a 30, 60, 90 degree triangle. So if you remember that from your high school days, that we're very much still engineers. And I think MIT characteristically, one of our claims to fame is our students. They're very industrious. They're very creative, and you'll see that here in this work. They pull these pranks. We famously stole Caltech's cannon one time. This is a prank where the students put a Cambridge police car on the top of the dome. This is actually a real photo. It's not photoshopped, and I like to start with that to highlight that we need to keep our students busy doing constructive things, because otherwise they'll pull these harmless pranks that are really flashy and showy. There's a long tradition of this going back since before I was born at MIT. So I would like to thank my group. This is an old photo. Of course, in the past year, we haven't been able to assemble on the mall, but I look forward to a time when, when the pandemic subsides and we can update this photo of the research group. I would say about a fourth of my work now is focused on plants, and I'm going to talk. Before I talk about the topic of my seminar, I just want to highlight some things that we work on. We're a nanotechnology laboratory, and we like to invent new things, do nanotechnology to solve problems. This is we invented a new kind of neoprene. It's actually a low temperature. It's actually the warmest garment that has ever been invented. It's a low temperature dive suit for the Navy, and we used nanotechnology and concepts that are related to enable this, what we call an artificial blubber. One area my group is working on are cell-size robots. These are tiny cell-size particles that have electronic functions, and it's a new and developing area, and we're very excited. But again, it's an application of nanotechnology to solve problems, combining microelectronics and colloidal science. And I work in the area of nanotechnology and thermal energy as well. We invented this technology called a thermal resonator. So these devices take temperature fluctuations from the atmosphere and turn them into electricity. And so few people know that you can do that. You can actually just use the random temperature fluctuations that surround a building or surround you can convert to electricity. And we invented the theory of this, and we invented the devices. It's called a thermal resonator. And so that's an area where we work on. I'm not going to talk about any of that today. I'll talk about our plant work. And journalists have certainly helped to overhype it. So I'll say that right now. They called us MIT geniuses to make a bomb-sniffing spinach plant, or we're working on a glowing plant, a light-emitting plant, but we also have made tools that can help agriculture, and that's really what I want to focus on now. But just if you're following along, I wanted to just highlight that my interest in plants, my perspective in plants, is really from an engineering standpoint. And so, just so you understand where I'm coming from, my interest in plants started maybe 10 years ago as the starting point for technology. So I noticed engineers tend not to think about living plants as a starting point to develop technology, but from an engineering perspective, they have a number of advantages. Of course, plants harvest energy through photosynthesis. They also store it in saccharides and starches. So they're a combination of, say, a solar cell and a battery. They have access to low energy water flow, just through transpiration. They're already adapted to harsh environments. So I'm trying to think about if you were to make technology that can go out in the environment and monitor things remotely, or become devices. They already can exist in fairly harsh environments with relatively little additional engineering. They have this property of autonomous self-repair, and I have some projects trying to make human versions of that. It's fascinating. The ability for plants to repair themselves. It's amazing. They're doubly negative carbon footprint. So I say doubly negative because they're made of essentially carbon dioxide from the atmosphere and other carbon sources. But then they also consume it as fuel. So it's kind of like if you had a car made of wood that ran on biofuels. It's doubly carbon negative. Of course, it's easy to recycle. So the reason why I'm going through this is because my perspective on plants, even from back when I started interacting with Larry Gilbertson, is I want to see to what extent plants can replace the things that we stamp out of plastic and circuit boards, right? It's a good question to ask today. It's sort of a sustainability question, but and I don't know the answer to that question yet, but I'm interested in the engineering rules. What would you have to do to turn a plant into into a sensor or Wi-Fi repeater or a lamp or optical communication device? What would you have to do? And that's part of my perspective. And then just from a historical standpoint, I started with my interest in the chloroplast, and then I'll get right. This is actually the start of I'll tell you about a very powerful theory that we developed in this way that underpins all of my work on nanoparticles and plants. So if you bear with me, I think this story is actually worth it. So I became interested in the chloroplast, extracted chloroplast. Those of you in the audience have probably, it's possible that you've worked with them. So it's an interesting organelle. It can exist outside the plant. Once upon a time, photosynthetic bacteria incorporated into the plant cell. It does this very important chemical reaction, this carbon fixation reaction, which you could argue more engineers should be studying. So from an engineering perspective, actually, I was surprised how easy it is to extract chloroplast. So you can take spinach. A lot of our early work was buying spinach from the grocery store. You can grind it. You do this Percol separation, and you get these things that look like mammalian cells. They're large, but they're relatively stable, and they'll still do this chemical reaction. And that really was what got me interested in plants. I was amazed that this little lipid bag of catalytic proteins could do this amazing chemical reaction. And I just noticed for the engineers in the audience, if you look at the number of publications, there are very few engineering publications on the chloroplast. So there was a Nature paper in 1980-81 on electrochemistry of mostly broken chloroplast, but very little work. A lot of work on photosynthetic bacteria, a lot of work on the reaction centers, as you can see. But anyway, so I took this. So we started looking at nanoparticles and the chloroplast, as a way to engineer them, and we found something very, very strange. And this was about the time I started interacting with Larry Gilbertson. So if you flow certain nanoparticle solutions across a chloroplast, they'll actually enter the chloroplast through the double lipid bilayer membrane, and they'll become kinetically trapped inside. So these are DNA-wrapped carbon nanotubes. There's an enormous literature on that. And this is a chloroplast that you can't see yet. And then as these particles are flowing past, they're actually entering the chloroplast, and there's so many, and they're near infrared fluorescent. So what you're seeing is the fluorescence of this chloroplast just lightens up because it's trapping so many particles. This is not expected, and there's a lot of discussion in the literature. I think it took a while to convince reviewers that what's really happening is the particles are going through this double lipid bilayer membrane and becoming kinetically trapped. It seems to traverse this barrier so easily. How can this be? And It led to some discussions back and forth. Some prominent plant biologists emailed us and said, what's going on here? There's a tremendous amount of literature data here. Like, one of the things you can do with nanoparticles is you can do what's called single particle tracking. And so, I mean, this is, so you can actually see here, this is also one of these DNA wrapped carbon nanotubes. You can actually follow it as it moves into the chloroplast and then bounces around. In other words, it shows confined diffusion. You can do confocal methods to show that it's not trapped on the surface. It's clearly inside. So the question is, what is going on here? Because this process can work even if you infuse certain nanoparticles into the vasculature. So this is a very common technique. You can take a nanoparticle solution, you can pressurize it to the leaf lamina, and you can actually backfill the mesophyll with your nanoparticle solution. And then depending on the type of particle, you can still see here. So here you're seeing the chloroplast, the fluorescence from the chloroplast. Here you're seeing fluorescent carbon nanotubes, and you're seeing that they can be localized even in vivo. So this mechanism, whatever it is powerful enough to place nanoparticles into this organelle, even from in vivo. So this led to a lot of questions. Our work, our nanoparticle of choice at first was single-walled carbon nanotubes, but the behavior I'm going to show you extends to really any particle, any nanoparticle, natural or synthetic or derived from any material. It can be anything. So if you to get a clue about what's happening, if you take a carbon nanotube, if you take a nanoparticle actually, and it has a strongly negative zeta potential. Well, oh, sorry a minute. I didn't. Let me see here. Sorry. There we go. Yeah, if you take a carbon nanotube and it has a strongly negative zeta potential, okay, then you'll see here. Here's the chloroplast. Here you're seeing lots of fluorescence. So this goes into the chloroplast. But if it's weakly negative, like this PVA SWINT, it's still negatively charged, but you don't find any evidence of the nanoparticle inside. Positively charged, if it's above a certain threshold, will also find its way inside. And then perhaps a very important clue is if you wrap the carbon nanotube, if you wrap the particle, the carbon nanotube can be wrapped in different things to change its charge. If you wrap it in the same lipids that make up the chloroplast, you don't get any uptake. So this was a very important clue. If you wrap the carbon nanotube in phospholipids. So just to summarize this, I mean, this is a result now that has been reproduced over and over and over again. So you've got negatively charged, and you see where the nanoparticles will localize. You can compare and contrast this with PVA. This is now an extracted protoplast, so we get a clearer picture of what's happening. So these PVA SWINT, they're negatively charged. They don't even go inside, so they're nowhere to be found. But you can find other examples where you're positively charged. You can find particles that are going inside. You can even find particles that seem to have sufficient charge, like negatively charged. These quantum dots, these streptavidin-encoded quantum dots, they're negatively charged, not as negative as, say, the DNA wrap. They will actually go into the protoplast. Notice that they will. They seem to go into the cytosol. They won't go into the plastid compartments. So, and you can see that here. You can do gold nanoparticles. So this is a very general mechanism. And what is going on here? So highly charged nanoparticles can localize in the chloroplast. More neutrally charged can wind up in just the cytosol, or in some cases, not all. They won't even enter the protoplast. Okay, so here's what we understand to be happening. This LEAP is a quantitative theory that we've made. We don't have to go into the math. This is the mechanism that we think is happening, and it seems to work on almost every nanoparticle. And this technique appears to work on a broad range of plants. I don't know if it works on all monocots and dicots and all the, but it's a very large number. So a nanoparticle, whether it's negatively or positively charged, it's going to produce a cloud. Under biological conditions, it'll produce a cloud of ions. So if it's positively charged, it'll attract an ion cloud. If it's negatively charged, it'll attract an ion cloud. Okay, the cloud contains both the ions and their counter ions. So this ionic cloud, when the particle impinges upon the membrane, it's known that this change in what's called the ionic strength will soften this membrane. Okay, this is very well known. Biophysicists have done detailed experiments of pressing on lipid bilayer membranes, changing the salt concentration, and this membrane softening is very quantitative. We understand it very, very well. What we propose that happens in a nanoparticle is that the softened membrane then undergoes a form of non-covalent chemical reaction. We call it a lipid exchange. So lipids from the membrane can exchange and bind to surface to unsaturated sites, if you will, on the nanoparticle. And as they do, they'll bring the particle inside the double lipid bilayer membrane. Okay, the membrane reheals, but then what happens is the particle itself is now a lipid complex nanoparticle. It now lacks the driving force to immediately traverse the boundary back again. So it becomes kinetically trapped, not forever, but it's under the duration of the experiment, it's actually trapped. And this explains a number of things: why plus and minus both seem to work, why the charge has to be big enough. It explains why there's this irreversible trapping, and it's a completely passive mechanism. It explains why it's going to work on a wide variety of plant species, and then also extracted chloroplast. Okay, I am actually going to skip over the math. This is not the audience for the math, and that's not to say anything about your ability. It's just not central here. But what I want you to understand is that LEAP puts equations to all those mechanisms and adds them together, and it gets this. It's an equation, and it predicts the charge on the particle. This zeta potential here. It predicts the charge on the particle required to traverse one of these boundaries. Okay, and the other thing just to point out is that all of the parameters, in theory, you can calculate or measure independently. So it is a fairly pure theory. We're not fitting anything here. You can there's the nanoparticle radius is known. The lipid membrane properties are known. Its thickness, membrane tension, surface energy, all of those are known. Thermochemical properties are known. So you have this equation, and it makes these blue lines. So these blue lines, it predicts the boundary across which, that separates particles that will go through the layer and not. And so, for example, our positively charged chitosan swint appear to cross the boundary. This lipid PEG swint won't. And so this blue line plus and minus controls what goes through the chloroplast. In subsequent work, we did this for protoplast too. So you can use the same equation, and I have more on that on the next slide. You can use the same equation for the single lipid bilayer membrane of the extracted protoplast, okay? And you get, in this case, you get, yeah, sorry, I should have made it a different color. In this case, blue is the protoplast boundary, red is the chloroplast boundary. And what you can see here is that we predict, even when others have seen, we predict nanoparticles that will enter the cell, but not go to the chloroplast. We predict particles that enter the cell, wind up in the chloroplast. We predict particles that will do neither. So it's a powerful theory. If you notice, it doesn't take into account the species of plant. That's because, to the best of our knowledge, it appears to work on almost every plant species we've tried. I think it's an active area of research to figure out which plants it doesn't work in and so forth. But it's a quantitative theory, and I'll show you how it works. For the protoplast, we took the membrane properties that are known for chloroplast. If you plug that in, you'll get the red line. For the protoplast, it's a thinner membrane. It has a different dielectric constant. You plug that in, you get the you get the blue line. So I mean, it's a very simple theory, but it seems to be extremely quantitative, and we can even put data from other people's papers, and it fits right on. This theory fits right on the data. So we think it's the theory itself is too simple to it's simpler, and it seems to extend. It extends to almost every nanoparticle we've tried, and almost every plant species we've tried. And the theory is too simple for us to deserve that. So we are constantly looking for exceptions, and it will definitely need to be modified. And we made the cover of this nanotechnology journal small for extending this theory that we developed to the protoplast. I'm going to try to stay on time. I'm going to skip this slide. We definitely turned our attention towards some of the longstanding problems in plant engineering, just by interacting with folks like yourselves, talking with Larry, and they're saying, hey, it'd be really interesting if you could do this or you could do that. And there are these longstanding challenges, particularly for delivering things like genetic material. We're going to hear about enormous advances on those fronts from the other speakers. I'm really looking forward to it. Our part of the technology is very, very small part, but we're going to hear about advances in agrobacterium mediated plant delivery to the cell. My understanding is that maybe this is fixed, is that this is often a technique that's limited from species to species, or requires a lot of work and innovation to get it to work with a particular plant species. The gene gun, the ballistic delivery, nobody has kind words for. Everybody that uses it hates it, and they say it's a non-selective bombardment. When it works, thank goodness, but it's a technique that I think everyone in the field, at least that I've talked to, is very interested in getting rid of. And it's interesting that it does in fact use gold nanoparticles. It uses particles that could be nano. And so there may be a leap type mechanism that's ultimately happening here. But that's complete speculation. I haven't. Okay, so can we design nanoparticles that target different subcellular environments, particularly in a species agnostic way? And this, I think, is something exciting. So we asked this question. This was almost a side project for us. We designed some nanoparticle carriers. We compared a number of them, but the one that works the best is a carbon nanotube that you functionalize with a chitosan. And this makes a positively charged nanoparticle. If it's sufficiently charged, it'll go right into the chloroplast. But in theory, you could the reason why we chose chitosan is because it's positively charged. You could take a plasmid DNA, which is negatively charged, and you could complex it. You could change that ratio to increase the net charge. And so, and when you know it, it works really, really well. And it follows the LEAP model perfectly, right? So one of the things that you can do is you can take the nano carrier, which is positively charged, you can change the ratio of plasmid DNA to carrier, and you can make the resulting complex more and more negatively or positively charged, right? So so this shows if you do a one-to-one ratio of DNA of plasmid DNA to the nano carrier, you're positively charged, but you're not positively charged enough to actually go into the into the chloroplast. But if you at a 1 to 3 and especially a 1 to 6, so you see this here. This is a 1 to 1. I'm showing here at the bottom a 1 to 6, a plasmid that encodes for yellow fluorescent protein in the chloroplast biosynthetic machinery. And you're seeing here you get yellow fluorescent protein expression, in the chloroplast here. So not only is that result interesting, but it's completely in line with LEAP. So LEAP is predictive and it's quantitative, and but every time it worked, it surprised us. It surprised. There are many reasons why this could have failed. To me, it continues to be amazing how this equation has some predictive value, right? So and not just that it delivered this cargo, but it predicted exactly when, under what conditions would it traverse this chloroplast boundary, right? And you can see here, this is actually in a mature our arugula plant. This is just additional data using confocal. So we have the tools of nanotechnology that convince us, but then I think we need to use plant biotechnology tools to also convince. And so what you're showing here is you're showing yellow fluorescent protein where the particles actually wind up in the chloroplast and merging these. So reviewers asked us, they said, well, what about other plant species? And I think we did a total of 6 or 7 species. And so the results were positive the same way in arugula, watercress, tobacco, spinach. We have since gone on to use this type of technique in rice, and you'll see data from oil palm and all these. So I hesitate to say with certainty that there is a I hesitate to say with certainty that the tool is completely species agnostic, but it certainly works for almost every nanoparticle we have worked with, and almost every plant species that we've worked with, and including some in the literature that others have worked with. So it seems to be fairly broad. I'm gonna skip over these 2 slides. Larry said there would be interest in particular in our work on gene delivery to oil palm, and this is actually a brief project that we had sponsored by Sime Darby, and that project has ended, but they're very excited about the results, and so they gave us oil palm pollen. Pollen has different structures and shapes and sizes. I'm by nowhere near an expert. I'm actually looking forward to some of the talks today. But for those of you who aren't familiar, the oil palm produces palm oil. It's an enormous industry globally, and there are longstanding challenges in oil palm that are very well known. Always trying to get high oil content and to increase the quality of the oil. Here, actually, we did through some engineering and some trial and error. We actually used to get a sufficient charge. We ended up using carbon nanotubes that were functionalized with this imidazolium group, which I'll call IIM. So it makes a positive. It's the same basic principle. It's a positively charged, and then you can complex the negatively charged plasmid DNA to it. And what you find is here now. What I'm showing you is an optical micrograph of the pollen that is germinating, and what you're seeing here is you're seeing the green fluorescent protein overlay of the plasmid that we delivered. And if you go through our paper says submitted, but it's actually in print. I'm sorry, I noticed that I didn't update that. You see that again. You see this threshold type behavior. The 1-to-1 ratio does not show much green fluorescent protein, but then you get you go to 1-to-2, 1-to-4, 1-to-6. You can clearly see a threshold between 1-to-2 and 1-to-4. Now, remember what that is, though. That's the net charge on the particle, which is what LEAP very simplistically, maybe overly simplistically predicts that it's really just the charge on this particle, right? When you have sufficient charge. Now, we don't know. We don't have precise information on the barriers in pollen and oil pollen. So I believe we don't quite know what the dielectric constant of the membrane is. So for our paper, we said our results are consistent if the primary barrier within the pollen has a dielectric constant, say, between that of a chloroplast and a protoplast. And its dielectric constant is, say, between about 3 and 4. And so it seems to be consistent. So, but there's definitely evidence for a threshold. And we also went on in the paper to use inhibitors of endocytosis, and we showed that it had no effect on the results. So there's definitely evidence for a passive mechanism. It sure does look like LEAP, and if someone can go in and measure more of the dielectric properties, in the pollen, if you can unfold it and study it, then you could confirm that. So it's definitely extends the field, and we can talk more about that. I'm happy to engage you online or offline. I'm very excited about it. And so these may be powerful tools to help plant biology, but there are other tools. I want to make a plug for some of the more engineering type of applications that I'm also interested in. This was working. I won't go through the whole thing, but this is work where we took spinach and we implanted. Now I'll talk more about this. We planted nanosensors in the spinach leaves that you could detect remotely that could monitor explosives. So we have in this case we have a fluorescent nanoparticle, and what you're supposed to see here is that it will show a fluorescence quenching when it binds to an explosive molecule. So in this case, picric acid, as an example. So in this study, we showed you could put nanosensors inside the leaf. This is a reference sensor, which you're seeing imaged, and a nanosensor. You can ratio the two of them to detect explosives that are transpiring in the plant. And you can do this on a cell phone and with very simple instrumentation. So this is a quick video, which I will use as hopefully this video is playing. Can you see this video?
Yes, it's playing.
Okay, good. Yeah, so this is a plant seedling. The roots are wrapped in cheesecloth. You put a dilute solution of picric acid. As it transpires, it's actually going to activate a nanosensor that's ratioed to a reference, and you'll see that here. And you can use very simple instrumentation for this, like a Raspberry Pi, a cell phone camera. And you see this here. You get this quenching as explosive molecules are transpired and trigger the sensor. So you can, we demonstrated this as a way to go from chemicals in soil transduced by the plant, sending it to digital information, as sort of an Internet of Things. And this when we first did this, I worried that my colleagues at MIT and maybe in the field, this was an idea that was very, very new, and it was it wasn't quite within the window of acceptability. But about a year later, so you see this is 2016, 2017, DARPA came up with their APT program, which they funded, and there they wanted research ideas on a plant that could detect things in the environment, and then it could then send that response. It could itself respond, and then it could also send a sensor signal. And it was very interesting. Like it had these stimuli in the environment, which were chemical, say, and then it could send a response. And so I said, where'd you get that idea from? I mean, maybe you got it from our paper a year earlier. So we we put in a big grant into DARPA. I thought we were a shoo-in for this because we just did it. We just used unique tools to solve their problem. But alas, we did not get funded. So if anyone here is from DARPA or part of that program, you need to know I still have sour grapes. I still do. But actually, it was for the best because we started to turn our attention inward, thinking about plants. Like instead of sensing things outward, what if we turned our sensors inward and we started to detect the internal signaling in the living plant? And I hope I hope you'll find this this latter part of my talk interesting, in that nanoparticles don't only just have to deliver things to solve all of these problems. I think if you're a plant biologists, you may be interested in the work that we're doing now, turning sensors inward, measuring the internal signaling in living plants. So plants are exquisite sensors of their environment. They have a lot of information about their environment and about their local conditions. They have that information long before humans discover it. So they have internal signaling on light intensity or light stress, changes in temperature, thermal stress, mineral and water availability. They have that information locally. They respond quickly. Pathogens, they respond with exquisite precision, very, very quickly relative to humans, at least. Insect mechanical injury. So the idea here is that could you take nanosensors, which are very powerful, can detect very low concentrations, and I already showed you can connect to the internet. I mean, clearly you can connect them to the internet. You could. So can you take those signals and can you collect them for human use, for basic research, for agricultural monitoring? Can you and how can you do that? And you want to do this non-destructively. So plants have a variety of exciting signals. You can see that. Maybe I'll try to. Maybe let's try to. That's sort of a duplicate. So I'm going to use another technology that we developed in Nano in my group, and I can explain it in a simple way. It's called corona phase molecular recognition. You probably heard me refer to the adsorbed phase of polymers or surfactants around a nanoparticle. In the field, we call that a corona phase. So you're supposed to see like the rays of the sun. It's called a corona. And there's I think in biology in medicine there's also called a protein corona when proteins adsorb, and they'll also make a corona. It's the same idea. Corona phase molecular recognition is the design or the discovery of these corona phases that give you molecular recognition. So it's an empirical. We do a screen of a library of these corona phases. You can find instances where these analytes will bind and cause intensity changes or wavelength shifts. Now it's a very powerful technique. It's been around for about 7 years, and a carbon nanotube as a sensor is non-photo bleaching, and it can emit in windows often where chlorophyll does not absorb, and you can use that as an optical window. And and we've done a lot of work showing their biocompatibility. So they seem to, if engineered correctly, they seem to work well with the plant system. So one of our first sensors that's worked exceedingly well is actually this sensor for hydrogen peroxide. It's a DNA oligonucleotide wrapped carbon nanotube that actually is exquisitely sensitive to hydrogen peroxide, and you can actually reference this with another DNA oligonucleotide that does not respond to hydrogen peroxide very well at all. And so the two of those can form a ratiometric sensor. And let me sort of skip over this here, and you could, well, I'll say this: if you we first used this sensor to study wounding, and if you mechanically wound a plant, it's known that through NADPH oxidase, you'll generate hydrogen peroxide, which then goes to signal calcium wave, and which sets off a chain reaction that propagates through the tissue. And the instrumentation that we developed in order to measure this, I want to point out a couple things. One, it's very simple, but two is that it's standoff. So if you have an optics background, you're going to know that if you can get this detection to work over a meter, you can get it to work over 100 meters or 1,000 meters. It's basically a free space detection technique. And so it's amenable to things like drones or long distance monitoring. Let me show you. So the infusion and the setup is similar to the explosive sensing. We've got our hydrogen peroxide sensor here, compared to our reference. And then if you wound the plant, you get a very interesting wave. Let me see if I can. It'll start over again. So you see a quenching that then slowly restores. You can see it here in the red. There's a quenching that then slowly restores, and so you're imaging in real time. You're imaging this wounding wave here, and you make a characteristic wave form, which you can actually show is a sum of logistic functions. And so I've done a lot of mathematical thinking about this wave. It's a very interesting, this asymmetric wave form is what propagates all throughout the plant. It's fascinating. This is with 10 plants, and I think this is done also in spinach, but you see we did it in a variety of other species. So you can launch multiple of these waves. You can so here what you're seeing is with a sensor construct in the middle of the leaf. If we injure at 1, and then injure at 2, then injure at 3, you're seeing these waveforms, right? And you're seeing and again, of course, you can detect this with nothing more than a cell phone camera. So just like a $20 CCD camera can detect this. So it's amenable to very low cost instrumentation. We started again. It's the same with the nano carriers. It's a species agnostic tool to the best of our knowledge. You can immediately use this to study the wounding waveform in. We did a lot of agricultural targets like spinach, strawberry, lettuce. Arabidopsis is the model plant, but we've also done tobacco, sorrel. So this waveform, it's interesting. You get the same waveform, the same asymmetric functional form. So the plant is telling us a function, but the parameters are different. So the amplitude, which is the height of this this diminution, the lag is the velocity of the wave, which you can show is actually related to biochemical properties. And the full width at half max also you can show is related. But so you can now study this wave and you can study these properties inside the plant. We did as something particularly showy. You can do insect feeding. This is a caterpillar as an elicitor, and every time the caterpillar seems to cause tissue damage, you get these diminutions, and so you're able to actually detect them in real time. And I'll leave you with this though. This is interesting. We then studied. So we studied the wounding response very, very well. I believe I understand that wounding response, and I have a paper coming out, where I'm working on that, and where I mathematically model it. I understand why it has the size and the shape, and you get this asymmetric waveform, and you can mathematically describe it. You can link it to basic biochemistry. It seems to appear in all plants. Very exciting. But if you do heat stress, and there's a characteristic experiment where you heat stress the plant, you see that the waveform is very different. I mean, this may not be completely obvious to you. It might look like a curve, but this mathematically, this blue is not the same as the red. And my theory that describes this, it says that under heat stress, something else is happening. And it's a very curious thing. Light stress also has a very different waveform. It's not a simple scaling factor compared to the wounding. And then also, if you do this FLG22, this pathogen assay, you also get a different. You get the green, so it's pointier and it has a different lag. These are different waveforms, and this introduces the idea that these living plants have waveforms. They encode for the type of stress they're experiencing, and for the first time in real time, we can detect that stress and send it to a cell phone, and we can decode that stress. And this will be a very powerful tool. Remember, the plant can generate multiple of these waves. You can see them in real time, and we can do mutant plants, and we can really piece together these pathways in a way that's much faster than grinding up plant tissue. So Very, 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 very exciting. I don't know how much time I have left. It's 10:55 in Eastern Standard.
You're doing great on time. We go till 15 more minutes actually. We do want to save some time for Q&A though.
Okay, yeah, I'll do one more example. So this is something. This is the satellite laboratory that I have in Singapore. I work with my partners there, the Singapore government. I should have mentioned that this DISTAP, this disruptive and sustainable technology for agricultural precision, we put together to solve problems in urban farming and also global agriculture. One problem they brought to us is a problem of arsenic. So arsenic is present in aquifers that are associated with agriculture around the world. It's largely natural arsenic. It comes from geological sources, but the problem is a lot of staple crops like rice and vegetables and tea will bioaccumulate arsenic. And this is a real problem. So one of the things we're doing to solve that is giving the community an exquisite sensor that works inside of a plant that can tell you about arsenic uptake. And this could be a very valuable research tool for understanding the pathway of taking up arsenic. So I'll go right to it. We applied LEAP to and we found a different DNA construct. This is a GT5. It's very, very different. It has really great selectivity for arsenic-3. I mean, you can see it here. It doesn't bind to really anything else. It binds to arsenic-3. It has a pretty good KD, and it's very reversible. So if I chelate the arsenic, you can see that it restores, and you can use this to study. So this is again spinach, and you're adding arsenic just to the water, to the ground water, and you can see here if you've got a sensor here, you're able to see in real time. You're able to see the arsenic transpire up into the plant, and you're able to measure the kinetics of this. You can see this in real time. You can ratiometrically do this. You're seeing the accumulation of arsenic-3, and you can even do this in rice. We've said so. In rice is a particular target in this sense. So we're interested in a sensor that can accumulate arsenic, and we even did this with this beautiful work from Mal et al. In 2001 on these arsenic hyperaccumulating ferns. So this brake fern can accumulate huge amounts of arsenic, and so we're just curious. We had to try it. So it turns out if you put our sensor inside the brake fern, you get much faster uptake, and you get a huge amount of arsenic that can be accumulated here. And this brought us. We did a lot of mathematical modeling of arsenic in the frond and in the concentration around the sensor. We came up with this general mechanism. So arsenic in the groundwater that then binds to receptors on the root, transport up the frond, and they're detected here, but then they actually decay as they're segregated, and it turns out this mechanism does a very good job of describing the uptake. But if you use a hyperaccumulating fern, one of the things that you can do is you can turn this plant into the most exquisite sensor for arsenic that has ever been detected. We think in our, not just in our opinion, but the World Health Organization, has a drinking water regulation of about 10 parts per billion, which is about 77 nanomolar. And in irrigation water, they relax it a little bit to 100 ppb. But if you have a 12-week-old hyperaccumulating fern, after about say 7 days, you can detect 4.7 nanomolar of arsenic. So we actually envision a technology where you could use a hyperaccumulating fern, to actually test for arsenic. You can detect it at concentrations far below, what you can detect with handheld monitors and other things in the field. So really like this idea of plants, coming into their own in the technology space. So my student Tedrick Liu really is featured in a number of the recent works here. So, and he actually he made this slide, which turns out to be a very good summary for the talk that I did today. So I showed you nanoparticle carriers for transporting really anything into plant cells, into chloroplasts, into pollen, right? And then sensors that can detect a wide variety of things, from wounding and other stressors to arsenic and inorganic materials. And keep an eye on DISTAP, our satellite lab, where we have a whole arsenal of very exciting sensors for almost anything you would like to measure in the plant. We're particularly focused on plant hormones, and the work is going exceedingly well. We're very excited about it. So with that, I will thank my group again. The picture needs to be updated, but yeah, very excited to pause here and answer your questions. Thank you for following along.
All right, thank you, Michael, for a great talk. By the way, did not advance to the last slide for some reason. Weird that it was great until that.
There we go. Okay, yeah.
Yeah, thank you.
Sorry about that.
All right, great, and luckily we've got time for questions, some good amount of time. So I'll ask a couple of questions in the chat. I do want to say though, I love these cross-disciplinary talks. I remember when I met you in Cambridge, you came to talk about photosynthetic capacity and how you can enhance that with nanoparticles, and my ears perked up when you said that they were coated with DNA. And so in this regard, we're here talking mostly about delivery today, but I think we're all intrigued by a lot of the things that you've talked about and the ideas about sensors. I look forward to getting an alert on my Apple Watch when my plants need nitrogen. All right, so I'm going to kind of group some of the questions. So the first couple of questions came in about the chloroplast delivery work was, what about other organelles? Why was it just the chloroplast? Did you see it go into the mitochondria? How organelle specific was it?
Yeah, so I showed a lot of that data. I think it'll take time to flip through it. We started studying the chloroplast, so we're focused on the chloroplast. We have the question of mitochondria is interesting. We haven't looked at it and investigated it to the same extent. LEAP makes predictions, like if you know membrane properties of the mitochondria, and it's also there's also an envelope there and two lipid bilayers ultimately, depending on where the particle needs to go. So I'll say our theory makes predictions about the mitochondria, but we haven't looked. We haven't investigated that. And also the other thing that I want to stress too, because the questioner asked about selectivity, is that our mechanism is a passive one. It's basically just using charge. It's going to be as selective as the differential in membrane properties. Our theory can tell you how selective you are. However, that doesn't exclude the possibility of using other anchoring recognition sites to in combination with LEAP to get more selectivity. I think you want to think about from a design standpoint. LEAP is a very good background mechanism that can get you through certain barriers. It's necessary in some cases, but not sufficient, depending on the charge. And there are lots of other techniques that have been successful in the literature, like these homing peptides and so forth. And I think there is a clear opportunity to combine the two of them. Yeah. So hopefully that's an answer. That's satisfactory.
Yeah, thank you very much. Somebody asked, what range of concentrations for the particles could be used to deliver something? Is there a concentration where the membrane becomes unstable?
We have not seen that. So with extracted chloroplast, yes. Nanoparticles themselves, though, have natural barriers to how high a concentration they can go to. So the nanoparticle solution itself can become colloidally unstable. So as you make nanoparticle concentration higher and higher, the first question to ask is, is your nanoparticle solution becoming unstable? Meaning, is it unstable to aggregation? All nanoparticle solutions have a concentration upper limit. The nanoparticle solutions that we're working with here are very dilute. You don't need a lot of them. They're essentially in the parts per million range. But LEAP absolutely does predict that if you saturate overly extrapolate. But it just seems to be the case that these tools and these mechanisms appear at first glance to be species agnostic. That does not mean that we'll find that we won't find conditions or species where there's something else that's going on. It looks like some of these, to the extent that it's basically structural elements in the plant cell and the plastid that are mainly governing the transport. Those appear to be evolutionarily conserved. So and I'm not the expert there, but it could be that the structure of the chloroplast isn't there's not a huge phenotypic diversity there that would cause LEAP to work in one case but not the other. But we should investigate that empirically. Yeah.
All right, thank you. Let's see if we can get a couple more questions in here. Susana Diaz asks, is there any data analyzing the effect of pH changes and ionic strength on the perceived preference of differently charged nanoparticles to penetrate certain organelles?
Well, so pH will definitely have an influence. It can protonate or deprotonate certain sites on the particle, and of course in receptors in biology. And I mean it's and what I'm describing in LEAP is primarily a charged interaction. So for example, one of the carriers I showed data for was chitosan. Chitosan is an aminated polymer, so pH clearly will change the charge. If you do a zeta potential measurement and you change the pH, you will see that the charge can be positive or go near neutral. So yes, there is a simplistic built-in ability for LEAP to predict that. Whether that's been systematically studied, I'd be happy to work with you and the community to study that. I think measuring compartmental pH, manipulating compartmental pH, those are things that you could do to try to control that and to get better selectivity. So I think you're on the right lines. You have the right type of thinking.
All right, so we're not going to get to all the questions. Dr. Strano did offer to answer by email. He's easy to find by email if you have any questions. I am going to ask one more question because I got 2 thumbs up, so several people are interested. I'm interested. Have you or your collaborators tried to pollinate with the pollen grains that were transfected? What happened if you did?
Okay, yeah. So I'm happy to follow up on that with you. I don't think I can talk about that now. I will say that we're not doing those experiments at MIT, but I am very interested in it, and it does get to where, like, to work with certain agricultural targets and to do certain experiments. You know, MIT is a small school, and we don't have an enormous, you know, agricultural infrastructure. I'm a tool developer, and I want to work with you. I want to. I know that I'm looking out into an audience of scientists that have access to laboratories that would allow us to do things we cannot do at MIT. And I have a scientific curiosity here. I'm very, very interested in exceptions to LEAP. I think we need to find situations where LEAP is not working. Species, plants, conditions, organelles, and the pollen, I think, opens up. I think Larry would agree. You would agree. The pollen mechanism opens up a lot of opportunities, and I very much want to. You could probably spend an entire career just exploring that, right? So I'm very interested in it. So hopefully that helps.
So we got to most of the questions. If I didn't get to your question, please follow up with Dr. Strano. And I wish we could all be in one place and applaud. So we'll just do a virtual applause. And thank you, Dr. Strano, for a great talk. So we're going to move on now to try to stay on time. And I'm going to turn it over to my colleague, Yurong, who is going to introduce the next speaker.
Thanks, Larry. Thanks, Mike, for the great presentation. Good morning, good afternoon, good evening. My name is Yurong Chen. I'm a cell biologist leading transformation optimization team here at Bayer Crop Science. It is my pleasure to introduce our next speaker, Dr. Langying Li. Dr. Li is a senior research scientist at Purdue University, and Ling Yang received her PhD from National Yanming University in Taiwan. In 1992, Dr. Li joined the laboratory of Dr. Claire Lawrence Caddo at UC Davis as a postdoc research fellow and started her involvement in agrobacterium-related research. In 1996, after joining the laboratory of Dr. Stan Galvin at Purdue University, Dr. Li became involved in agrobacterium research on the plant side. As a card-carrying microbiologist, she applied her specialty in bacterial genetics on modifying agrobacterium to study plant and microbe interactions to better understand the mechanism of agrobacterium-mediated transformation. Her current research focuses on developing mutant agrobacterium strains for conducting integration-free TDN transfer and on improving an alternative non-agrobacterium-mediated plant transformation systems. Dr. Li enjoys teaching and training young people as well as traveling. The title of Dr. Li's presentation today is Agrobacterium and Non-Agrobacterium Delivery Systems. Take it away, Dr. Li.
Good morning, everyone. So today I'm going to talk about agrobacterium and the non-agrobacterium gene delivery systems. So first, I want to thank Yurong to invite me for this symposium. It's my honor to be able to speak along with so many specialists in different areas, especially after we listen to Michael's talk. And then it's amazing how currently the nanoparticles can be applied not only in other fields, but also in the plant science. So with the improvement of genome editing and the sequencing tools, progress in plant synthetic biology and bioengineering has advanced. In agriculture, genetic engineering can create crops with increased yield, altered nutritional values, or resistant to herbicides, insects, diseases, and stresses, such as drought or heat. In pharmaceutical and therapeutics, genetic engineering of plants can support the synthesis of valuable small molecular drugs, recombinant proteins, and so on. So in the plant cells, there are 3 transformable genomes. They are nuclear, chloroplast, and mitochondria. So currently, gene delivery can be achieved by either agrobacterium-mediated transformation. This could be Agrobacterium tumefaciens or Agrobacterium rhizobium, or non-agrobacterium-mediated transformation. This could be bacteria-mediated, viruses-mediated, or other non-microorganism mediated. For bacteria-mediated, non-agrobacterium-mediated transformation, so far a lot of use of rhizobium species. And there's also Ensifer and the Acrobactron, which is used in the company Corteva now. And in the past, there are reports talking about using viruses to deliver the gene into the plant cells, but not so much nowadays. As for other methodologies, I'm going to just briefly describe and introduce either biolistic particle delivery, protoplast transfection, and electroporation, and nanoparticle mediated transformation. Of course, after the previous talk, I think I can limit this part. I just give a brief summary on this part. So biolistic particle delivery, also called gene gun or particle bombardment, basically is coating either gold or tungsten or silver particles with biomolecules such as DNA, RNA, or protein. And then with the force of high pressure and shoot all these particles into the plant tissues. These methods, as we all know, is quite easy to use. It's rapid and versatile. Transient and stable expression is possible. Cold delivery of the multiple plasmid and high levels of the cold transformation are possible. No carrier DNA is needed, and no extra genes or protein are delivered. This method can be used for many plant species and cell types, including those non-dividing cells and the plastic genomes. There are also disadvantages of this method because it requires specialized facilities and expensive materials. It also requires use of a substantial amount of DNA, enables low label and sporadic expression. Integration incidence is low if it occurs as random, and often causes plant tissue damage due to higher bombardment pressure and exposure to vacuum. So not many people are using this technique so far. We also can deliver a gene into the protoplast. Protoplast is the plant cells after the cell wall removed. We can use either polyethylene glycol mediated transfection, or we can use electroporation to deliver the gene. So there's a good thing about this method. Delivery of multiple plasmid with high levels of cold transformation can be achieved. No need to make a binary vector. High frequency transformation efficiency. Most of the plant species are amenable to protoplast isolation and transformation. There are also disadvantages of this method. It's limited to transient expression and also limited plant species that are amenable to regeneration from the protoplast. And among those that can be regenerated, the technique is time-consuming, labor-intensive. So as for nanoparticle-mediated genetic engineering for the plants, I don't have to describe too much, but basically nanomaterials is defined as materials with at least one dimension below about 100 nanometer and have a unique and tunable physical and chemical properties. And here just to show a figure of the plant cells and show the relative size of different organelle and different micro and nanoparticles. And then because the size exclusion limit of plant cells is a set search, so all these different type of nanoparticles has to be assisted from the mechanical aids, such as ballistic, ultrasound, vortex, or electroporation. This type of force-independent internalization. Of course, like Michael just described, many of their nanoparticle experiment was use nature diffusion and into the microorganism, into the organelles after extracted. So it's different applications. So in the literature, there are several examples of nanomaterials used in biomolecular delivery in the plants, such as mesoporous silica nanoparticles has been able to deliver plasmid DNA during the co-cultivation with the Arabidopsis root, and then detected the fluorescence expression. And then there are use clay nanosheets, also deliver RNAi molecules through topical application to tobacco plants. There are also DNA origami nanostructures can deliver the small interfering RNA into tobacco Nicotiana pentamina, and then detect the protein and RNA synthesis after the delivery. So as carbon nanotubes probably is the most potential nanoparticle can be used nowadays. And then like Michael already mentioned, their group and the other group, they have been successfully delivered either plasmid DNA or siRNA into variously model or non-model plant species. So the potential of these carbon nanotubes is because if you change the physical and the surface chemical properties, the charges or the size or the ratio you put in for the carrier and between the carrier and the biomolecular, you can govern these nanoparticles into different subcellular destination, different organelle compartment. And also optimization of this particle is possible for different tissue types, such as callus or meristem. And this delivery usually yield transient expression, and there's no tDNA integration. So it is possible to make a transgene-free plants. And the potential for gene delivery to the plastic. So these nanoparticles has a very great potential, just like Michael said. If we can combine the biology and this chemical engineering, and then we can open a really bright future for gene delivery into the plants. So nowadays, we all know agrobacterium-mediated transformation is still the most popular technology to introduce genes into the plants. However, other bacteria can also transform plants with tDNA and the virulence genes. So these bacteria species of a genus are listed here. So many of them are rhizobians, and then cynorhizobian, and then there's Ochrobactron and the Ensifer. So in the review written by Rothel and Mullen in 2018, they summarize several groups of non-agrobacterium method, which is successful result in transgenic plants. They are listed here. I highlight in blue. So for example, Transbacter is a combination of 3 different bacteria species. And then this system need to provide binary vector. For rhizobium-mediated transformation, Ensifer-mediated transformation, rhizobium-atala-mediated transformation, or Ochrobactron-mediated transformation, we all need to provide both tDNA and the virulence vector. So they can be used in either binary vector or unitary vector systems. All except Rhizobium atallite, this bacteria actually has its own virulence plasmid. So we just need to provide tDNA binary vector during the application. So all these system has a different host range tested, some with a narrow host range and some with a higher host range. So about the advantage and the disadvantage, many of these bacterial systems, the advantage is they use the free living bacteria or plant soil bacteria. And many of the bacteria, such as Rhizobium and Ensifer, they are non-plant pathogen, which is important as for the regulatory issue. And then some of them has been used in many of plant species. Some of them only used in very limited host range. Okay, and then some of them has a higher transformation efficiency compared to agrobacterium, and some of them still have a lower transformation efficiency. And except Ensifer, I think none of them has many literature we can follow, especially for this Ochrobactron. The only things we can follow is their patterns. So now I want to switch my gear into the research we are doing in our laboratory. There are 2 parts I'm going to talk about. The first one is improving agrobacterium-mediated delivery of genome editing reagents. There's potential for improving tDNA delivery and regulating tDNA integration. And the second part is a reduced host defense response supports Ensifer-mediated transformation as an alternative to agrobacterium-mediated transformation. So first, how do we manipulate agrobacterium tDNA integration? This work is conducted in Dr. Stanton Gilban's lab at the Purdue University. So agrobacterium tDNA integration usually into the plant genome is random. So this is useful for generating knockout libraries. However, random integration can result in host gene disruption and transgene silencing. tDNA often integrates at the multiple loci with multiple tDNA molecules per locus. This can result in transgene silencing and can present the problem with tDNA segregation in the subsequent generations. Integrated tDNA can be linked to vector backbone or even bacterial chromosomal DNA. So this can result in regulatory problem. So there are circumstances where it may be desirable to avoid tDNA integration. For example, when introducing a correction template for homologous dependent repair, HDR, it's often preferable to avoid the ectopic integration of tDNA containing these templates because random tDNA integration events will need to be segregated out of plants later on. Second, when introducing genome editing reagent, in order to generate non-transgenic edited organisms, tDNA containing genome editing reagents need to be segregated from the plant genome. This is time-consuming, costly, and difficult for plants with long generation times, such as trees or species that are vegetatively propagated. So if we can manipulate the system to suppress random integration and enhance target integration, then it's possible to facilitate high efficiency routine crop genomic engineering. So our goal is to develop a new technology, non-integrating tDNA transfer, to facilitate the use of the CRISPR-Cas and other genome editing reagents. So we are using several approaches in our lab to achieve this non-integrating tDNA transfer. And today I'm going to talk about to identify VOD2 mutant proteins that return tDNA transfer, but not the tDNA integration ability. So why use VOD2? So Agrobacterium virulence protein D2 is essential for tDNA transfer because it's known that the VOD2 will nick the tDNA border regions at the tDNA and then subsequently covalently attach on the single-stranded T strand, and then we'll export this T strand through type 4 secretion system into the plants. We also know that VOD2 is also important for integration. The fact is, Dr. Warren made a VOD2 omega mutants long time ago. He substituted the omega region of the VOD2 protein with several serum residues. And then when we test this VOD2 mutant in either transient or stable transformation, and we found that it's only about 20% of the transient transformation and about 1 to 2% of the stable transformation is caused by this VOD2 omega mutant when compared with the wild type VOD2 proteins. Therefore, we know that the VOD2 is also important for tDNA integration. So in order to uncouple this tDNA transfer and tDNA integration functions, we try to generate Agrobacterium strain that introduce tDNA into the nucleus, but do not efficiently integrate into the plant genome. So such mutants will be useful for delivering but not integrating genome editing at reagents and the HDR repair templates. So in order to do the experiment, first, we make a VLD2 deletion on the TI plasmid, and this deletion is non-polar to the downstream VLD3 and VLD4 expression. We also subchrome one region from the TI plasmid, put it on the replicating plasmid in agrobacterium. This region containing the VLD promoter, VLD1, and VLD2 genes. We modify this VLD2 fragment and make it easy to switch this VLD2 fragment. We use PCR mutagenesis to mutate VLD2 genes, and then we replace the wild type of VLD2 with all the mutated VLD2 gene back to these replicating vectors. So we then sequencing individual plasmids and then confirm the mutation of the VLD2 genes. So this figure just showing that both nucleotide sequences and amino acid sequences of these agrobacterial VLD2 proteins. So I use the red color to indicate altered nucleotide and the amino acid sequences. So normally, if you have a nucleotide change, you will change the amino acid residue in different ways. Sometimes you can get the early translation stop codons by just changing the nucleotide sequences. And of course, you can get some of the nonsense mutation, which we consider that will make the wild type of VLD2 protein. So you can see for this example I have shown here, the mutation is pretty random. So after we verify all these individual VLD2 mutation, I have to mention that most of the VLD mutation is with either one, most of them is with one single nucleotide and single amino acid residue change, and only very few of them has multiple changes in the amino acid sequences. So we then put this plasmid back to in the agrobacterium. This is a tumorigenic agrobacterium strain containing a VLD2 deletion on the TI plasmid. So we are hoping this incoming VLD2 mutant can or cannot complement the VLD2 function here. And the bacteria also contains a gas intron expression cassette on a binary vectors. So because this is an oncogenic strain, so we can first analyze all the bacterial colonies for the tumor formation ability on the kalanchoe plants, because we know the tumor formation is an indicator of tDNA successfully integrated into the plant genome. So we then discard or put aside those bacterial mutants which still give a tumor, and choose those didn't give tumor, and for the next assay. The next is we use transient gas expression as our indicator for the tDNA delivery into the plant and expressed as a transgene. So we use tobacco plants for agro-infiltration. So usually 24 hours after infiltration, we can determine whether this particular bacterial mutant can induce a transient expression of the Gus gene or not. So we will pick those are able to induce the transient expression, and then we analyze these colonies in our Arabidopsis root assay to reconfirm both transient and stable transformation efficiency. And then our final goal is to identify some bacterial mutant which has no stable but still with a good transient transformation efficiency. So this slide is to show you the Kalanchoe tumorigenesis assay. So the Kalanchoe leaves can allow the multiple inoculum on the one same leaves. So here I have a positive control, wild-type VOD2 induced the tumor about 10 days after the inoculation. There's a negative control, and there are various mutants I apply to infect the leaves. And for these mutants still maintain their tumor formation ability, we put aside, and we continue to analyze these mutants which already lost the tumor formation ability. So this is just to show you the tobacco leaf infiltration result can be used to identify some of the VOD2 mutants still supporting transient transformation while losing the ability to form tumors. So we then pick these transient positive bacterial strands and then to do further confirmation in Arabidopsis. I'm sorry, I saw this signal said that network quality is still okay.
Yeah,
Hello?
we still can hear, but a little noisy background, but still okay.
Okay, okay, I can continue. Okay, so this figure just to show we use Arabidopsis root assay to retest some of the Agrobacterium mutant strains and then to see their transient or stable the transformation efficiency compared to the wild type plants. So our goal is to pick those still have transient transformation efficiency. Of course, we didn't find them as good as the wild type, but we still see some has a pretty good transient ability, and then with a quite low the stable transformation ability. So we will further analyze these bacterial strains. So one of the systems we use to analyze the function of these VOD2 mutants is using the CRISPR-Cas9 editing system. So we make a transgenic Arabidopsis plant which is containing out-of-frame GFP genes. So within this GFP genes, we inserted one extra nucleotide to make the GFP out of frame. So there's no GFP expression in this plant. Then we use Agrobacterium strains carry various VOD2 mutants, and also the CRISPR editing reagents and the sgRNA targeting to these regions, and try to correct this GFP reading frame. So if the editing is successful, GFP can return to correct the reading frame, and then we can detect the fluorescence. We then analyze these plants to detect whether there's any tDNA integration events. So to show this method is actually working, we also use, we first use Arabidopsis root, which from this transgenic plant carry out-of-frame GFP to do the agro infections. So the tDNA we have The tDNA we have, in addition to this Cas9 SG DNA to correct the GFP, and also a plant to also have a plant active red fluorescence protein genes allow us to detect a transient transformation. So these are the callus about 1 month after the infection. And then you can see some of the callus indeed show GFP fluorescence signal. That means the GFP reading frame is corrected correctly. And then you can also see the RFP signaled still maintained in some of the callus, but not some of them. That means there's no tDNA integrated in this callus in this event, but with positive CRISPR editing successfully. So to conclude this part of the talk, we used mutagenic PCR to alter the sequence of VOD2. We also obtained mutants that could not stably transform plants. And some of these mutants continue to support low efficient transient transformation, suggesting that some DNA had entered the plant cells but had not integrated them. Some VOD2 mutants are able to deliver CRISPR reagents to Arabidopsis and support genome editing. These VOD2 mutants are now being tested in various plant species. So this concludes my first part of the talk. So next part, I'm going to talk about ANCIPR. This work was initiated by the former graduate students, Rochelle Leppen, in Stan Gilvan's lab at Purdue University, and current graduate students Ashok Govidan in Dr. Ewan Mullen's lab in Chegas, Ireland. So there are advantages of the ANCIPR-mediated transformation over agrobacterium-mediated transformation. Because using ANCIPR, we can avoid the complex pattern landscape surrounding agrobacterium-mediated transformation. And ANCIPR is not a plant pathogen. Therefore, the bacteria or the resulting transgenic plants will not be regulated in the same way as those generated by agrobacterium. And because ANCIPR is not a pathogen, so plants do not respond to it with the same necrotic defense reactions that often accompany inoculation with agrobacterium. And ANCIPR has been used to transform potato, rice, Arabidopsis, and the tobacco so far. So first, we ask several questions. First, with regard to the Arabidopsis transcriptome, do agrobacterium and ANCIPR elicit the same response when inoculated onto the roots? In particular, do both bacterial species induce a defense response and signaling genes? Second, does the Arabidopsis use the same plant genes to mediate the transformation by agrobacterium and ANCIPR? In particular, do plant defense mutants respond similarly to the two bacterial species? And third, can ANCIPR-mediated transformation increase the transformation of a crop variety that may be more recalcitrant to agrobacterium? So to begin with the work, Rochelle and Ashok, they conducted the Arabidopsis, their transcriptome analysis on Arabidopsis root when infected either by agrobacterium and ANCIPR. So this figure just summarizes those differentially expressed gene during the either agrobacterium or encephalopathy infection. And then this figure obviously to tell that the Arabidopsis root transcriptionally responds more to agrobacterium than to encephal infections. So if we categorize or group these regulated genes into different gene groups, and then you can note that Arabidopsis here shows in blue differentially regulate more of the defense and the stress response and the signaling genes than does encephal, which is shown in the red. So we first look at the agrobacterium and the encephal infection either transiently on various Arabidopsis ecotypes, because our lab, we do most of the work in the model plants. So we look at the different Arabidopsis ecotype, and then the result showing that whenever you use agrobacterium, we have a different transformation efficiency on certain ecotype. We will obtain the similar relative efficiency when the plant was infected by encephal. So that means when ecotype, for example, CO0, WS, ANG0, they're all very successful to agrobacterium infection. And then the same thing, they all successful to encephal infection. And then there are some ecotype which is recalcitrant to agrobacterium, and also they are recalcitrant to encephal infection. We also look at the unknown genes important for agrobacterium-mediated transformation. Here we use the example of 3 genes, red 1, which encodes arabinoglycan protein 17, which is important for agrobacterium binding. The red 5 protein encodes histone H2A1, important for protecting T strand and for tDNA integration. Importin alpha 4 is an importin alpha protein important for virulence effector protein nuclear targeting. And since these genes are important for agrobacterium-mediated transformation, therefore, when we use the mutant plants, has a deficiency in these genes, and we often get a resistant transformation efficient pattern. So these mutants respond the same way to encephal infection. So these essential genes are also important for encephal-mediated transformation. So we also know that for Arabidopsis, agrobacterium use EFTU, the elongation factor, as a PAMP signal for Arabidopsis. So if we look at the mutant, which is lacking the receptor for this PAMP signal, and then it will be very, very highly susceptible to agrobacterium infection. So we use the same mutant to check whether encephal has the same response. And the result here shows that there's no differences. These mutants response compared to the wild type plant. Therefore, we conclude that for encephal, it's an EFTU protein is not the PAMP for Arabidopsis. So we also check the FALS2 mutant, which is lacking the receptor gene for flagellin, which is a PAMP for many other plant pathogens, but not for agrobacterium. So it's not a PAMP for encephal either. So because Arabidopsis does not respond to ensifer by recognizing flagellin or EFTU as a pen, so does it respond to ensifer in other ways? We then examined different signaling proteins. We located different mutants such as BAC1, BIG1, or RBOHD, and to see their response to Agrobacterium and the ensifer infection. So these results indicate that the 4 mutants we look at, 3 of them, which is BAC1-5 and the BIG1 and the RBOHD mutants, they all become highly susceptible to both agro and ensifer infection. So that means these are These are the signaling proteins that Arabidopsis responds to both agro and ensifer. So we also check those genes differentially expressed after infection by ensifer or Agrobacterium. So here we pick 4 different genes, and we we use the mutant of these genes, and then do the infection assays. And then here you can see that for some of the genes, both ensifer and Agrobacterium and ensifer, they showed lower response, means these plants are more resistant to Agrobacterium. So this gene may be important for infection. And then for other genes, sometimes Agrobacterium and ensifer, they have opposite response. So AgConnect also tested some of the plant defense response in potato stems. So he inoculated bacteria, either Agrobacterium or ensifer or flag 22 with the potato stems. And then he can show that after 120 hours after the infection, and the Agrobacterium elicit a greater defense response on potato stems than does ensifer or the flag 22 controls. So AgConnect also tested different potato varieties, and using both ensifer and Agrobacterium mediated transformation. And the transient transformation results shows here. So he used either Agrobacterium 3 days, ensifer 3 days, or Agrobacterium 5 days, ensifer 5 days of co-cultivation. And all the results showed that I just give you a conclusion. For most of the potato varieties, ensifer-mediated transformation is at least as good as is Agrobacterium-mediated transformation. And for some of the variety, ensifer has a higher transformation efficiency compared to the Agrobacterium. So, but I want to point out the important thing is for some of the variety, the tissue will show severe or some mild necrotic reaction to Agrobacterium, but it shows nothing to the ensifer. So based on what we have so far, I think we can give answer to our previous questions. With regard to Arabidopsis transcriptome, do our Agrobacterium and the ensifer elicit the same response when inoculated onto roots? Ensifer elicit much less of a response, especially for defense response and the signaling genes. The second question, does Agrobacterium use the same plant genes to mediate the transformation by Agrobacterium and ensifer, sorry, this Arabidopsis. In general, yes, but Arabidopsis does not respond to ensifer EFTU as a PEM. Some other plant genes also respond differently to Agrobacterium and ensifer. Can ensifer-mediated transformation increase the transformation of crop variety that may be more calcitrant to Agrobacterium? Yes, as I just shown in the previous slides, for various potato variety, ensifer actually works better. So it looks like ensifer is a good candidate to substitute the Agrobacterium. However, there's a current limitations of ensifer as a transformation platform, because ensifer is resistant to many commonly used antibiotics. For example, kanamycin, neomycin, spectromycin, and the rifampicin. So to be able to work with this bug, we need to eliminate some of this resistance to manipulate the bacteria. And second, ensifer is not as virulent as is Agrobacterium. So we need to increase the virulence and our manipulate infection conditions. So our strategy is try different disarmed TIR plasmid with different set of virulence gene. Or we can add a PCS gene, or try using constitutive VOG genes, or try manipulate plant genome genes. So with that, I have introduced our work regarding Agrobacterium and ensifer-mediated transformation. So your comments and questions are mostly welcome.
Thanks, Lin-Yan, for the great presentation and great summary. And we do have a few questions on the chat already. Actually, the first question is from Larry. There is a low-level integration within VERD mutant. Do you analyze any of these inserts to see if the junction between tDNA and the chromosome are similar or different compared to wild type?
Can you repeat the question? Sorry, before that, how can I get back to the meeting part? How do I stop sharing?
You just unshare?
I couldn't see the screen here. Okay, yeah, okay, that's good.
So Larry asking, do you analyze those inserts to see if the junction between tDNA and the chromosome are similar or different compared to wild type compared to VERD mutant?
Oh, okay. So far we haven't done that step yet. Okay. So Larry, you asked about the current VERD2 mutant. Is that correct?
Yeah.
Okay. Yeah. So far we haven't done that part yet. So we are still testing our mutant in different plant system. And then we are looking for some more of the people to work with us if we can work in different crops. And basically, we can only work with Arabidopsis in the lab. So we haven't analyzed those integration events yet.
Okay. The next question is, how long will the trans expression be active in VERD2 mutant?
Which reaction? How long will the
trans expression, is a mutant trans expression be shorter or would it be the same compared to wild type?
What reaction? Can you repeat again?
No, sorry. People ask the transient expression of VERD2 mutant is compared to the wild type, would it be longer or shorter, or how long this is going to be?
I would say it's about the same. because during our observation, okay, if we consider transient, it's about up to 10 days, and we can see similar reflorescence signals, okay, which is our indicator, because we cannot see the GFP correction right away. So they are about the same. Either we use wild type bacteria or the VOD2 mutants. So the time for transient expression is about the same.
Okay. Well, another question is, how high was the rate of editing in Arabidopsis in your VOD2 mutant?
Good question, sir. How high? I think the editing efficiency is high. However, our system depends on correct reading frame correction. Okay, so we can only hope we have one deletion or two insertion, and then that will make the GFP reading frame correct again. So we don't see many of incorrect editing. So we are currently trying to use a different system, and then we can collect all the edited genes for sequencing, and then we can show people how capable the editing works. Okay, so based on our the system we design now is not really suitable to answer these questions. Because if you have a big deletion, small deletion, or any insertion bigger or small than 2 base pair, then you don't see the GFP reading.
Okay. So another question may be similar. The question is, how did the rate of editing in Arabidopsis varies between VOD2 mutant and wild type?
Wild type, so far, what I saw, depends on the mutants. Some mutants give more editing events than the others. However, wild type, I didn't see really strong editing event. Didn't see much more editing event. Okay, so, but this is all work in the Arabidopsis using our system now. So, for example, we are giving our strain to our colleagues in Japan, and then they try to work in the rice transformation system, and then we try to set up the tomato system to look at the same experiment.
Okay. So I have one question, Liang Ying. So for the VOD2 deletion, the trend expression is about 20% of normal, right? But how can you improve the trend expression in this VOD2 mutant?
So our idea is if we have a different mutant, actually this VOD2 is not the deletion mutant. Okay, it's exactly the same size as wild type VOD2. Okay, in terms of the gene size. And then this VOD2 mutant, we have a different group of VOD2 mutant. Some of them give a really strong transient, and some of them give lower. So we are thinking about to swap the domains or swap a different region of this VOD2. So if you can get the N-terminal from this D2 mutant, and then C-terminal from this D2 mutant, and maybe there's a chance to improve the transient transformation efficiency higher than what we can get so far. But this is just idea thinking and need further work to show whether it works or not.
That's great. Yeah, we have one more question here coming. What is the regulatory status of ENCIFER media transformation?
Because ENCIFER, I have no idea. I think this question probably need to ask Dr. Mullins. He is in Ireland and he's working for the government facility. So he's part of the regulation for this ANSIFER. And I know that they can produce the potato, okay, manipulated by this bacteria. And then my only knowledge is since it's not the plant pathogen, so probably much easier to regulate. And especially if we are going to do the transgene-free transgenic plant or transgene-free editing, so probably it would be easier, but it's only my personal opinion.
Great, we are on time. Thanks again, Lang Ying, for great presentation.
Thank you.
I think we are now going to break. Annie, I'm going to make some announcement. I will just show the slides.
Annie, you're on mute.
Thank you very much, Edward. Just want to thank again Dr. Lee and Dr. Strano for kicking us off this morning. We've had some excellent talks, and yes, we are going to break right now. I do want to address a number of questions that have come in at the chat around the event recording. And so I'll put this up on the slide right now, but the event recording will be made available to all registered participants after the event. And so please stay tuned through LinkedIn for details on how to access that recording. But don't go anywhere. We have a series of talks coming up next that are quite exciting. So we will resume back here at 11:20 a.m. Central Standard Time with Mark Johnson's presentation. So thank you. And let me make sure this is visible. And I'll see you back here at 11:20 a.m. Central Standard Time. Thanks. All right. Hello, everyone. Welcome back to the Cellfies Symposium. I am going to turn it over to my colleague, Dr. Huachen LaRue, to introduce our next speaker. Huachen?
Yeah, thank you, Annie. Welcome, everyone. So my name is Huachen LaRue, a scientist in Bayer Crop Science. Currently, I'm leading reproductive system team in global breeding. It is my great pleasure to introduce Dr. Mark Johnson as our speaker. Mark joined Brown University's faculty in the Department of Molecular Biology, Cell Biology, and Biochemistry in September of 2004 and promoted to associate professor in 2010. Mark completed his graduate work in microbiology, cell and molecular biology at the Plant Research Lab at Michigan State University, where he studied mRNA degradation with Dr. Pamela Green and began working on plant reproductive development while he was a postdoc fellow at the University of Chicago with Dr. Dathan Pruse. Mark's research focus has been on molecular mechanisms responsible for pollen tube ovule interactions and for plasma membrane fusion of sperm and female gametes that lead to successful fertilization in flowering plants. Mark and his group have recently begun using tomato as a model system to study how temperature stress affects pollen tube pistil interactions, and are particularly interested in identifying gene variants that allow fertilization to succeed at high temperatures. The title of Mark's presentation today is Advances in Pollen Biology, Opportunities for Breeding and Crop Improvement. Welcome, Mark.
Thank you. Okay, can everybody see my presentation?
Yes.
Great. Thank you very much, Huachen. It's a real pleasure to be here in the symposium today. And it's a great pleasure to offer everyone a pollen selfie. It's been really interesting this morning to listen to how everybody is thinking about new applications and new ways of thinking to apply what we've learned to agriculture. And I'm definitely very interested in hearing from you about ways that we can apply what we're learning about basic pollen biology to agriculture. So my plan for today is to give you an overview of pollen development and function. And then I'm going to talk about a few project areas that are ongoing in my lab. One is on signaling that's required for sperm release from the pollen tube into the female gametophyte. And then a project on the mechanisms responsible for fusion between the plasma membrane of the sperm and that of the two female gametes. And then at the end, I'll touch briefly on a relatively new project aimed at understanding how pollen performs under heat stress. So I hope to be able to highlight cell-cell interactions required for fertilization. I think this, in many ways, will be a theme that resonates with the first two talks today. And then along the way, I also want to make sure that I highlight resources that have been developed, not only in my lab, but in the pollen research community that allow us to study pollen gene expression and function. Okay, so we'll start with this picture of an Arabidopsis flower. So we'll go right into the important parts for plant reproduction. So here's the anther, where all of these yellow dots are being made. These yellow dots are pollen grains. And in Arabidopsis, which is self-fertile, the anther conveniently deposits the pollen grains right on the stigma as the flower is opening, providing contact between the male gametophyte and the receptive stigmatic surface of the flower. Okay. So flowering plants have a unique system that brings male and female gametes together. The male gametes are locked inside of these pollen grains, which are encased in a really rigid cell wall. And the female gametes develop in ovules, which are, in this cartoon version of the flower, sort of buried within floral tissue. So I think I need to get my pointer back. there we go, to show you this movie. So I'm going to show you a cartoon version of pollen tube growth and guidance within the pistil. So the pollen gland starts off dry, it lands on the stigma and then hydrates, and it forms this tube that grows down through pistil tissue and then responds to guidance cues emanating from the ovule. It will target one ovule, it will grow inside the ovule, it will burst and deliver 2 sperm cells for fertilization. So reflecting on what we've heard so far this morning, obviously the pollen tube provides a great opportunity to deliver DNA to the germline, because that's precisely what it evolved to do. And thinking about this, in contrast to agrobacterium, the pollen tube, if you will, is the plant's pilus that delivers DNA from the male germline to the female germline for fertilization. Okay, so we can think of the pollen tube as a DNA delivery machine. So I just want to focus your attention on this cartoon just so everybody's familiar with the cell type. So in this case, the pollen tube is in red. The pollen tube nucleus is this gray oval here. And directly behind the pollen tube nucleus are a pair of sperm cells shown here. So what you're going to see as this cartoon continues is the pollen tube growing toward one of 2 synergid cells that are here in pink. The pollen tube is going to grow inside one of the synergid cells, and then the pollen tube and that synergid cell will simultaneously lyse, and the sperm will get released to a position where one can fuse with the egg and the other with the central cell. Okay. So that results in successful double fertilization. So there are a lot of really interesting cell-cell interactions that happen along the way here. The pollen grain has to germinate and make this tube. The tube has to burrow its way through the cell wall of the stigmatic papillar cells and gain access to the transmitting tract. The pollen tube exits the transmitting tract, and we now understand in pretty good molecular detail that the pollen tube is guided to the micropyle by small proteins called lures that are secreted by the synergid cells. And the pollen tube expresses receptors for the lure proteins, which then redirect the pollen tube tip growth toward a high concentration of lure proteins at the synergid cells. Okay, and the focus of our work has been on the next steps, so sperm release, and then the step that comes after that, gamete fusion. All right, and just so you, I know that many of you are interested in other plants besides Arabidopsis, and just so you can sort of port over some of these drawings to other plants that are certainly more important agriculturally, I'm showing here a tomato pistil, which has a relatively long style and a long transmitting tract, and ovules that are arranged in locules within the tomato ovary that will develop into the tomato fruit. So the pollen tube has quite a long distance to go in the tomato style before it reaches a tomato ovule. And here I'm using barley as an example of a grass. Sorry, yeah, as a monocot species. And here in barley, as in maize, pollen are captured on these projections from the style, which is a silk in maize. The pollen tube will then germinate and grow down the silk. The silk will lead to an ovule where the female gametophyte develops. And just as in Arabidopsis and tomato, the pollen will deliver 2 sperm for double fertilization. So double fertilization produces seeds. So these are Arabidopsis seeds 1 day after pollination. You can see the zygote developing here with the zygote nucleus, and then several endosperm nuclei that have already divided. And then a few days later, you can see the embryo beginning to develop where one of the sperm cells fertilize the egg. And in Arabidopsis, the endosperm is quite advanced 4 days after pollination. Of course, double fertilization also produces really important seeds. So every kernel of corn, every kernel of wheat, every kernel of rice requires this process. Each one requires successful growth of a pollen tube and fertilization for these very important agricultural products to be produced. Okay, so I think they're just, I'd like to take the opportunity to explain briefly pollen development because it's really interesting and is relevant to some of the things that we've talked about so far. So inside the anther, tapetum, which is a specialized structure where the microspore mother cells are, they will undergo meiosis to form a tetrad of 4 microspores. And each of those microspores will then undergo further microspore development. This culminates with a really interesting cell division where the germline divides away from the haploid cell derived from meiosis, and it actually buds into the pollen vegetative cell, resulting in the pollen cell with a second cell inside of it that's called the generative cell. So you'll note that the generative cell has its own plasma membrane and its own nucleus. The pollen nucleus exits the cell cycle, while the generative cell re-enters the cell cycle and duplicates its DNA and divides to form the 2 sperm cells. So in Arabidopsis, this happens before the pollen are released, but in other plant species, for example, tomato, the sperm cells don't divide until after the pollen tube begins to grow within floral tissue. So I just want to point out this picture of an Arabidopsis pollen grain and thinking about delivery of DNA using nanoparticles into pollen. It's really interesting to look at and think about the really tough sporopollenin wall that encloses the pollen grain. But I do want you to note that in most pollen, there are these apertures which present openings into the pollen wall that give access to more access to the plasma membrane of the pollen that's inside, presenting a potential route for nanoparticles such that they could avoid this tough sporopollenin wall. Okay, so another important thing for breeders is the recognition that the haploid nucleus in the pollen tube of the pollen cell is the same exact haploid nucleus as that present in the 2 sperm cells. So the male gametophyte of flowering plants is shown here. It comprises a pollen cell and 2 genetically identical haploid sperm cells. All right, and just a little bit, just briefly about meiosis, because I think this is a really important point for potential breeding applications of pollen and also the female gametophyte. So if we imagine a plant that's a hybrid, say between 2 breeding varieties that one is interested in the diploid cell that will undergo meiosis, you can think of, in this case, this is a representation of tomato, which has 12 chromosomes. The hybrid is going to be heterozygous for every gene, every chromosome in the genome. But when meiosis happens, on average, there's about 1 crossover, or maybe sometimes 2 on each chromosome. And that's going to result in individual unique haploid genomes that get packaged into gametophytes. Either the pollen and sperm or the female gametophytes, which I will tell you about their developmental program in a second. Okay, so this then is a little packet of genetic information that can be really useful. So some of the things that I'm interested in are identifying traits that are important for the sporophyte using the pollen phase, using pollen to screen for interesting combinations of gene variants in the pollen phase. These 2 are really relevant for our new project on trying to optimize pollen tube growth for high temperature. The capacity to capture a genotype in the haploid phase, double that, and generate a sporophyte receives much attention in many different species as a way to accelerate breeding of the sporophytic part of the life cycle. Of course, it would be interesting to breed and engineer pollen traits, for example, resistance to high temperature. And as we talked about earlier, it would be really interesting to use pollen as a way to deliver transgenes to the egg, as Michael Strano's work is getting at using nanoparticles. Okay, so I just wanted to briefly go over female gametophyte development in the ovule because there are some important differences. So the megaspore mother cell also undergoes meiosis within a single developing ovule. The major difference between male and female gametophyte development is that only one of the female products of meiosis survives. The other three degenerate. And then a second major difference is that the female gametophyte undergoes an extra round of cell division. And in this case, all of the cells stay in the cell cycle and complete division until female gametophyte development is completed. So we go from 1 haploid cell to 2 haploid cells to 8 haploid cells, and then the two polar nuclei, so one from this pole and one from this pole, migrate toward the center here. They'll fuse to form the central cell nucleus. The cells at the micropilar pole take on really interesting identities, the 2 synergid cells, or SC, that are responsible for attracting the pollen tube, and the egg cell, which constitutes the female germline. And then at the other pole are 3 antipodal cells that we don't really know a whole lot about their function. So this is just another way of looking at that structure now as a cartoon inside of an ovule. Here you can see a pollen tube growing toward a pair of synergid cells. You can see the 2 sperm cells located, situated next to the pollen tube nucleus, and the 2 haploid synergid cells, the haploid egg cell, the central cell, which is the product of fusion of 2 haploid nuclei, and the antipodal cells here. Okay, so the first little pollen selfie vignette that I'm going to tell you about is work from my group that was done mainly by Alex Layden and more recently by Nathaniel Palmvere. They were both graduate students who completed their PhDs in my lab, and they provided us with some new insights into how sperm release signaling works. Okay, so this project starts with a really interesting movie that Alex Layden made. So we can grow pollen in vitro in a couple of different ways. One way we call the semi-in vivo pollen tube growth approach, where we take the top part of a flower, we clip off the stigma and place it on the surface of a petri dish. And in this case, we've pollinated the stigma with pollen expressing GFP. And then on the surface of the media, we open up the pistil and we excise ovules and also place those on the surface of the media. And then just below the ovules, we just place pollen grains that are not growing through pistil tissue, but are just placed directly on the surface of the media. And just so we can keep the 2 sets of pollen tubes separated, we've used pollen in this second experiment that are expressing DS red. So the way this looks on the surface of the plate is shown here. So this experiment was done by Alex Layden, but it was used a technique for this semi-in vivo pollen tube growth approach that was developed for Arabidopsis by Ravi Palani-Velo at the University of Arizona. All right, so I love this movie because it really demonstrates the dynamic nature of pollen tube growth and highlights some of the really interesting aspects of pollen biology. So you can already see some GFP expressing pollen tubes have started to grow out of this pistil explant. The ovules are here, and here are in vitro pollen tubes that we just put right on the surface of the media. And I just want you to watch this movie, and as you watch it, make some observations. Try to think about what's different between the green pollen tubes and the red pollen tubes as you watch this. So already we can start to appreciate some bright green dots that are forming inside the ovules. Those are positions where the pollen tubes have reoriented toward the micropyle, entered the micropyle, and burst to release GFP and their cargo of 2 sperm cells into the synergid cells. One thing that you may have noticed is that the red pollen tubes that didn't grow through pistil tissue, they grow more slowly. And also in the cases where they do come near the micropyle, they don't turn into the micropyle. So the pollen tubes that haven't grown in contact with the pistil tissue are not competent to respond to those lure guidance proteins in vitro. So as a way to begin to understand what's different about the pollen tubes that have grown through pistil tissue and those that are just growing on the media, several years ago now, we collaborated with Ravi's lab to do transcriptome analysis using, at that time, microarrays. And the really interesting thing about that study was that we found that there were about 1,200 genes that pollen tubes, those GFP expressing pollen tubes, express, that the pollen tubes grown in vitro don't express. So the conclusion from this was that something really important and significant happens to pollen tube physiology as it grows through pistil tissue. So as a way to try to understand that transcriptional response, we looked for transcription factors that were expressed in pollen tubes growing through the pistil tissue. And we identify a member of the large Arabidopsis MIB families, MIB 120, that is not expressed in unicellular pollen, not expressed in bicellular pollen, not expressed in tricellular pollen, isn't expressed in dry pollen grains, but then only begins to be expressed in pollen tubes that have been grown in vitro, and then is particularly highly expressed in pollen tubes that have grown through pistil tissue. So a postdoc in Ravi's lab named Yuen Chin isolated bundles of pollen tubes that had grown through pistil tissue for this microarray experiment, which has been since been repeated with RNA-seq. So we identified 3 MIB transcription factors, MIB 120, MIB 97, and MIB 101 from this small MIB subclade that all seem to be really prominent in growing pollen tubes. And we made a triple mutant lacking those 3 transcription factors and found a really interesting reproductive phenotype. So if you pollinate a wild type pistil with wild type pollen, you see nice seed formation up and down the pistil. But if you do that with triple mutant pollen, you see these, you see a few fertilized seeds, but most of the seeds were unfertilized and were beginning to shrivel up at this point in development. So we used the LAT52 pollen express GFP and to characterize what a wild type pollen tube does when it enters the ovule. So here we can see this is a synergid cell. This is the outline of the rest of the female gametophyte shown in red autofluorescence. And when a wild type pollen tube enters the synergid cell, it bursts. It makes this big green dot in one of the 2 synergid cells and releases its cargo of sperm. But what we found with most of the triple mutant pollen tubes is that they enter the synergid, the female gametophyte, but they continue to grow, forming these coils of pollen tube that fail to release sperm. Interestingly, this phenotype that the MIB mutants showed phenocopies observations that are made when you do interspecific crosses. So for example, Here is an Arabidopsis by Arabidopsis cross-pollination. And what we're looking at here is an aniline blue stained pollen tube growing into the micropyle and bursting. If we use an Arabidopsis pistil and cross it with the closely related Limarabidopsis pumila pollen, that closely related pollen can find the micropyle, enter the ovule, but then it grows into one of these characteristic coils that's very reminiscent of the MIB triple mutant phenotype. So our hypothesis about the MIB transcription factors in the pollen tube is that they are controlling the identity of the pollen tube that must be recognized by the female gametophyte in order for proper signaling to occur that allows the pollen tube to release its cargo of sperm. Okay, so we wanted to develop a technique that would allow us to identify the key genes that these MIB transcription factors are regulating in a pollinated pistil. So the problem that we have here, and we wanted to get away from the semi-in vitro system because it's invasive, and we were worried that that pollen tubes growing through out onto the surface of media might be mounting some form of stress response. The problem, though, is that it's impossible to extract intact pollen tubes from pistil tissue, and pollen tubes represent only about, our guess is about 1% of the cells in a pollinated pistil. So we really needed a way to separate the pollen tube transcription signal from the pistil transcription signal so that we could identify the genes that the MIB transcription factors are regulating in the pollen tube as it grows through pistil tissue. And we decided to take a bioinformatic approach to do this that takes advantage of polymorphisms that you can take advantage of if you cross pollen and pistil from two different Arabidopsis accessions in this case. Okay, so if you do an RNA-seq experiment on a pollinated pistil with pollen and pistil from the same Arabidopsis expression, and you map reads and quantify reads to specific genes, you're not going to be able to tell whether these reads are coming from the pollen parent or the pistil parent. But on the other hand, if you use the pollen parent from one accession and a pistil parent from a different accession of Arabidopsis, and this would also work with different varieties of tomato or maize or whatever your favorite crop plant is, you can then use polymorphisms in the RNA-seq reads to determine whether a particular read came from the pistil parent or the pollen parent. And in so doing, we can then quantify, in this case, gene A, we would know, comes strictly from the pollen parent. Gene B comes strictly from the pistil parent. And for gene C, which has reads that come from both the pistil parent and the pollen parent, we can quantify the relative proportion of gene expression from the two parents. Okay, so for our first experiment using this system, we used the Cape Verde Island accession, CVI, as the female and crossed it with Columbia pollen. And those two accessions have a SNP, a single nucleotide polymorphism, about every 180 base pairs. So interestingly, about 60% of the genes in the Arabidopsis genome with these two accessions had a SNP that allowed us to identify whether an RNA-seq read came from the pistil parent or the pollen parent. Most of the SNPs are in exons, but any SNP in a 5' UTR, a 3' UTR, or an exon is useful for this bioinformatic analysis. And just one thing I wanted to point out, it was kind of surprising that about 8% of the reads that had polymorphisms that allowed us to differentiate pollen from pistil were coming from the pollen parent. So the pollen tube is expressing a significant amount of transcript relative to the pistil in a pollinated pistil. So we use this to further characterize, we use this to do experiments where we compared wild-type pollinations with MIB mutant pollinations. And we found a really interesting set of large gene families that were not expressed in MIB mutants. So these MIB transcription factors of this type are known to be activators. So we were looking for genes that were failed to be expressed in the pollen tube. And we found families of small secreted proteins, cell wall modifying enzymes, and transporters. And so our model is that the pollen tube is expressing these factors that allow it to properly interact with the synergid cell, such that in a wild type case, the pollen tube and synergid cell exchange signaling that results in this simultaneous lysis of the pollen tube and the synergid cell, so that the sperm cells get released for fertilization with the egg and central cell. Okay, so an area of work in our lab now is to try to identify the key MIB target genes that are signaling back and forth to the female gametophyte. On the female gametophyte side, it's already known that proteins encoded by the Feronia, Lorelei, and Nordia genes are all important for this same signaling event. So loss of function in either of these three female mutants results in the same pollen tube coiling phenotype as the MIB mutants. So we're really interested to determine whether any of the MIB target genes are interacting with the synergid cells for proper pollen tube, for proper sperm release. So one approach that we've taken is to use the yellow chameleon calcium sensor, which in this case is being expressed by the synergid cells, which are shown here. And here you can see a pollen tube growing toward a synergid cell. So previous work done by Thomas Dresselhaus's lab and by Uli Grossniklaus's lab had shown that when the pollen tube arrives and contacts the synergid cell, there's a characteristic calcium oscillation. So Nathaniel Palmvere in my lab is using that system to determine whether the MIBs elicit the same synergid calcium response or not. And here's just an example of one of his movies. You can see the pollen tube now touching the synergid cell, which starts this oscillation of calcium within the synergid. The pollen tube is about to speed up as it interacts with the synergid cell just before it bursts just there. And when it bursts, you see a big burst of calcium as both the pollen tube and one of the two synergid cells lyses. So what we've found so far is that MIB mutants and pollen from closely related species are able to elicit a Feronia-dependent calcium response. So this tells us that pollen tubes are controlling a signaling event that's downstream of the Feronia-mediated synergid calcium response that's important for this interaction between the pollen tube and the synergid cell that results in sperm release. Okay, so next I want to tell you about some things we've learned about the interactions between the sperm cell, the pair of sperm cells, and the female gametes. And this project led to the identification of a protein that we called HAP2, that's also called GCS1, as the sperm-expressed gamete fusion protein that's likely responsible for double fertilization across the flowering plants. And this is work that was done by two graduate students in the lab. The project was mainly started by Kristen Beal and then was later worked on by Jennifer Forsina. All right, so I'm going to start to orient us with a movie from Tetsuya Higashiyama's lab that was taken using a confocal microscope some years ago now. And this provided us with the first glimpse of live imaging of double fertilization. So here, the sperm nuclei are expressing a histone related protein, HTR10, fused to RFP. So the sperm nuclei are expressing RFP. The pollen tube is expressing GFP. So what you're going to see is the pollen tube moving, it's into the synergid cell. It bursts really rapidly. I'm just going to play this again, and you can see the two sperm nuclei getting ejected to right here. And then they fuse, the plasma membranes of the sperm nuclei fuse with the egg here. And then you're seeing the nucleus undergo karyogamy with the egg nucleus, and the other sperm nucleus undergo karyogamy with the central cell nucleus. So interestingly, Yuki found that the sperm, after they're released, they sit at this site of fusion for about 7 minutes. And my lab has been particularly interested in what's going on during those 7 minutes before the plasma membranes fuse. And that's because I had identified a mutant that I called HAP2 that completely blocks the ability of the two sperm cells to fuse with either the egg or the synergid cell. So this is, I'm sorry, either the egg or the central cell. This is an experiment that Kristen Beal did, where she was using that same HTR10 RFP sperm marker. So the nuclei of the sperm are shown here in RFP. And in this image, this confocal image, the egg cell nucleus and the central cell nucleus are expressing a nuclear localized GFP. So here in a wild type, if you pollinate with pollen expressing this marker and come back about 16 hours later, you can see that the sperm nucleus has already begun to fuse with the egg nucleus, and the other sperm nucleus has begun to fuse with the central cell nucleus. But if you do the same experiment with HAP2 mutants, the sperm cells will just sit at the site of gamete fusion. And because their plasma membranes can't fuse with either the egg or with the central cell, the nuclei then can't migrate to those two target genes. So HAP2 is conserved across many eukaryotic genomes. And now we call it an ancient gamete fusion protein. So its HAP2 sequences are present in plant genomes, genomes of chromista, genomes of some animals, but not vertebrates, and also in protist genomes. So far, HAP2 has been shown to be essential for gamete fusion in Arabidopsis, the data that I just showed you, and in Chlamydomonas reinhardti, also in Tetrahymena and Plasmodium, also in Nematostella vectensis, a basal animal, and in Dictyostelium discoideum. So this is an ancient protein. It's plausible that it was present in the first sexual eukaryote, and that this mechanism that I'm about to describe was the first mechanism that eukaryotes evolved to enable two germ cells to fuse with each other to form a zygote. So the real breakthrough for this project came when Juliette Fedre, who was then a graduate student in Felix Ray's lab in Paris, they had read some papers about HAP2 and decided that it might have the same structure as a viral fusion protein. So Juliette set out to find the structure of HAP2, and here is the structure of the Chlamydomonas HAP2 from her Cell paper that was published in 2017. And here's the structure of the tick-borne encephalitis virus glycoprotein E, which is the protein that TBEV uses to get into a host endosome. This structure is showing a trimer of the protein, and this is the conformation that occurs after gamete fusion. So this is representing the fused membrane, which is not a part of the structure, but is this is the part of the protein that interacts with the membrane. Okay, so class 2 fusion proteins insert into their target membrane. So when the protein is on the viral envelope, it's in an inactive form. When it's activated, the second domain, domain 2, interacts with the host membrane, and part of domain 2 inserts into the host membrane. And then upon trimerization, there's a conformational change, that pulls the two membranes in close proximity, squeezes out the water that prevents fusion of the two membranes and allows the two lipid bilayers to merge. So this is the state that's been, the structures that we have to date of HAP2 are in this conformation, and they lack the transmembrane domain of HAP2. So Juliette also obtained the HAP2 sequence from Arabidopsis, and it had this really interesting alpha helix that we called alpha F, or a fusion helix, in its apex. This is showing a trimer of the structure. And this is just a zoom in on the residues that are in that helix. So the idea is that these residues interact with the lipid bilayer of the female gametes, allow HAP2 to insert into the lipid bilayer, which then allows HAP2 to trimerize and pull the two membranes together. So through genetic experiments that Jen Forcina in my lab did, and biochemical experiments that Juliette did, we were able to show that the residues on the apex of alpha F are required both for insertion into artificial liposomes, but also for fertilization in vivo. And so this is just a reminder of the way that the sperm look like when they're still in the pollen tube nucleus. One of the sperm cells is wrapped around the nucleus of the pollen tube, and the other sperm cell is attached to that sperm cell. And here you're looking at confocal micrographs that Jen Forcina took of HAP2 YFP localized to the pollen tube plasma membrane in the growing pollen tube. So the pollen tube tip is oriented to the top of these images. You can kind of see the tail of one of the pollen tube, of the sperm cell that's wrapped around the pollen tube nucleus here. You can see that a plasma membrane localized RFP also localizes to the plasma membrane, and those two proteins co-localize. The mutants that we made that disrupted fertilization in vivo don't disrupt localization. And so this is really showing the nice interaction between the two sperm cells that are really delivered as a packet into one of the synergid cells so that one sperm can fuse with the egg and the other with the central cell. Okay. So just to summarize what we've learned so far about HAP2, it's an ancient gamete fusion protein. It's broadly distributed across eukaryotes. Interestingly, the membrane interaction surface of HAP2 is variable. So Juliette also has obtained the structure of HAP2 from T. krusei and Chlamydomonas, and the apex is very different from the flowering plant apex. Although a recent paper showed that the Chlamydomonas HAP2 also has an amphipathic alpha helix that inserts into the plasma membrane. So this apical fusion helix appears to be functionally conserved across all of the flowering plants. So we were able to take the rice sequence of that amphipathic helix, substitute it into the Arabidopsis sequence, and rescue our HAP2 mutant using that construct. So it seems that this mechanism of gamete fusion, where the sperm expresses a fusion protein that, when activated, can mediate fusion with female gametes, is conserved across the flowering plants. It's really interesting that this protein has adopted to the broad diversity of sexual systems in which it functions. So the environment inside of the flowering plant ovary is a very different environment from where, for example, Chlamydomonas or Tetrahymena is undergoing gamete fusion. It's probable that HAP2 function is regulated in different ways across these systems, and that's an area that we're really interested in pursuing in the future. Also, now that we know more about the structure of this fusion protein, it'll be really interesting to see if we can identify HAP2-like proteins in organisms in which gamete fusion mechanisms are not currently understood. And there's some really important organisms, for example, humans, for which we don't have a concerted biochemical mechanism for how the sperm fuses with the egg cell. And so it will be really interesting to see if eventually we identify a HAP2-like protein that's not identifiable in the human genome by primary amino acid sequence, but could be identified by virtue of its structural characteristics. Okay, so our current model for double gamete fusion is that when the sperm are traveling in the pollen tube, they're in an inactive form. And so either they're dimerized, as shown here, which is one of the mechanisms that negatively regulates the fusion protein in viruses, or it could be that HAP2 is interacting with a partner protein that we don't know about yet that keeps it from being active while it's in the pollen tube. So upon release into the female gametophyte, into the synergid cell, where it has access to the egg cell and the central cell, HAP2 is activated, and where we think that the apex of HAP2 then inserts into either the egg or the central plasma membrane, egg or central cell plasma membrane. There's really nice work from Stephanie Sprunk's group on a protein called EC1 that's secreted by the egg cell that seems to activate HAP2 because loss of function of EC1 prevents HAP2 function. So it could be that EC1 is involved in this activation step. And then the final step would be this conformational change to form the structure of the protein that we got from structural analysis that would bring the two plasma membranes together and merge them to form either the zygote in the case of fusion with the egg or the primary endosperm in the case of fusion with the central cell. Okay, so obviously this is a really important mechanism to understand because this fusion event is what triggers seed development and fruit development across our really important crop plants. Okay, so with the last couple of minutes, I'm going to finish with a more recent project that we've started in the lab, and that's to really think about how pollen performance is affected by heat stress. A graduate student in the lab named Sorel Imga is working on this project along with an undergraduate named Ben Styler. Okay, and the motivation for this work is that the climate is getting warmer. So this is one of the climate maps that you can find showing the expectation that between 2040 and 2060, much of the globe will be experiencing warmer summers than they've ever experienced before. This is going to have a big impact on agricultural productivity for many reasons, but we think that one important impact is going to be on pollen development and function. And our lab is particularly interested in how high temperature is affecting the ability of the pollen tube to navigate the pistil tissue and deliver sperm cells for fertilization. So for this project, we're using tomato as a model system. So if you pollinate a tomato pistil with a full load of pollen, and you allow those pollen tubes to grow at 28 degrees Celsius, which is the optimum for many tomato varieties, and you wait about 12 hours, plenty of pollen tubes will grow down and find dozens of ovules, deliver sperm cells to each one, and the result of that is a nice big tomato fruit with dozens of seeds, and a nice tomato. On the other hand, if you do the same experiment at 37 degrees, what happens is many of the pollen tubes will germinate, but they'll fail as they grow down the pistil tissue. And the result of that in the case of tomato is flower loss. So an unpollinated tomato flower will just abscise. The tomato flowers that stay on and develop into fruits will have reduced seed numbers because very few of the ovules will get fertilized by pollen tubes. And this results in loss of tomato fruit, or for the fruits that do get produced, a decrease in fruit weight. Okay, so we've been doing experiments like this one where we grow pollen tubes in vitro at 28 degrees, in this case for 6 hours. And you can see that over 6 hours, we start to get pollen tube germination at 28 degrees, and we can compare that with pollen tubes grown in vitro under temperature stress. And these experiments were done a few years back by a group of undergraduates, Danielle Alvarez, Tu Pham, and Grace Cinderella. More recently, Sorel has been developing live imaging that allows us to couple some software that he's written that automatically tracks the pollen tube tip, which provides us with a much richer view of pollen germination and the dynamics of tube growth over a time course. But these early experiments showed us that if we take thermosensitive varieties like VF36, which is a commonly used variety of tomato, or M82, which is commonly used for genetics systems, if we grow pollen tubes at 28 degrees versus 34 degrees, we can see that in VF36, the average pollen tube length goes down. In M82, it goes down significantly. These green bars represent the relative mean length of 34 divided by 28. So high temperature decreases pollen tube function for most varieties of tomato. But we're using also a set of varieties that have been bred for thermotolerant fruit set that we obtained from the UC Davis Stock Center. Tamaulipas, San Marzano, Nagkarlung, Malinka, and Hotset. And if you grow their pollen tubes at 34 degrees, in some cases, for example, in San Marzano, the pollen tubes grow much better at 34 degrees than they do at 28. So for San Marzano, Nagkarlung, and Malinka, they all do better at 34 degrees than they do at 28. Whereas Tamaulipas and Hotset in vitro did not show thermotolerant pollen tube growth. So these data suggest to us that when breeders have been identifying germplasm that results in fruit set at high temperature, they've been selecting for gene variants that provide enhanced pollen performance at high temperature. So the goals of this project are to define the pollen tube and pistil transcriptional responses to heat stress, identify genes conferring pollen tube thermotolerance using a haploid phase mapping approach that we're doing that allows us to directly identify gene variants that confer enhanced pollen tube performance at high temperature, with the overall goal of identifying gene variants that are necessary and or sufficient for thermotolerant pollen tube function. So let's see. Yeah, I think the timing is about right. I'm going to wrap up there and rejoin you. So I just want to thank the people in my lab who have worked on sperm release and gamete fusion, mainly Jen Forsina, Kristen Beal, Nathaniel Palmvere, and Alex Layden, and acknowledge the NSF for support for those projects. And I also want to acknowledge the pollen heat stress project team that brings together collaborators from the University of Arizona, UNC Charlotte, and Wake Forest to try to understand how pollen responds to high temperature and to discover gene variants that allow it to perform under temperature stress. And that project is also recently supported by the NSF. So with that, I will stop and would be really glad to take some questions. Thanks.
Thank you, Mark. I think Jody has posted a couple questions in the chat. I'm not sure if you can see the chat. If not, I can read it out. So there's one question is, in the mature or germinating pollen grain, where is the gene expression coming from? Is it come from the vegetative nuclei of sperm cell or all of them, you know?
Yeah. So the short answer is all of them, and each of those cell types has its own gene expression program. Just as an example, the HAP2 gene that I talked about that's important for fusion of the sperm with the egg and central cell, that gene is only expressed in in sperm. It's not expressed in the pollen nucleus, and it's not expressed anywhere else in the plant. So that gene expression program for the sperm doesn't begin until the generative cell is formed and becomes distinct from the pollen nucleus. On the other hand, there are many genes, including the MIB transcription factors, that are expressed exclusively from the pollen tube nucleus, but are not expressed in the sperm cells and are not expressed anywhere else in the plant. So the short answer is that each of those cell types has its own gene expression program. And I should point out that other work has been done to isolate the sperm cells from the pollen tube cells using cell sorting techniques, and RNA-seq analysis has been done. And there are data sets available that allow you to identify all of the sperm expressed genes and separate those from the pollen expressed genes. And the same is true on the female side. So the egg cell has a different gene expression program from the two synergid cells and from the central cell.
Okay, I have another question. I put them in the chat. It's pretty long. Has the SNP-based RNA-seq approach with pollen tubes been used in interspecies crosses to identify where compatibility barriers exist at the molecular level, or be used to identify and express factors that overcome those barriers?
Yeah, there's actually a really nice paper from Tomato that explores that very phenomenon. So this is, the first author is James Peace, who's now a collaborator on our current Tomato project. But James was using interspecific incompatibility in tomato that's mediated by a pistil expressed ribonuclease to characterize genes that the pistil expresses and that the pollen expresses during both incompatible and compatible pollinations. That paper came out, I think, in 2016, was published in Molecular Ecology, and yes, you can find lists of genes that are associated with compatible pollinations versus incompatible pollinations. And that technique is, I think it's interesting because it doesn't require marker lines, it doesn't require transgenic plants, and as long as you have two polymorphic parents in a cross, it can be applied to any system.
I think we're just right on time. And thank you so much for your awesome presentation.
Thank you.
Annie?
All right. Thanks again to Dr. Johnson and to our other two speakers this morning, Dr. Lee and Dr. Strano. I want to thank you so much. So we are going to go on break for now, a longer break, a half an hour break for lunch, depending on the time zone that you're in. It might be a very late lunch for some individuals on the East Coast there. So with that, I will put us on break, and we will resume at 12:50 Central Standard Time. So please don't go too far, but please get some coffee, tea, or lunch if you haven't had lunch yet, and we will resume at that time for the second half of our presentations with Dr. Scanlon, Dr. Key, and Dr. Voitis. So thank you, and we'll be back at 12:50 Central Standard Time.
Hello, good afternoon. Welcome everybody back to the session. I hope everyone had nice lunch break and refreshed. So for those just joining this afternoon, so my name is Yurong Chen, cell biologist leading a transformation optimization team with Bayer Crop Science. And it is my pleasure and a great honor to introduce our next speaker, Dr. Michael Scallon from Cornell University. Mike is a professor at Cornell University, and he received his PhD in genetics in 1993 from Iowa State University, working with Alan Myers and Don Robertson on maize kernel development. Michael was an NSF postdoc fellow studying leaf development with Michael Freeling at UC Berkeley from 1993 to 1997. After starting his own lab at the University of Georgia in 1997, Mike then moved to his laboratory to Cornell University in 2006. Research in Dr. Scallon's lab focuses on the mechanisms of plant development and evolution of plant morphology. Using comparative developmental genetics and functional genomics, Dr. Scallon is especially interested in the processes whereby meristems make leaves and embryos make meristems. Dr. Scallon's lab exploits forward-reverse genetic mutant of maize, Arabidopsis, tomato, and Mars as a foundation for comparative analysis of fundamental processes in plant development. The title of Dr. Scallon's presentation today is Maize Shoot Development at Single Cell Resolution. Take it away, Mike.
Well, thanks very much, Sharon, for this introduction and also for this great invitation to this symposium. I'm learning a lot. So, thanks a lot. Yeah, so as, oh, sorry, I just hit forward. Next slide, please. Yeah, I'm going to talk today about work that was mostly done by a fantastic graduate student in the lab, Jack Satterley, who's on the left. And then also it was helped by John Strabel, who's now just begun his own lab at North Carolina State University. He was a postdoc on this project. And it's all been funded by NSF. Next slide, please. So yeah, my lab is really interested in mechanisms of plant development. So how organs, tissues, and individual cells acquire their specific fates. And for this lab, for this talk, it's going to be focused exclusively on maize, which is my favorite organism. And on the left, you can see the study sample for this project. It's basically a 2-week-old seedling. And at that stage, shown on the left, you can see about 4 leaves, but it's actually made, this plant's made about 14 total leaves, most of which are buried inside each other in these whorls. And that white square at the very base shows where the position of the shoot apical meristem is that made all of these leaves, all of these stems, all these lateral organs, everything you see above ground in the shoot, I should say, of a maize plant is coming from the structure on the right, which is called a shoot apical meristem. And we're looking at here is that dome is the shoot apical meristem, or SAM, and those 2 bumps labeled P1 and P2 are the youngest and the next youngest leaves that have been made from this meristem. And the meristem of maize will make about 20 or so leaves in this inbred background before it basically becomes an inflorescence meristem and starts making, in this case, the tassel, which is, of course, the male flower. So it's really hard to get at the meristem, and we've done lots of different kinds of studies of this, genetics, and also lots of laser microdissection, carving up different regions of this meristem to understand how cell fate acquisition happens. But ultimately, the best way we think to understand how cells acquire their fates is to look at single cell gene expression. Next slide. So that's what we were trying. But there's a problem. I know many of you have seen lots of studies are coming out in scores now, different single cell analyses of different parts of the plants, but shoot meristems happen to be extremely difficult to make protoplasts from. We're not really positive why that is. The meristematic cells, as you can see in that previous slide, they have relatively very thin walls, but they're very hard to make protoplasts that survive. So we've tried and failed for several years at getting good numbers of viable protoplasts from the shoot apical meristem. We get them from other parts of the plant, but not the shoot apical meristem from the seedling. So Jack actually found in literature from the 1980s at a single lab at Yale University that was then studying how to make protoplasts from oats, and found that using different concentrations of mannitol, but more importantly, adding polyamines to the solution of protoplasting really helps the viability. So Jack did a number of tests and found that we found the best viability when you add this amount of 10 millimolar arginine and 0.65 molar mannitol. He also found, next slide, that if you adjust the pH to higher levels, the protoplasts from the meristems tend to survive much better. So there was a paper from 1980 that showed that the pH actually of the shoot apical meristem is much higher than other cells in the plant. So using these optimized protoplasting methods, next slide. Jack first tried this method using the 10x technology, and we didn't get any cells from the shoot meristem. So we don't know why, but the shoot apical meristem cells, even with this protoplasting improvements, are not surviving the 10x system. I suspect that the meristem cells are quite fragile. I haven't tested this, but I suspect that maybe the cytoskeleton is quite weak, and these things don't survive the 10x pressure system. So what Jack did was he dissected down to the shoot meristem shown on the left, plus 2 leaf primordia labeled P1 and P2. And this thing is only about 100 microns tall, the shoot meristems, which is very, very small. He dissected about 75 of these, protoplasted them for 2 hours in this modified system, and then basically put in a vital stain called FDA+, so he can distinguish dead cells from living cells. And he basically handpicked thousands of these cells, put them in wash solutions, and put them into 96 well plates, several 96 well plates. So really a Herculean effort to generate these protoplasts from the meristem. Again, they don't survive. They don't seem to survive the 10x system. And so after a couple of years of trying to get this to work, the next slide, please. Jack was able to basically generate this UMAP. It's basically dimensionality reduction of complex data, showing his data for a single cell analysis of the shoot apical meristem plus 2 leaf primordia. And so as you probably are familiar, each one of these little dots in this figure represents a single cell. And they're grouped according to their similarities in gene expression. So you can see maybe at the very base, those purple cells are where we think are coming from the SAM tip using known markers. Then there's the meristem, different regions of the meristem are shown in blue just above that, primordia connected to that, and then epidermis over there on the left. And the one thing that we noticed right away was these don't seem to sort into distinct individualized cell clusters like you see in many samples. They form a continuum. So that tells us that these states are really in a continuum, a dynamic flux of differentiation. The only ones that really seem to stand out separate from the others are the vasculature cells, shown there on the far right. Okay, so what are these cells? Next slide, please. The first thing we wanted to look at was what was happening in the meristematic tip. Now, there's a marker that was described by Maya Timmermans. I collaborated with Maya Timmermans for over 15 years, still collaborating on previous projects. And one of the things that she did with her lab was microdissect the shoot apical meristem bulk tissue, but microdissecting the tip versus the primordia versus the peripheral zone and things. And she found this great marker for the meristem tip, which we think represents where the stem cells are in the shoot meristem. And that's shown on the left in the situ from Maya's lab of this dynamin gene in the tip where we think the stem cells are. So we use that as a marker for stem cells. And then we asked, okay, what other genes are expressed in this stem cell tip, which is really, we don't really have any mutations yet in maize that remove the stem cell, like we have in Arabidopsis and other species. So next slide. using Dynamine as a marker, I should basically say there's been a comparison made between the stem cell niche in animals and plants. This is from a cool review in 2014. Similarities and also some differences. So on the left here, you can see what we're looking at is the developing intestinal cells of an animal, of a vertebrate intestine. And we see basically stem cells shown in blue, those stripes in blue near the bottom in the panel cells, are pressed next to stem cell organizing cells. Then immediately above that, basically are what are called transient amplifying cells. Then above those are differentiation zone where these differentiated cells are found. So there's a stem cell niche shown in the bottom. There's a proliferation zone, which is adjacent to the stem cells. And then final differentiation happens at the tip. And this study makes cool comparisons to this structure in an animal, and also on the right, what we know about the Arabidopsis shoot meristem, where the stem cells are at the tip, shown in blue. And they're organized by what we'll talk about, a gene called Wuschel, which is beneath the stem cells, well below the tip, those purple cells. And on the peripheral is basically where we think these transient amplifying cells in plants, we call these peripheral zone cells, are basically transferring their identity from stem cell into eventually differentiated cells. So they're in the process, in the middle of that procedure. Okay, next slide. So using Dynamine as a marker, we asked what other genes are differentially expressed in these tip cells. And we found there are a lot of genes that are involved in RNA-dependent DNA methylation, to keep heterochromatin, we think, from jumping around, and transposons from jumping around, and keep heterochromatin from being expressed. We also found a lot of genes. Next slide. Next slide, please. Oh, I can't see. Oh, sorry. Previous slide. Sorry. I've lost the slides. My toolbar is covering the bottom of the slides and I can't see it. But right now I can't see the slides. I don't know.
Let me unshare, Dr. Scanlon, and I'll try sharing again. You can't see the slides right now?
I can't see the slides.
1 second.
Yuck. I think I hit something. There it is. Okay, great. Yeah, the toolbar is blocking, so I can't see. But basically, we also found lots of genes that are involved in DNA maintenance, DNA repair. So it looks like this is also found in stem cells of animals. There are a lot of genes that seem to be involved in maintaining genome integrity, stopping the increases in genetic load, stopping the accumulation of mutations. This is also found in animal stem cells. Next slide. So yeah, the big question here is, the problem of the germline. So this is an extreme example, Methuselah tree, over almost 5,000 years old. How does a structure that's this old keeps on generating new structures like a plant does throughout its life cycle? How does it keep from accumulating mutations, not just in somatic tissue, but more importantly in the germline, that would basically increase the genetic load so incredibly that this thing would be so mutagenized that they couldn't possibly function anymore? So how does this work? So we think maybe this is what's happening in the stem cells with the expression of these RDRM genes, and also these genes involved in DNA repair. But also, next slide. The question was, what happens to the germline in plants? So this is a really cool summary review, I should say, published a couple of years ago, about different ways different organisms are proposed to basically maintain germline integrity, to keep the germline from accumulating mutations. And this study actually points out that we think we know a lot, but actually the evidence for some of this is not all that extremely Great. So on the bottom, we see what we think is happening in most animals, where there's early segregation of cells which will give rise to the germline, and they differentiate into germline cells, shown by red here, also quite early. On the top, A is what many people think, this is kind of like a dogma for what happens in most plants, where there's a late segregation of the germline cells from somatic cells. And they also differentiate very late. So in this case, the shoot meristem will make lots and lots of vegetative structures, and no cells in that shoot meristem have yet been segregated, put aside, or have yet been differentiated to become germline. This is what we think is happening in most plants. And in the bottom is perhaps early segregation of the germline, maybe sequestering it away so it's not undergoing lots of cell divisions. But again, late differentiation. Not really good evidence for differentiation of the germline early in any plants that have so far been studied. So again, the question is, how do you keep these mutations from accumulating? Next slide. Next slide, please. So what Jack did was he found that meristem cells exhibit relatively low rates of cell division as measured by the expression of genes, especially the G2 M phase and S phase genes, mostly G2 and M phase. And this is quite different than animal stem cells. Animal stem cells, pluripotent cells, seem to divide very rapidly and very frequently, whereas in maize anyway, these stem cells seem to exhibit very low rates of cell division. So the next slide, Jack actually used two different markers for these two different phases of the cell cycle. H3 on the top here in B, looking at a marker of S phase, and then Psych1 as a marker of G2M. And he partitioned the meristem into evenly sized domains, even height, I should say, 1 through 5, and he basically counted how many cells were expressing these different markers of cell division of S phase and G2M phase. And he found indeed the shoot meristem tip is very, very low in expression of these cell cycle genes as compared to the rest of the meristem. And particularly, it's not easy to tell in these longitudinal sections, but a lot of these cell cycle expression genes were especially high in the periphery and the outer edges of the shoot meristem, and not in the tip. So perhaps this is another way to keep basically these cells that are eventually going to give rise to the germline from accumulating lots of mutations through errors in replication, through lots of cell divisions, and so on. Okay, next slide. The other question that we addressed in this paper was the mechanisms of maintaining these stem cells. And as many of you are probably well aware of this Clavata-Wuschel system, which has been well described in Arabidopsis, tomato, lots and lots of other eudicots and some monocot plants, where Wuschel is basically a gene which is expressed in the organizing center below the stem cells, and it organizes stem cells in the meristematic tip above. And this gene is basically repressed by clavata function, this clavata-3 ligand, binding to its receptor clavata-1, which then somehow represses transcription of Wuschel to maintain this homeostasis in the stem cell size, to keep it the right size, not too big and not too small. And it's a classic, beautiful negative feedback loop where Wuschel actually activates expression of clavata-3, which encodes this ligand, which will bind clavata-1, and they work together to somehow repress Wuschel. And that mechanism is really unknown. Okay, so that's in Arabidopsis and lots of other species. This is really a canonical system, which any meristem review will pretty much reference this system. Next slide. But what we found was we didn't find any evidence of Wuschel expression in the shoot meristem of maize B73 anyway. And the same thing was found in laser microdissection analyses done by Meyer Timmermans, our collaborator, a couple of years ago. We did find some of these classic receptors, many of which, many more than clavata-1. I'm not going to talk about all of them in this summary. I've been described in Arabidopsis. But we know from previous work from Dave Jackson's lab that mutations in clavata-1, which make the meristem very, very large in Arabidopsis and many other species because they're not repressing Wuschel, and it keeps on making lots of stem cells, Mutations in clavata1 in maize do not affect the shaded buccal meristem at all. And the gene is actually not even expressed in this tip region. It's expressed in the primordia, and we're not really sure what it's doing. It is expressed, and it does cause this classic clavata-like phenotype in inflorescence meristems. So in the inflorescence meristems, which make the ear and the tassel, this Wuschel system has been demonstrated by Dave Jackson, Wuschel and clavata, maintaining stem cell size, but not in the shaded buccal meristem. Next slide. And so we presented this to reviewers. They didn't believe us. And so we actually did some RT-PCR and referenced Maya's previous paper. Looking at expression of Wuschel, there's two paralogs, I should say, in maize, WUS1 and WUS2. And this RT-PCR on the left shows after more than 35 cycles, really, we can't detect any evidence of a transcript in the shaded buccal meristem. We do find it in the ear, in the tip of the ear, actually, inflorescence meristem, and not in leaves. So yeah, there is no Wuschel-like function that's maintaining this stem cell pool in the B73 maize meristem. So the question is, next slide, please. What's going on in the meristem? What's going on in this so-called organizing center? So we're presuming that the tip of this meristem, where we saw dynamine expressed before, is where the stem cells are. Then logically, you might think that the organizing center should be below it, like we found in Arabidopsis, but Wuschel's not there. So what is there? So again, Maya's group in a collaboration that we were on with them showed this gene, which is the unknown function, no mutations ascribed to this gene, called unknown one. It was expressed in a region where we think the organizing center should be. And its expression is shown in the UMAP in the middle, in the meristem fairly broadly, mostly in the organizing center, above the tip, as you can see, not in the tip cells. So we then said, okay, this is the organizing center. We're presuming what other genes are expressed there. Are there any Wuschel-like genes, any other wax genes that are expressed there? What's going on in that region? So we looked at co-expression of cells that are expressing this unknown. Next slide, please. And this just shows the kinds of genes which are expressed there. Some of these we'll talk about later, but the next slide I'll talk about a few of them and where they're actually expressed. Next slide, please. So yeah, there's this gene called DRM1, which is previously described, and on D, you can see it's expressing roughly in this organizing center and also a little more broadly in the shaded buccal meristem. And in the panel beneath that, in F, you can see its expression is really more intense in the axillary meristem, which will make the ear in this case. At this stage, the structure in F, which is showing that purple color, is making husk leaves later on will greatly enlarge and form an ear meristem and basically make flowers. We also found expression of this grass homolog called GRAS33 in the organizing center and also in the peripheral zone and also in leaf primordia. And again, its expression in the axillary meristem is shown in panel E. It's also expressed there. So we have no idea pretty much what this grass homolog was doing prior. We had some evidence that this DRM1 gene was somehow involved in oxygen-induced dormancy. Might be why it's expressing really highly in this axillary meristem. In maize, most of the axillary meristems that form in every node of a maize seedling do not actually become functioning branches. So maybe this is undergoing some kind of dormancy. We're not really sure. But the next slide shows phenotypes we found for the ham homolog, which we're calling GRAS33. So this is important because in Arabidopsis, this hairy meristem or ham gene interacts with Wuschel, the stem cell organizer, to basically somehow repress clavata3, keep it from repressing Wuschel expression, and to maintain stem cell homeostasis. So this in Arabidopsis, this ham gene interacts with Wuschel, and it basically maintains the stem cells. Next slide, please. So what Jack did was he found mutations, uniform mu insertions in two different paralogs of this, the maze paralogs of this ham gene, which we're calling GRAS32 and GRAS33. These are they seem like orthologs of hairy meristem. And he found that when they were mutated, indeed, the meristem is smaller. Looks like the stem cell pool is probably smaller. They're not as tall as their siblings. Their width is also reduced, although not significantly in the double mutants, but the height is greatly reduced. So yeah, so it looks like this ham is functioning in maize as it is in Arabidopsis, but it's clearly not working together with Wuschel like it does in Arabidopsis. So the question is, what is the stem cell organizer? Right now, we don't really have a very good idea as to what it might be. Next slide, please. Looking at the other Wuschel-like genes in maize, we found just 2 that were expressed in the shoot apical meristem. None of them were expressed in the organizing center. These are WOX3 and WOX9. I know some colleagues are working on reverse genetic mutagenesis of WOX9. We've got some WOX3 reverse genetic mutants right now in the greenhouse. And it doesn't look like their function is really involved in stem cell maintenance. So again, the Wuschel-like function in B73 of maize anyway is unclear. Okay, so one of the coolest things you can do with stem cell, single cell, I should say, RNA-seq, is reconstruct these differentiation trajectories, which we call pseudotime, to basically infer gene expression throughout ontogeny. As you go from indeterminate cells shown on the base of this UMAP, marking expression of knotted-like genes and knotted, which I'll describe in more detail in a minute, and then also as gene expression changes as you basically form epidermis, form lateral organs, and ultimately form vasculature on the upper right part of this pseudotime trajectory. So basically, knotted 1 shown on the right here is a gene which is, I'll talk about this a lot more later, is implicated in basically stem cell maintenance, indeterminate maintenance, I should say. It's expressed throughout the meristem and also in the stem. And we think this its function is to maintain indeterminacy of these cells. And we'll talk about this a lot. So this is a good marker for indeterminacy. And we can use this basically to tell us which genes look like they're so-called meristematic. And we use other markers, as I'll show in the next slide, to tell us which genes are representing different stages in ontogeny as they basically differentiate. So the next slide, please. So this is what the pseudotime trajectory showed us. Again, in the stem cell niche, we find knotted-like genes, related genes, like the CNF1 gene, which is expressed in the stem cells. The next stage in this pseudotime trajectory, basically through ontogeny, is boundary-expressing genes, like this one in the middle, this HYPE1 gene. It's expressed at the boundaries between, in this case, leaf primordia and stems, but also it's expressed at the boundaries between young leaf primordia and the shoot meristem. Throughout shoot ontogeny, we see these boundary genes. And the last step in pseudotime trajectory is basically differentiation, represented by genes expressed in developing primordia, like the YABIs, RF2s, and others. This WOT1-like gene, it's expressing in primordia. Okay, and this was done first for the SAM plus 2 leaf primordia. Next slide, please. We also did this on the SAM surrounded by 6 leaves. Okay, so in this, it's shown on the right part of that black figure. You can see the sample that Jack actually took. The meristem is represented by that little is in this region in that little bracket at the very base of that little piece of whitish tissue. So this would actually contain the shoot meristem, lots of stem tissue, and lots of leaf primordia, up to 6 leaves from the meristem. And for this analysis, he basically was able to use a 10x genomics platform, which gave him a lot more cells, but it didn't give us any meristematic cells. So what we're looking at here is what's the pseudotime trajectory profile for later stages in shoot development. This is basically replete with genes from later stage primordia from stems and from vasculature, not from early stages. And again, this is what the trajectory looks like, going from indeterminate cells. This mid phase is like the boundary cells where you're starting to make organs. And then there's determinate cells, including leaf primordia, the vasculature, and also some mature stem. Next slide, please. And what he found, maybe not surprisingly, was what the same kinds of genes, markers of pseudotime trajectory in a young shoot with the meristem plus 2 leaf primordia, the same kinds of genes were found in an older shoot. Okay. So this thing is continuing to make leaf primordia as a shoot meristem does, continuing to make stems. And so the same kinds of genes are used early as are used late, which we think is pretty cool. Next slide, please. So the last part of this part of the talk is looking at the effects of a gain-of-function mutation in NODD1 on cell fate progression. So I introduced NODD1 before. Its expression is shown in an in situ on the left by David Jackson many years ago, a beautiful in situ. So wherever you see that blue, that's the NODD1 expression. This was the first homeobox box gene ever identified in a plant by Sarah Hake over 30 years ago. And it was basically first identified by its effects in dominant mutations. So on the right here, and actually in the right middle, is a plant which is a NODD1 plant, which is basically a dominant mutation, which we now know overexpresses NODD1 ectopically. So in the wrong place in the wrong time. On the left middle is a wild type plant, and you can see basically the structure of the leaf. The sheath surrounds the stem, and it basically holds the plant close, holds the leaf close to the stem, and it also adds strength to the stem. And then the blade, the upper part, the distal part, projects away. This is the major photosynthetic part of the plant. And in between, at that kind of triangular-like junction, is the ligule auricle, which I've studied a lot in the last couple of years. And basically there is a structure called the ligule, which forms at the boundary between the sheath and the blade. Okay. Now, in wild-type plants, NODD1 is expressed in the shoot meristem and the stem, but we don't see a lot of it in the leaf primordia. It's turned off. It's a marker of leaf initiation. Okay. These plants that have these dominant mutations in NODD1, they express NODD1 in the wrong place at the wrong time, so topically. And there's a cross-section, a transverse section through a maze shoot showing these coils of leaf primordia. And this dark blue expression is the expression of NODD1 over the vasculature, which is not normal. So this is the ectopic, wrong place, wrong time expression, okay, of a gene that we think promotes indeterminacy in leaves. And when it does this, it makes plants that have these incredibly cool bumps, overgrowth of tissue over vasculature, because that's where the ectopic expression is. This is all work that was done in Sarah Hake's lab. So we thought this was a cool kind of mutation to look at the effects of this mutation on pseudotime progression. Next slide, please. Okay, so what we're looking at here is a UMAP projection that Jack did of seedlings. This was the SAM plus 6-leaf primordia. So again, it's enriched for older stages of ontogeny, not for the shoot meristem or very young primordia. They don't survive this system. And where we see in the top, the expression of knotted shown in purple in wild type, and its expanded expression in lots of other cells in the NODD1 dominant mutant. And more importantly, we used this gene GA20 oxidase, which is a known target of narrow shoot transcription factors. So this gene is a known target. So this, the cells that are expressing or ectopically expressing GA20 oxidase, we know are actually having the effects of overexpression of NODD, the transcriptional effects of overexpression of NODD. So in wild type, on the left bottom, you can see those orange cells are expressing GA20 oxidase, and it's greatly expanded in the cells in the NODD mutant. So we use those cells, those orange cells, which are expressing GA20 oxidase, as the real markers for NODD1 ectopic function. And we said, okay, what does this do to pseudotime progression? Next slide, please. So we expected that if you overexpress in the wrong place at the wrong time, knotted 1, this marker of indeterminacy, that it would basically slow down pseudotime progression, but actually the opposite happens. It accelerates pseudotime progression. These cells that are expressing in the knotted mutant seem to go through pseudotime progression or ontogeny faster. It's accelerated. So that didn't make much sense. I first asked Jack if he got things swapped, but of course he didn't because he's really good. So we were kind of baffled for a while. What does this mean? Next slide, please. So basically we went back and we looked at the knotted mutation. And if you look on the right, those panels, upper and lower, are showing knots on the right there in the knotted mutant that are found in the leaf blade of a knotted 1O plant. And again, these are basically, it looks like sheath tissue is being expressed where it should just be blade tissue. And at the junction between this ectopic sheath caused by knotted expression, we think, and the normal blade, you get a ligule because ligules form at the boundary between blade and sheath in maize. And to show that this is actually sheath, what Josh did was Josh Strabel, he basically made double mutants of knotted 1O and a gene called ligulus 1, which removes the ligules from the blade sheath boundary. The boundary is there, but the ligule is not formed. And when this happens, the ligules at this and these knots are gone. So in normal mutant knotted plants, you have sheath plus ligule. And in these knotted 1O ligulus mutants, you just get knotted, suggesting this really is sheath. And Josh also looked at the lengths of the blade and sheath in knotted mutants versus wild type, and he found that sheath is actually overrepresented in these tissues. So this is good evidence that these knots in knotted 1O are ectopic sheath, which Sarah Hake has been saying now for over 30 years. Next slide, please. So we looked again at really old kind of in situ hybridizations and immunos of knotted 1 expression. And the mRNA is shown on the left. This is done by Dick and Hake many years ago. And on the right, there's an immuno using a knotted antibody. And you can see, especially the protein, is not absent from all regions of leaf primordia. It's turned off, the gene is turned off. On the left, you can see where the next leaf is going to be initiating this patch of loss of expression of knotted, and this pretty much stays off for most of the leaf on the left. But you can see at the very base of these primordia, shown by these arrows, we do see what looks like knotted protein coming up into the very base of these primordia. And so we've speculated for a long time, Sarah has for a very long time, we just published a paper that we think that these cells, which are expressing knotted in a wild type plant there, is where the sheath is being made, which would infer that in these primordia, the amount that's sheath identity is actually very, very small at this very early stage in development. Next slide, please. So yeah, and basically lots of fate maps done by Scott Pothig and colleagues indicated that sheath identity happens much, much later than blade identity in maize leaf ontogeny. So on the left here in wild type, you can see 2 different stages of leaf primordial development, a very, very young one on the left and a slightly older one on the right. Still a very young primordia, maybe P3, P4 stage, 3 or 4 leaves from the meristem. Young leaf primordia, according to Pothig, are almost completely composed of cells that will eventually become blade. This is all done by fate map. Almost no sheath identity. In a little bit later stage primordium, P2, P3, there seem to be some cells, but very few at the very base, that are fated to become sheath at maturity. And indeed, I've done some analyses which show that if you treat these seedlings with inhibitors of auxin transport, you get a fusion of the sheath up until the 4th leaf from the meristem, suggesting that, first of all, auxin is required for the sheath to separate from the other side, but also the entirety of the sheath is not fully out of the meristem or out of the stem in this case until pretty late in ontogeny, maybe P4 stage. So sheath happens late. So if that's the case, we have a knotted mutant where it's making a topic sheath, a later stage identity of leaf development. In the blade, this could explain why we get this acceleration of pseudotime progression, because it's making sheath, which should be made later, it's making it earlier in the knotted mutant. That's how we think what's happening there. Next slide, please. So I'm going to summarize this first part. I can't really see what time it is. Okay, good. Yeah. So we think that single-cell RNA-seq can provide great insight into gene expression, the landscape of gene expression in the shoot apical meristem. Next slide, please. It's kind of awkward with the. Yeah, we think that maybe stem cells express a lot of genes which prevent them from accumulating mutations, and they exhibit a slow cell division rate, which we think is a mechanism in these plants to keep the genetic load down in these cells, which will eventually, maybe later, really late in ontogeny, have to form germ cells, keep the mutation rate down in those germ cells. Again, we don't find any evidence for a Wuschel expression organizing center, a stem cell organizing center in B73. We don't think that there's good chance that other WOX genes are doing this, Wuschel-like genes in the shoot meristem. So what is actually making stem cells, organizing them in the meristem is still unknown. Next slide. Next one, please. Yeah, we think pseudotime progression is a huge advantage of single cell analysis. You can't do this kind of a study in laser microdissection where you're grabbing bulk tissue. You can't look at pseudotime progression. You can't use this to look at gene expression throughout ontogeny. And we think the ectopic knotted one actually accelerates rather than delays pseudotime progression, even though knotted one is a marker for stem cell indeterminacy or cell indeterminacy. Okay, I want to just quickly, next slide, talk to you about a project that we just started, also using single-cell RNA-seq, on an old mutant that I've been working on for a way long time called narrow-shoot. And this is basically shown in this series of slides, a series of pictures in this slide. The wild-type leaves are on the left, and then the mutant leaves are on the right. And you can see it causes a huge reduction in mediolateral growth of the leaf, of the blade, and also into the sheath, as you can see in these pictures. Very, very skinny leaves. It doesn't form this sheathing leaf face. On the far right panel, you can see on the left of that far right panel, looking at the node and the insertion point of the sheath, which wraps around the stem completely in a wild-type plant and covers the stem. You can't see it to form this sheathing leaf face, which is so common in many monocots. And then on the right, there's a narrow sheath where the sheath tissue doesn't come anywhere near to surrounding the stem, and you can see the stem in these plants. So it basically affects mediolateral outgrowth. Next slide, please. And this gene was cloned a long time ago in collaboration with Wolfgang Ware's lab, and it's basically encodes a Wuschel-like, or WOX, homobox transcription factor called WOX3. This is not one of the ones that we found in the shoot meristem proper in the single cell. That was WOX3A and WOX3B. The names are really horrific, but we call our genes narrowshoot1, narrowshoot2, but these are orthologs of WOX3 in Arabidopsis. And if you do a transverse section through the meristem, that's shown by that arrow on the left, you'll see the expression is in initiating leaf primordia in the first one from the meristem called the P1. And then later on, it's resolved to the very marginal tips of these leaf primordia, as you can probably, hopefully you can see at the edge of the P2 leaf primordium. So it's in the meristem, but really in the peripheral zone where the leaf is really starting to initiate, so it's not meristem proper. It's basically cells that are becoming leaf. And we think this expression somehow makes the leaves grow wide. Okay, next slide, please. So we just did some, a lot of cool work was done by former postdoc Phil Conklin. We just published this last summer. Basically, if you look at the expression on the left, you can see the mRNA and also the protein. We've got a nice antibody to this, to narrowshoot1. I should say the phenotype was caused by recessive mutations in duplicate factor genes, narrow sheath 1 and narrow sheath 2. They both encode pretty much identical WOX3 homeobox transcription factors, and they both seem to be expressed at the margins. So we use a probe for narrow sheath 1. It picks up both. But as you can see on the left, it really is localized in later leaf primordia to the very tips. Okay. And these don't grow out at all in narrow sheath mutants. The protein also localizes to the tips, shown by that white staining. So what Phil did in a nutshell was microdissected. This is bulk microdissection of the tips of leaf primordia from wild type, which is on the top in B here, and also from the very truncated blunt-edged margins of the P2 and also the P3, the second and third leaf from the meristem of narrow sheath, and look for genes which were bound by narrow sheath and modulated by this combination of ChIP-seq and laser microdissection RNA-seq. We found 52 genes, and most of these genes were repressed. The WOX genes are known to function mostly as transcription repressors. And I can't see what the bottom of that says, but I think it's something about ARC2. Next slide, please. Yeah, previous slide, sorry. So basically what we found, can you show the previous slide, please? I can't remove this toolbox from the bottom of the slide, so the bottom of my slides is blocked. But one of the genes that we found that was greatly transcriptionally repressed was 2 ARF2 paralogs, auxin response factor genes. There's 2 of these in maize, and in Arabidopsis, these were known to be basically repressors of leaf and other organ outgrowth. So it looks as if, we published this already, that narrow sheath, one of the ways it functions to make leaves grow wide is that it represses the expression of repressors of growth in the margins. And we think what its main job is, either directly or indirectly, to activate rampant cell division at these margins to make these leaves grow wide. So it represses a negative regulator of growth, which is a common theme, actually. Next slide, please. So we thought this was a great system to maybe look at the single cell level. We looked at gene expression in the previous analysis by laser microdissection, bulk tissue, the very tips of those leaf margins. But again, this is brand new data. There's only one replicate that Jack just found, and again, indeed, he found narrow sheath 1 expressing cells, not so many of them, but they were in a region of the epidermis, shown on the upper right. Those orange cells are all narrow sheath expressing cells, almost all localized to the epidermis. Said, okay, what other genes are differentially expressed in the very cells that are expressing this activator of cell growth, this repressors of cell growth? Narrow sheath 1. Next slide, please. So one of the things he's found was there's a lot of differential expression in these narrow sheath expressing cells, and at least one gene that promotes gibberellin catabolism, the breakdown of gibberellin, GA20 oxidase. So on the left is in situ that Jack did. It's hard to do this without a pointer, but in the very center, it's a transverse section. The meristem is in the center, and then it's surrounded by these overlapping leaf primordia. Do I have a? Yeah, right. And it's also expressed in the margins. But again, it's not just expressed in the very tips where narrow sheath is expressed. This gene is expressed throughout much greater regions of the leaf primordia, including the margins. And in narrow sheath, somehow we see expression in these younger leaf primordia. We suspect that this leaf, I can't do this, but maybe the second or third leaf from the meristem, you can see good expression in those margins. We think that expression is in the region, in the very tip of the leaf, in the blade, where narrow sheath doesn't seem to have a phenotype. But outside that region, if you look in the other leaf primordia, they're very, very blunt edged. They don't wrap around the apex like wild types do, and there's no expression. So the expression here is removed from regions where we don't really see narrow sheath expression. And again, no evidence that narrow sheath protein, at least in Arabidopsis, moves. Next slide, please. This is my last slide, and I'll end it up. But yeah, we found lots of examples where genes on the bottom row here, a number of them, we won't name them all, are expressing in rather wide regions of the leaf primordia, some closer to the margin tip but not exclusive to the margin tip, where that expression is either greatly reduced or not there in narrow sheath mutants on the top. What we're showing here on the bottom, narrow sheath 1, narrow sheath 2, these are plants which have just one wild type copy of one of these narrow sheath duplicate genes, and these plants look completely normal. Okay, these are redundant functions. So we see expressing, for example, this FFP1, this is a putative subtilase, a putative protease, expressing in the very tips of these margins. And on the far right, this Ralph-like is a putative ligand, which we suspect, we hope, might be actually interacting with this FPF1, but we're not sure. But again, they're both affected, and we're now making mutations in many of these genes to understand how we get this change in expression in regions which are, much, much greater than narrow sheath expression domain. Okay, and I think this is brand new work. This is from a couple of weeks ago, and Jack just actually collected the second and third reps of this narrow sheath, and we're going to be working on this for the next year or so to see what we get. So if anybody has any questions, I will take them. Thanks for listening.
Thanks, Mike, for the presentation. I think we do have a few questions here.
Okay.
Actually, I also posted the chat as well. So, first question is, are cells comprising the same susceptible to aggro infection? Are there any physiological differences or conditions that would alter the susceptibility to aggro infection?
I've never studied that. This is not when they do transformation, as you guys probably all know, of maize, they're using somatic embryos. There is a meristem there, but I'm not sure if that's the prime target. They make callus from these embryos, so I'm not sure if the meristem itself is actually infected by aggro. I've never done any kind of cell tissue culture experiments or aggros, so I'm not really sure.
Okay. Next question, I think you referred earlier to the work in Wolfgang Wehr's lab doing the expression of maize wuschel. Look like a pattern is different. The question is, does it suggest difference between how maize and Arabidopsis meristem are maintained or organized?
So, yeah, I'm not sure if those in situs published many years ago of wuschel in maize can be reproduced. Dave Jackson has a marker, RP marker of wuschel, and he doesn't see anything in the shoot meristem. He's published this, it's in the inflorescence meristem. And intriguingly, he also says he finds it very early on in the embryo, but in the shoot apical meristem, we don't see it. So those studies have not been reproduced. In rice, a similar situation occurs. It's not wuschel that seems to be the stem cell maintenance WOX gene. It looks like it's WOX4, which in other plants has been described as being involved in expressing the vascular meristems in the procambium. But in rice, like in maize, wuschel mutants do affect the inflorescence meristems. So again, in maize and rice, it doesn't look like wuschel is doing this job. In B70, I should say, in B73. We haven't looked in other species. Sorry, in bread lines.
Cool, thanks. So another question is, that's an interesting question. That's a pretty long question. I think the main point is there any indication of such application basically reduce transpiration by further understanding of shooter growth in maize?
Transpiration?
Yeah.
I never actually, I can admit I've never even thought about that. Sorry. But I guess there could be, you know, we tried overexpression of narrow sheath and we thought it would make the leaves wider, but boy, when we did this, it was by 35S. We know 35S doesn't express well in the meristem, but this was in the older primordia. Most of the time when we had these plants, we published this, about half the time we would see a phenotype. It was just a little bit of margin ruffling, a little bit of overgrowth at the very margins. And when we found those plants, we always found little patches of ectopic narrow sheath expression, which correlated with this overgrowth, this thickening growth. But it was very, very restricted. It wasn't like, even in leaf primordia, where we know 35S should be expressing, narrow sheath was not expressing throughout those leaf primordia. Something is really working to keep this gene localized. And there were a few escape events, we think, where there was some ectopic expression, and it caused this outgrowth and its proliferative ruffles, but we couldn't make the leaves wider. So something is really keeping us from overexpressing narrow sheath, and that's another area which we're really interested in. So maybe not a great way to increase transpiration. Maybe not.
So one more question here is, can NS phenotype, what is this phenotype, can be rescued by GA inhibitor treatment?
Yeah, we tried that. Great question. We tried that, and we found it was additive. So the leaves get a little bit wider, but narrow sheath doesn't make a normal margin, and it has this characteristic shape. And when we treat with GA inhibitors, both the wild type and the mutants get a little bit wider. The cells are longer, but we don't restore the lost kinds of tissues, the lost tissues that never differentiate in the mutant plant. And we made double mutants with narrow sheath and various GA biosynthetic mutants, and again, they look like they're additive phenotypes. So yeah, we don't think it's we don't think it's in the same pathway.
Okay, thanks. We have one more question here. Say, how far along the progression from SAM to germ cells to fit mapping study extended?
How far? Oh, you're talking about the early work that was done by Pothig and others, including myself, actually. Yeah, so they could tell by, they knew what stage and how many leaves were generated when they x-rayed, these were done by x-rays, when these x-ray induced sectors were induced, and they could tell by that staging and by where these sectors lay in the plant, relegated to one leaf or to several leaves, whether they were in the meristem or whether or not they were in leaf primordia and what stage. And so they were pretty accurately able to tell, yeah, at the early stage in leaf leaf initiation, those leaves are mostly all blade. They're almost all blade. And only a little bit later you get sheath. By counting the numbers of cells and what leaves were they, numbers of leaves in the seedlings, and what leaf number those sectors were on, they could tell particularly where those were when they were x-rayed.
Okay, probably you almost answered some of them already. As a follow-up question, say, do they link specific stem cell all the way to germ cell precursor in maize or other species?
Well, you know, there's a lot of somewhat controversial, again, fate mapping studies from over 30 years ago. And some of that data has been retracted. But, you know, if you can induce a sector, they did, when the plant was 2 weeks old or maybe a week old, it had made, you know, 2 or 3 leaves. And you can find meristematic sectors. We know they're meristematic sectors because they go over many, many leaves. And you can find sectors that will mark 4 or 5 or 10 or more leaves, then go into the tassel, mark the tassel, and then we'll mark actually the anthers and things. And so clearly those cells, before they were differentiated to become anther, they made lots of vegetative tissue. So they used that as evidence that there was no germline specification and perhaps no sequestration. The specification, I believe, I don't think we really understand about sequestration. Maybe some regions of the shoot meristem have very, very low cell division. And I think there's some evidence for this from Sobolewski's lab and others in Arabidopsis. And I suspect that's what's going on in terms of this.
Okay. Thank you, Mike, for the great presentation, a great answer to those questions. I think we are on time. Then I'll pass to my colleague, Larry, to introduce our next speaker.
All right, thank you, Yurong. Hello, everyone. And for those of you who weren't here in the morning, I'm Larry Gilbertson, a scientist at Bayer Crop Science, where I lead one of our teams that is working on gene editing called Applied Genome Modification. It's my great pleasure to introduce our next speaker. You know, we've had a lot of great talks already on shoot development, the one we just had, pollen biology, a couple talks on various forms of delivery, whether it's nanoparticles or agrobacterium or non-agrobacterium. And one of the reasons why we're also interested in that is because of how we might apply it to technologies such as gene editing. And so it's really great that our next speaker is a true expert in gene editing, is going to focus on some of the tools of gene editing. Dr. Yiping Qi received his PhD degree from University of Minnesota and then did a postdoc there working with the speaker who will come after him, Dr. Dan Voitas. And during that time, he made contributions to the development of plant genome editing systems based on zinc finger nucleases and talons. Dr. Qi started his own lab at East Carolina University in North Carolina in 2013 and stayed there until 2016. When he moved to University of Maryland as an associate professor in the Department of Plant Science and Landscape Architecture. In recent years, his lab has developed CRISPR-Cas9, Cas12a, and Cas12b systems for plant genome editing and transcriptional regulation. He's interested in developing genome engineering and synthetic biology tools and how they can be used to boost plant fundamental and translational research. Just as a reminder, if you have questions during the talk, go ahead and put them in the chat. I'll moderate the chat and we'll do a Q&A at the end. And with that, I'll turn it over to Dr. Yiping Qi.
Hello, can you all hear me and see my slide?
Yes.
All right. Well, thank you, Larry. Very nice for the introduction and inviting me. I've so far enjoyed so much with this symposium. So today, I'm going to talk about developing efficient CRISPR systems for multiplexed genome engineering in plants. So basically, my lab is really interested in plant genome engineering for both foundational and also translational research, where we would like to engineer tools which go into the genome and to modify genes for knockout or refine gene expression, or just really engineer metabolic pathways, sometimes for gain of function of endogenous genes. So over the past few years, with collaborators like Dan and Yong and Tao's group, and we have shown that we also developed CRISPR-Cas12a, formerly known as CPF1, or Cas12b, formerly known as C2 or C1 systems for genome editing in plants. So I don't really have to introduce you about CRISPR-Cas9 because it's well known. And then compared to Cas9, Cas12 systems are slightly different. Just a few aspects that I just quickly touch upon. So first of all, Cas12a and B systems, they actually recognize the T-rich PAM. So the PAM sequence is really the sequence recognized by the Cas protein. So when we talk about CRISPR-Cas genome editing, we often simply thought sometime about that targeting specificity was really contributed by guide RNA. It's actually contributed by both the guide RNA and also the Cas protein. So because these Cas12a or B protein recognize the triple T or double T PAM sequence first, and then lead to the guide RNA complementation of DNA. So second of all, these Cas12 nucleus, they make cleavage away from the PAM. And then unlike Cas9, Cas9 is only cut 3 nucleotides upstream of PAM. So where these nucleus cut away, which means if you only introduce very small change there, it's likely the guide gonna still recognize the modified DNA again for another cleavage. So there are potentially multiple rounds of cleavage, which potentially gonna accommodating like a homology repair or making larger deletions. So finally, Cas12 A and B proteins, they are making staggered end cutting, which again is unlike Cas9 because Cas9 make blunt end cutting. So all these different characteristics may kind of give these nucleus potential different applications in genome engineering. I don't really have time or want to get into detail because there are so many nice review papers in the field that we can all refer to. So one thing, after we demonstrate CRISPR-Cas12a and B genome editing for plants, another question is we want to know how specific are these nucleus, right? Because specificity is kind of a major problem for sort of human genetics or pharmaceutical applications, not less so for plant application, but still from regulatory perspective, we want to know how these nucleus are specific in the genome of plants. So working with many collaborators, we were able to investigate this problem specifically in maize and also in rice. And so for the maize work, I worked with Cam Wang group and Keith Jones group. And so what we have done in maize is really, because maize genome is huge, contain many transposons that are highly repetitive. So what we end up doing is using Keith Jones labs, the so-called CircuSeq in vitro system to digest genomic DNA with our CRISPR-Cas9 and Cas12a system and to identify putative off-target sites. And then we just did amplicon sequencing specifically to investigate those potential off-target sites, whether there are really indeed a mutation introduced or not. So the answer is actually not. We were sort of unable to find any of the in vitro identified off-target sites, find any evidence of off-targeting in those maize lines expressing CRISPR-Cas reagent. And similarly, we also produce a transgenic line in rice with CRISPR-Cas9 and also Cas12a. And because rice genome is rather small, and we can sort of with collaborator, we can sort of afford financially and also technically to sequence the genome and to really annotate the genome to see whether we can find the genome-wide off-target mutations. And again, the long story short, we actually sort of failed to identify any of the off-targeting mutations except only for one guide RNA, where we deliberately designed sort of as a messy guide RNA, which we know the guide RNA can mismatch to other genome sites, only that guide RNA resulting off-target mutation. So again, the take-home message here is Cas12a and Cas9 are both very specific in inducing a targeted mutation in the genome of plants. And then, well, based on the data from human cells, people have claimed that Cas12a may be even more specific than Cas9 in terms of targeting. But anyway, so after all this work, it's kind of encouraging because really that suggests that Cas12a is a nice alternative genome editing tool to Cas9 for genome editing in plants. So I have two stories to tell today. Both are kind of unpublished. We kind of just organizing to manuscripts. So the first story is Cas12a genome editing tools, continuing our Cas12a work. So this is done by a postdoc, Yingxiao Zhang. So there are three objectives for this project. First of all, Yingxiao wants to know whether we can identify Cas12 orthologs from different bacterial species or engineer Cas12a variants. Any of them can recognize a relaxed PAM because the typical canonical PAM is triple T, which is rather rigid for us to find many target sites. So the way we can relax PAM allow us to target many sites we wish to target in the gene or genome of interest. So second of all, Yingxiao wants to find, again, Cas12a orthologs or variants which can potentially work nicely at lower temperature for editing a plant genome. Because it is well known with our lab's work and along with other people's work done in human cells and also Drosophila, that Cas12a is actually, many of Cas12a also actually were very temperature sensitive. So they require higher temperature than 32 or 30 Celsius above to be highly active. So that's a question for us because for a lot of transgenic work or when we play tissue culture in plants, we are using really a lower temperature, like 25 Celsius degree, which is not really optimal temperature for Cas12a nucleus activity. So that's a second object. So last object for Yingxiao is after she, let's say, was able to successfully achieve both objectives, whether then she wants to apply the improved Cas12a nucleus for multiplexed genome editing in a plant, right? So because the way we can simultaneously editing many sites in the genome will be very, very powerful. And then the work is mostly done in rice, which we consider is a model plant and also it's a major crop. So for Yingxiao to look into Cas12a ortholog, should look into some earlier work and then identified a bunch of candidate Cas12a orthologs. Many of them were shown to be working in E. coli to recognize, actually, rather than triple T, they also recognize triple T, but their PAM actually are double T. Instead of TTTV, the V stand for ACG. And then Yingxiao found these a bunch of Cas12a ortholog recognized only TTV, right? Because even though you have one timing shot, that actually relaxed a lot of, giving us a lot of opportunity for targeting. So total she picked 9 of these Cas12a orthologs, and then use ALB Cas12a, which is really the standard, or I can say the best Cas12a nucleus in the market. It has been used for genome editing widely in human cells as well in plants, which is ALB Cas12a. So that was kind of like a positive control for us to benchmark our technology. And then here, what I showed you here is plotplast assay, right? Just expressing these reagents using this dual promoter expression system where both the Cas12a and also the guide RNA, CRISPR RNA, were expressed by maize ubiquitin-1 promoter. And then because Cas12a use only CRISPR RNA, which is only 43 nucleotide short of RNA, and it has to be precisely processed. So we use a double ribozyme system, HH and HDB ribozyme, flanking this CRISPR RNA sequence. So this is done early. We should earlier. This system is very efficient. So anyway, in the plotplast of rice, we found that surprisingly, nearly all these Cas12a orthologs showed activity, and some of them are comparable to ALB Cas12a. So which is very encouraging. And I just highlighted in the green window here, there are 4 Cas12a, ER, ALB5, and BSMB2. These 4 are really we picked on top of our list, even though some other additional Cas12a are pretty efficient as well. So the next question is, instead of doing whole genome sequencing to find how specific these ortholog nucleus is, and we just use an easier way to assess the targeting specificity of these top 4 Cas12a orthologs. So first of all, what Yingxiao has done is basically there's some permutation of making a systematic mismatch mutation of every 2 nucleotide in the protospacer. So the protospacer we use for Cas12a orthologs here is 23 nucleotide. So slightly longer than typical 20 nucleotide Cas9 guide RNA protospacer. Although I have to say, if you use only 20 nucleotide protospacer, it works just fine for the Cas12a orthologs. But anyway, if you look at the data here, again, these are all done in plotplast, and then many of the mutation kind of completely abolished activity of any of these Cas12a nucleus. So which suggests it was pretty specific. And Yingxiao again did a second experiment where systematically truncating the length of a protospacer from 23 nucleotide to 15. So as you can see, when the protospacer getting shorter and gradually it's losing activity. And especially when you reduce length below 19 nucleotide, and then the activity really drastically reduced for certain Cas12, like ER or BS and also MB2. So which again suggests the pretty good specificity for these ortholog Cas12a system. So the next what we want to really show is that we can whether we can get stable edit in transgenic line of rice. So again, we just only choose 2 target sequence. One is targeting OSDP1 with a triple T TTTC PAM. The second target is targeting another gene, rice OS EPF9 with triple T again and TTTG PAM. So again, we kind of did a different number of T0 transgenic lines and assessed 10 of these, well, I would say 9 of these Cas12a orthologs. And as you can see, they showed a different efficiency. Many of them showed quite efficient, almost close to the best Cas12a, which is ALB. And then this is also happened to for the second target. So again, this is kind of surprising, but it's kind of good because we kind of confirmed in rice, in fact, many of these Cas12a ortholog can mediating target mutagenesis in stable transgenic plants. And in fact, many of the mutation kind of more biallelic mutations. And then we, in fact, follow many of the mutant lines to next generation from T1 to T0, from T0 to T1 generation. And the genetic analysis suggests these mutations are indeed germline transmittable, can be inherited into next generation. So our InShot's purpose is really try to find that whether we can reduce the sort of PAM requirement, and the ability to identify so many Cas12a ortholog working in plants for genome is kind of exciting. But whether we can really improve that or making or discover better Cas12a, which recognize relaxed PAM, is really the key sort of discovery we hope to make. Hence, InShot targeted all these reagents to a second set of sites which has only double T PAMs. Here is a GTTG and another one is a GTTC, basically double T PAM. So rather than triple T. And not surprisingly, many of these Cas12a ortholog nucleus, they sort of work but poorly, right? This is kind of done in rice plotplast followed by NGS amplicon sequencing and analysis. And then interestingly, and we find only that MB2 Cas12a here and consistently showed across 2 different target sites and the best editing efficiency relatively compared to all remaining Cas12a ortholog, including the best Cas12a we are all using, ALB Cas12a. Again, this is not surprising. ALB Cas12a basically failed to edit efficiently at both only double T PAM sites. However, MB2 seems to be pretty good. So which is kind of very encouraging. And then InShot tried to target many other VTTV PAMs to see how general this MB2 Cas12a is able to edit these sites. And then here is the result showing you here on the lower left panel, where InShot targeted an additional 18 VTTV PAM sites. And then she found that 13 out of these 18 sites, actually, we were able to get more than 10% editing in plotplast of rice. So this is pretty good. I mean, with 10% editing in plotplast of rice, we can typically translate this efficiency to about 30–40% and above, basically, in stable transgenic rice line. So this suggests, looks like MB2 Cas12a indeed was able to edit many of the VTTV PAM sites. And again, we actually just picked 2 random sites, did a stable transformation into rice, and then we indeed find 80% out of 20% editing efficiency on these 2 double T VTTV PAM sites. So next InShot wants to look at is whether we can further sort of reduce the PAM requirement. And then this is based on one work we have done earlier. And a couple of years ago, we published a paper in Molecular Plant reporting that if we engineer a triple mutation to NLB Cas12a, and we were able to make this variant recognize relaxed PAM, which is basically TATG and TATA. And we pretty much reproduce that result again here. So for example, if we use the triple mutation here, basically the N to R, K to V, and the N to R again here, this for ALB, and then that variant can recognize the TATG, 2 TATG sites, independent sites, and also one of the TATA sites. And so that was for ALB. And then InShot basically harnessed the same, did the protein and structure comparison, and then harnessed the same mutation, install them into all 4 top Cas12a orthologs, and to assess how these variants perform against these relaxed PAM sites. And interestingly, and what she found is that actually almost all these Cas12a ortholog perform slightly better than the originally we published ALB Cas12a ortholog. Because if you look at this TATG PAM site, and all these 3 out of 4 worked better than, were as good as ALB. And at the TATA site, and again, all the 4 Cas12a variant worked at editing these 2 independent TATA PAM sites, but ALB only editing at one site with lower efficiency. What's more strikingly, and what we found is at the TATC site, we used to unable to sort of edit with the RVR, so-called RVR variant of ALB, and now with MB2 RVR variant, we were able to edit both TATC sites. And interestingly, other Cas12a orthologs also failed to edit the TATC site. So interesting again, because I showed you this slide here earlier that NB2 Cas12a is only one recognized VTT VPAM. Now with protein engineering, and Yingxiao was able to show actually the RVR variant of NB2 Cas12a was also superb, when compared to other Cas12a ortholog, whether the earlier ALB Cas12a ortholog variant we made or the newer variant we engineered here. Because it's only NB2 Cas12a RVR variant can edit across all these TATG, TATA, or TATC PAM sites, and hence they can recognize collectively TATV. So which is a pretty, again, very promising. And then Yingxiao went ahead to further introduce a fourth mutation. So this mutation is K625R mutation. And this mutation we've done earlier work with ALB Cas12a. And here, after install this fourth mutation to MB2, and so now we basically make this MB2 RVRR. So we call that a super variant because it's kind of interesting because the data really suggests it's kind of like a super variant you expect to have. And this super variant, we show that across many target sites, and we summarize the data here again in panel E here. And then not only this super variant retain its ability to target a very canonical triple T, TTT VPAM, it also retains the sort of wild type MB2 Cas12a's ability to target the relaxed VTT VPAM. And in addition, it retains the triple mutation, the RVR mutation ability to target a TAT VPAM. Now, the fourth mutation enable, on top of all these recognition, now enable to target additional PAMs, including TYCV, a Y equal to C or T, and also a CCCV or CTCV. So again, this drastically expand the targeting range of a Cas12a nucleus as we know. So now if we just do some computing of the genome in rice or maize or bread wheat here. So you can see here on this panel that the MB RVR super variant now can target more than twice as many as the sites offered by the targeting ability of canonical typical SP Cas9. Everybody, it's mainstream Cas9 everybody's using, right? So this really strikingly showing you how good this this variant of MB2 is in order to help us to expand the targeting scope in plants. And then the second question Yingxiao wants to find is, can we really identify any ortholog or variant which works sort of more robustly at lower temperature, let's say room temperature, for gene editing in plants? And then what Chef done here, I showed you here, is basically test MB2 Cas12a along with three other, right, the top four Cas12a at editing two independent canonical TTTV site at OSDP1 and also in the OSEPFL9 gene. And interestingly, what Yingxiao found is that only MB2 Cas12a, this is interesting, really Cas12a seems to be less sensitive to temperature. And because other Cas12a clearly showed a little kind of higher editing activity, but reduced drastically at lower temperature, like room temperature of 22 Celsius degree, right? But MB2 was not really showing much difference at both temperatures across both sites, which is kind of interesting because MB2 is also able to target a relaxed VTT VPAM, as I showed you earlier. And Yingxiao can also show that indeed it's actually at these two different temperatures, MB2 Cas12a seems again doesn't really seem to be much reduced activity at lower temperature. So we kind of we are still gonna think it's gonna be further testing needed in different species because this is only done in the protoplasts of rice. And but however, these data point to an interesting phenomenon that MB2 Cas12a appear to be less temperature sensitive when comparing to other Cas12a. So in that sense, so the mission to identify a Cas12a, which is relatively temperature resistant, sort of achieved. So the third aim for Yingxiao for the project is really to apply the Cas12a reagent and to conduct large-scale multiplex genome editing in plants. So, but before we were able to do that, we need to figure out which is really the best multiplexed genome editing system, right? Because there are some work has been done in different groups, yet there's no very comprehensive research has been done to really identify the best performing multiplex Cas12a system in any plant. So that really gave us a big motivation to assess this area. And to do that, InShell compared a dozen, basically 12 different idea or strategy to express multiplex Cas12a system. And I don't really want to get into detail. It just basically the idea is really we kind of fix the expression cassette of Cas12a because that's nothing too fancy here. We always use a POR2 promoter like maize ubiquitin 1 promoter, for example, to express Cas12a, and with a specified terminator. Here is a NOS terminator, for example. But for for the CRISPR RNA, because again, Cas12a utilizes very short CRISPR RNA, and then for CRISPR RNA expression or multiplexing, that's really a lot of strategy, and that's why we focus on how to multiplex many different CRISPR RNA. And then one idea, for example, is use a small RNA promoter like a U6 promoter, and or we can excuse me. So U6 promoter or just POR2 promoter, like again ubiquitin promoter to express that, and or we can use single transcript unit which means basically we use a single promoter to drive both Cas12a and the guide RNA without any, well, we just put some artificial poly sequence as a linker. We have shown earlier work that this strategy worked, and we want to further include this strategy for comparison. And then there's a lot of other ways to process the guide RNA. For example, I showed you earlier, we can use double ribozyme, HDV ribozyme, to process different guide RNA, just randomly erase them. Or we can use tRNA, because tRNA has been shown for processing guide RNA of CRISPR-Cas9. And tRNA also contains some internal promoter activity. And or we can use, interestingly, one thing I didn't mention is Cas12a nucleus. Interestingly, Cas12a nucleus also have RNase activity, which means that it can process its own sort of a CRISPR repeat of guide RNA, and hence can sort of piece them, use its own protein, and without any other additional mechanisms for processing. So we also use that idea, just using CRISPR array, just stacking different CRISPR guide RNA one after the other. And so basically, this is all 12 strategy meant to explore. And because there's so many constructs to transform to rice, we really don't think just worth effort. But to streamline the process, we did make all the construct and targeting 4 genes simultaneously. But we didn't really want to screen like 40 plants. That's too much work. We'd rather look at only like 4 plants, but only looking at like 4 independent plants already giving us immediate feedback. What we really discovered is actually anything showing any strategy showed a really good editing is really based upon a utilization of a POR2 promoter, like a ubiquitin promoter. And using U6 promoter or U3 promoter didn't really show very robust editing activity. So that's why we decided to focus only on the POR2 promoter. And for some construct, we included the POR3 promoter because just that's mainstream used, and we just want to include that for comparison. So this red window highlighted a 7 strategy, which is the one we mainly want to focus on for further comparison. To further compare that, again, InShell tried to use them to target 4 different genes in rice genome. And then there are 7 different strategies, and we just named them B, D, G, H, M, I, A, just different indication for different strategies. And as you can see for this figure in the middle, that clearly this B strategy, again, I highlighted the construct here for easy to see here. So again, this is based on using 2 different POR2 promoter, same POR2 promoter, but always use POR2 promoter to drive Cas12a as well to drive the guide RNA array here. And the guide RNA is being processed by the double ribozyme system. Okay, so this is system, long story short, we identified as most efficient multiplex system for Cas12a, which is again show you here. For example, here you have targeted we have targeted 4 different genes here, and then nearly every, again, InShell made 36 transgenic line. And Basically, every single transgenic plant line is edited at all 4 genes simultaneously. So in short, we get 100% multiplex genome editing. And that can be translated into another figure for easy view here. For example, Inshal analyzed what's the multiplex editing efficiency. Again, the B strategy is deeply kind of blue up top to 100% editing efficiency, the highest you can imagine you can get. And then Inshal also looked at what's the multiplex biallelic editing efficiency. And only because that's only for DP1, that somehow for one site you only get 70% of the biallelic editing. And that's why, so we reduced, but other than that, all other 3 genes are biallelic editing for every single lines. So this is really remarkably efficient. But the question is, this is done with only one, the sort of mainstream used ALB Cas12a, right? Because our group now have identified MB2 Cas12a, which showed a very relaxed targeting in the genome of plants. So we want to know whether MB2 Cas12a can be coupled with this multiplex editing strategy. And the answer is yes. And then Inshal basically just harness the same expression cassette and to express MB2 Cas12a rather than LB. And then this is showing you individual 11 line of transgenic line of rice. And as this is just showed you the empty sort of grid just shows you no editing like wild type. Half-filled grid just showing you one monoallelic editing, and because rice is the sort of diploid. And then the fully filled grid just shows you biallelic editing or homozygous editing. So as you can see, and across many different lines, in many lines showed you here, show the biallelic editing across all targeting genes. So this again sort of demonstrate that indeed this general multiplexing strategy works pretty well when paired with different Cas12a nucleus. So next, Inshal wants to sort of demonstrate some application of this efficient multiplexing systems. And so one thing she did is really try to target 6 sites on 5 genes in rice. And for the first 4 sites was actually targeting the promoter of sweet 11, 13, and 14 gene, because sweet genes are considered as a disease susceptibility gene because these genes were being manipulated by a tail effector of Zesomonas pathogen. And when Zesomonas pathogen infect plants, they secrete the tail effectors and different tail effectors like tail ABR-X7 or PTO-X03. These tail effectors are actually artificial DNA binding protein. They are actually transcription activators. They can activate host genes and to manipulating host metabolism. For example, they can manipulate these sweet genes because all these sweet genes include sucrose transporter. So they force plant to make more sucrose transporter to so shuffle the nutrient toward the intercellular space so that pathogen can really thrive in that space. So this is the pathogen, how they smartly harness the plant metabolism to really benefit them. But the way to engineer resistance against such pathogen is really just to go to the genome to mutate the cis elements or the binding sites of these effectors, and so that they cannot recognize and modify or manipulate, and so that we can really gain sort of a resistance, and that is kind of so-called bacterial blight resistance. So this is the idea. And the additional 2 genes Inshal targeted is actually a protein coding gene. One is GS3, a second gene is GW2. And if we mutate the GS3, we can enhance green size. If we mutate the GW2, we can enhance green widths and also green sort of weight. So again, we can make a sort of a larger and heavier grain. So the idea is try to use our multiplex Cas12a system to install 2 unrelated sort of traits. One is really disease resistant to bacterial blight. Another one is higher yield. So anyway, if you look at only 4 independent lines here, right? So as you can see on the target site, most of the target site are being mutated biallelically. So across 4 independent lines. So this again suggests very efficient. And indeed, if you look at closely, and the tail binding site are indeed being mutated. And so that suggests probably we have really installed a resistance against bacterial blight, even though we need to do some kind of like a pathogen test to fully confirm this. But I will be surprised if that's not the case. And similarly, we have achieved biallelic mutations on these 2 green yield genes. And our earlier work we already published with Cas9, and we show a genome editing of these genes can really enhance rice yield. I wouldn't be surprised, So the same can be achieved now with Cas12a. And one characteristic of Cas12a induced mutation is large deletions. I just want to highlight here. As you can see, many of the deletions are more than 10 nucleotide base pair, where Cas9 typically install 1 base pair insertion or deletion, and also up to 3 base pair deletions in the plant, such as in rice. So this is kind of interesting aspect for us. I think using Cas12a is better than Cas9 because inducing larger deletions can give us a better chance to modify or delete these tail effector binding site or any other cis regulator element we are interested in studying. So that could be a story, but Yingxiao come to talk with me during meeting time, say, whether we have enough, but, then we talk about, is there any, limit? Because when I go to meetings, there's people from the industrial side asking me always, how many sites is really the upper limit, right? How many sites we can really target in multiplex editing with Cas9 or Cas12a? So I try to be, little bold on that, but also want to be, conservative, try to get it to work. So I suggest just adding 10 more sites to 6. So then that's why Yingxiao ended up randomly picking 16 sites in rice genome. And as you can see, the sites has been depicted here, targeting 9 chromosomes of total of 12 chromosomes in rice. And then Yingxiao look at individual 21 individual lines and use the whole genome, well, not whole genome sequencing, but amplicon sequencing to look at all 16 target sites across all 21 lines. That is a lot of data. But after going through all the data, so she was able to analyze this way here. If you look at basically target 1, target 2 to target 16. So first of all, if you can see here, most of target give us very high efficiency of editing, like about 90% or higher, many cases 100% editing. Basically every single line we got, this target has been edited. And in many cases it's biallylically edited here, right? So it's pretty efficient. And for some guide RNA like this target 2, this guide RNA didn't work well. So that just suggests maybe the poor guide RNA design. And if we, because other guide RNA works very well. And then if you look at below this table here, we just showed you how many across individual lines, how many target has been simultaneously edited. So I think the statistics also pretty impressive because across different transgenic lines, we can find that pretty much, you will find that 14 or more target has been simultaneously edited in this, transgenic plant. For example, I only highlighted transgenic plant number 2 because in this line, every target, all 16 target has been edited. And in fact, only 1 target is the monoallelic edit. And then all remaining 15 targets have been edited biallylically. So I think, again, this suggests pretty high efficiency of multiplex editing with our systems. So because the technology, our lab is developing all the tools. We want to make sure the researchers can, not really be overwhelmed, rather find it's kind of straightforward to use the technology. And then we made all our vectors deployed to AgConnect. And we also, Yingxiao, also put in supplementary of data, like how to assemble all the, the toolkit to use them. And then there's other system I don't have time to get into because I only introduced the most efficient multiplex genome editing systems. But in our toolkit, we actually have also gene regulatory aspect in that for gene repression. And although the efficiency wasn't very impressive, I would say there's more room to improve for Cas12a based gene repression. So, but anyway, that's kind of concluded the first story. And I'm going to use remaining time to really shift gear to a second story about the CRISPR Act 3.0 for gene activation. Because earlier work, I really showed you how can we knock out genes or modify promoters for that. But here we're talking about how can we achieve gain of function. So this is done by another second postdoc, Changtian Pan. So the objective for this project is really 4 here. So number 1, Changtian wants to improve CRISPR activation systems, and then he kind of wants to make sure it's really efficient because he wants to assess that system by only using 1 guide RNA to target for 1 gene. So this kind of 1-to-1 relationship allows us to spare more room for multiplex genome, activation, right? So if we can use 6 guide RNA, then we maybe really be able to activate 6 genes simultaneously. That's the idea. So then, of course, we want to do, again, this is for multiplex gene activation. So hopefully we can demonstrate metabolic engineering, but that may be sometimes too naive, think about, because we maybe overlooked the complex post-transcriptional regulation in the cell. But nevertheless, the tool has been developed, then other people can use them for metabolic engineering or, really to perturb transcription network in the plant. So finally, again, we are sort of developing tools. We want to make sure the tool sort of easy to use. So just a little background, because we have done actually this kind of work earlier. So in fact, we have developed two sort of CRISPR activation systems. And based on, this is really based on, for the audience who are less familiar about this, basically for CRISPR-A, it just means CRISPR activation. And then, which is really a very kind of simple, interesting idea, where we can introduce two mutations and to sort of destroy the nucleus activity of Cas9. So now you become, you have dCas9. And then now you can fuse a transferrin activator like VP64. And this fusion protein now can steer a complex with guide RNA to target a promoter sequence in this case, right? And then this promoter, then this activator now is able to recruit the transcription machinery to overexpress the gene downstream for gene activation. So for example, when we make this VP64 fused to dCas9, we can only find really a few fold activation of our target when we make transgenic lines in Arabidopsis, and not very efficient. And then about 2 years ago, we published a paper that further improved that. We named that the CRISPR Act 2.0, where we still have the VP64 fused with dead Cas9. And however, we further engineer the guide RNA. We make guide RNA 2.0, where you have two stem loop with basically with MS2 stem loop, which can accommodate binding to the MCP RNA binding protein. So if we fuse VP64 to the MCP RNA binding protein, and then they can really be recruited to this stem loop. And then because MCP MS2 binding is, they form a dimer first to bind a single MS2 stem loop. So you actually having two stem loop, you potentially can really recruit 4 individual VP60 activators. So this indeed really helped us to achieve much more sort of higher fold activation for targeted genes in Arabidopsis. So for example, this gene, we only were able to achieve about up to 7-fold with the first generation, with second generation CRISPR 2.0 here. So we can achieve 40% activation. And for this FIS2 gene, so don't be fooled by this 1,000 level, activation. This can be a little bit cheating here, because this FIS2 gene is interesting. It is just really an imprinter gene, which means in rosette leaves, in mature rosette leaves, and this gene is pretty much silenced. So you can consider it's a pretty much silenced gene. That's why we can actually get so much higher activation with our activators. Anyway, so this is kind of the state of art we have, and a few years ago. But we recognize that still will be room to improve these activation systems. And we believe only when we are able to improve that, then the system will be more robust, that can be widely used by the community. So that's kind of the motivation for us to do that. And Tang Tian, in fact, tried many different ideas to really fuse a lot of different activators to Cas9, because the beauty of this is really you can think about engineering of focus on either Cas9 fusion or focus on the guide RNA, right, or both. So this is literally what Tang Tian has done. So in the end, just long story short, what he kind of found is actually there's a lot of potential just to play with the guide RNA scaffold rather than the Cas9. So for example, we end up having this CRISPR Act 3.0 system now, this platform. This is really mostly based on our guide RNA 2.0, which contain 2 MS2 stem loop installed in the middle of the guide RNA. And then because we have this stem loop optimal here, and then that can accommodate the binding of MCP RNA binding protein, and then we can fuse something to the MCP. One thing worked pretty well is the SUNTAG technology published earlier by multiple groups. So the SUNTAG is basically a sort of a tandem array of GCN4 repeats peptide. And for example, if you have 10 repeats, and here just indicated here, and then each repeater can be bound by a single chain sort of antibody, which is SCFV here. And so then this single chain antibody, again, each of them binding to 1 repeat. If you have 10 repeats, again, you can have 10, these antibody bind to these 10 repeats. If you fuse our transcription activator, right, to this SCFV single chain antibody, and then all the transcription activator will be recruited, right? So you can end up having a many sort of activator being sort of recruited sort of by a single CRISPR complex here. So this is the idea. So this is the sort of we kind of harnessed the SUNTAG idea to, because the SUNTAG idea was previously demonstrated only by fusion, just repeated to the Cas9, to the C terminus typically. But instead of doing that, what Changtian found is actually fuse SUNTAG to the MCP and then allow that to be recruited by the guide RNA scaffolder is more efficient because potentially we can use 2 scaffolder to stem loop to recruit 4 SUNTAG chain, and which really allow us to amplify the signal. So this is the idea. Again, this is, and in fact, we didn't really find that having a additional VP64, helped at all. We can remove this VP64 from fusion of Cas9. We just kept there because it didn't really help much. The most activation power is coming from this system, the SUNTAG system recruited by the guide RNA. Anyway, so this is how Changtian found the most efficient platform. But still, he need to improve or test a lot of parameters. And one thing he kind of become very ambitious is he doesn't believe that this guide RNA having 2 stem loop is better. And he said, what about we have 8, right? Can we really have many more? The activator being recruited by having 8 MS2 stem loop here. This is he tried this called a series 8 times MS2 scaffold. And then to test that, he used activator, which is the VP64, which is the one we used for our CRISPR Act 2.0 system. And then what he found here is actually, interestingly, more stem loop doesn't mean better, because in the end, it is still the guide RNA 2.0 showed the more robust activation across at 2 different target genes here in rice. This is assessing protoplast of rice. And this guide RNA MS8 MS2 didn't really work well. So we reasoned that could be a lot of structure hindrance and impacting the efficiency of this system. So that's why we kind of in the end we end up still using this guide RNA 2.0 have 2 stem loop. And then he also compared having a 4 repeat versus 10 repeat, and it appears that having 10 repeats seem to be slightly better than having 4 repeat, and that's why we kind of end up having 10 repeats in future work. And we also compare this system to our CRISPR Act 2.0. As you can see, our CRISPR Act 3.0 drastically outperforming the 2.0 system a lot. So anyway, so the next one is Changtian doesn't believe this VP64 is really the best activator after all. There are so many other activators we can choose. And then Changtian decided to compare, right? So he compared by adding additional 4 different activators, including a 2 times tail activation domain from the Zasamonas tail effect proteins, and also then 2 times tail further fused by 1 molecular VP64, and also a TV, which is earlier published another 2nd generation activation system using 6 tail repeat and a VP64 fusion. And finally, VPR is basically a chimera of 3 different transferrin activators earlier being shown in human cells, resulting very robust gene activation. So anyway, so Changtian tried to recruit all these 4 in addition to VP64 to, again, he wants to finally compare the 4 repeats versus 10 repeats. So interestingly, what he found is actually the best performing system is actually having 2 tail repeat as a transferrin activator, coupled with a guide RNA, again, 2.0 system to recruit the SUNTAG system with 10 repeats. So this system is resulting 200 over 200 activation, fold activation, very high for this gene. And this is system we really think is the best among all. And then we need to benchmark this CRISPR activation 3.0 system. And in addition to our 2.0 system, there are additional 3 at least a 2nd generation activation systems. One is use SUNTAG system, again, like I said earlier, should to work well in human cells, and then later also being shown in plant works very well for gene activation, and also the TV system, and also both of them are rely on a fusion of activator to the dCas9 protein. And then this dCas EV 2.0 system recruited ED area by dCas9 and recruit VPR system using a guide RNA scaffold called 2.1, which also have 2 MS2 stem loop. So to benchmark our CRISPR 3.0, we compared all these systems together, and nicely, what we were able to show here is our system outperform all these second generation systems very much, actually by a big margin. And in fact, the best system in our hand is still for the second generation is the DCAS and TV system. Indeed, is pretty good. And that was published a few years back by Jennifer Lee's group in Nature Plant. So our system indeed very active very well. And in fact, we even tried to use a port ubiquitin promoter to drive the guide RNA. And we also used just a typical USLE promoter to drive. We found a very compatible activity for gene activation. But you would ask me, okay, all the work you have showed me is actually at mRNA level, right? So you have gene activation, and then mRNA to protein, that's another level called a translation. So how much we know that's, more RNA can be, fully, we are fully convinced it can be translating to more protein. That's a very good question. So to at least to assess that aspect, and what Changtian has done is he make these two independent reporter system where he has fused a promoter which we kind of empirically validated as a lower activity promoter, and then put that promoter to drive the mCherry expression. And then he only used a single guide RNA, only one, because we reasoned that if the system is robust, one guide RNA would be good enough. And to express, then that would be allowed us to activate the gene expression. And indeed, that's the case in both cases. And we were able to activate the gene expression. As you can see, you have light up red color of mCherry cells. And that suggests indeed the activation is translating to protein level. And then Changtian made this multiplexed system for other people to use. And this system he specifically want to investigate is actually use tRNA to process the guide RNA. And earlier we have published a multiplex system just by really using Golden Gate cloning to putting together the guide RNA cassette with individual promoter and terminator. And Changtian compared this tRNA system with this conventional multiplexing system. And he showed actually this tRNA system slightly outperformed this conventional multiplexing system, and almost as good as you try to activate the gene one at a time. So then we think this tRNA system works pretty well. And here he should only just prove concept to activate 3 genes simultaneously. But all the work is done in rice. So we want to show whether we can achieve that for Arabidopsis, which is a dicot model. Rice is a monocot model. So then Changtian basically did a multiplex editing construct here, just randomly designed 2 guide RNA for activation of FT gene, a floral gene in rice, in Arabidopsis, and the 2 guide RNA to activate the TCL1 gene. So then because FT activation can give us early flowering, very easy phenotype to score. Indeed, that's all happening for the transgenic line. Very clear early flowering, and in fact, you can quantify the number of the rosette leaves and to show clear early flower phenotype. And what's really being remarkable is that from seed to flowering has been reduced almost a month. That's how we can reduce the life cycle of this plant from 2 months to nearly almost a month. So this really reduced almost the Arabidopsis life cycle to half. And then that is not surprisingly, it's because the gene has been highly activated. We have achieved over 100-fold activation of FT across different transgenic lines. And importantly, that phenotype has been genetically stable in next generation of T2. And then we also look, because we also activate second gene, which is basically TCL1, and we actually only activate this gene only a few fold. We were a little bit disappointed, but as you know, not every single gene is really regulated in the same mode. That's a kind of black box for us to understand how much the gene regulatory feedback has been installed for individual endogenous genes in the plant. And then we don't know how much at the transcription or post-transcription level, right? So, but nevertheless, with careful study, Changtian actually were able to show that even with a few fold activation for this gene, we can actually observe the phenotype, anticipated phenotype, which is reduce the trichome number on the surface of the leaf. And this is a statistic showing indeed as a small sort of reduction of the number of the trichomes. And importantly, all these phenotypes can be really transmitted to generation to generation. This is again showing you another generation of T3, clearly for early flowering and clearly reduce the trichome number. And so again, this is a kind of like suggests that all the components of CRISPR Act 3.0 are really stable. Nothing gets silenced across different generations. So finally, because we also assessed many different guide RNA, and we kind of have a lot of data, we can kind of try to have a look. Is there any clue for us to design a guide RNA allow us to achieve efficient gene activation? And Changtian looked at that, and it looks like if we target design guide RNA to target non-coding strand of the DNA, and then between anywhere from the 40 to 180 sort of a nucleotide ahead of this transferring start site, and we can achieve we have higher chance of having a very high level of activation. And for the GC content, well, kind of you need to balance the GC content that looks like from anywhere from 45% to 65% GC content to achieve more robust activation. So though the data set is kind of small, and we envision we need to do a lot of this kind of have a lot of data to maybe to further refine this sort of guide RNA design rule, which will be very useful for the community to use, and then coupled with our activation system to really apply them for their interesting research. So with that, I would like to sort of summarize what I have just introduced, two aspects of the research. So first part, I introduced to you about Cas12a, a new Cas12a genome editing tool, allows us to really achieve multiplex loss of function. So I wouldn't necessarily say loss of function because I also showed you promoter editing or promoter sort of, which is kind of allow us to refine gene expression as well. So second part of the work is I kind of introduced a new multiplex gain of function for genome engineering. So now we can not only sort of generating loss of function, we can also generating robustly gain of function. So which is kind of powerful, I think, for basic science as well as engineering. So finally, I would like to thank all my teams. And so team members here, and particularly, again, the talented postdoc, Inshao, and worked with Cas12a project with some support from a collaborator, Yong Zhang's group, and then Chang Tianpan for the activation system with some support of collaborator with Patrick Shee's lab at UC Davis, and very much appreciated the external funding as well as the startup funding from UMD. I'll stop here. I have around 2 minutes for questions. Thank you.
All right, thank you, Yiping. That was really great. Man, that CRISPR-A is pretty impressive. All right, let's see if we can get in one, maybe two quick questions here. One question was, are you planning to test it? So I guess most of your editing was in genes. Are you planning to, or have you tested in intergenic regions? And I guess related to that, have you seen chromatin effects on editing frequency?
Yeah, that's a very good question. I think, actually, I don't really have a defined answer for you, but based on our limited data set, and I think we didn't find a clear chromatin effect, even though there's data from mammalians, from other plant researchers showed a little bit of chromatin effect because people have fused with a sort of a chromatin relaxing peptide and opening chromatin to Cas9, for example. They were able to show that in some sites you seem to be observed the higher editing efficiency. That indicating if you open up chromatin, and then actually that helps. But our work actually didn't really see that closed chromatin, at least I don't think that's prevent us from targeting, right? Because we have targeted closed chromatin, like silence the mesolytic region, we can actually activate gene expression with our dead Cas9, which kind of suggests that we can get access to those regions, even though the access may be not most efficient. So, but that's a very good question.
All right, yeah, thanks. You would think intuitively open chromatin would be easier, just not a lot of evidence, at least in plant cells. It was very impressive about the multiplexing and especially for expressing the guide RNA. So one of the questions, let me see if I can find it. What do you think is the mechanism for why the POL2 transcript approach to guide RNA production works so much better? What's the mechanism compared to POL3? Well,
yeah, I think one main contributor is the POL2 promoter is evolved to drive along a sort of transcript. And POL3 is really, when you do multiplexing, you actually need adding the length, of the guide of the transcript. I think if you only express one guide, maybe POL2, POL3 are very equivalent. But when you try to do more, you quickly lose, POL3 promoter meant to express only short guide RNA being outperformed by the POL2 promoter because it meant to drive long guide RNA. That's one aspect. Yeah.
Yeah, no, that's a good point. Kind of related to that, so Mark Johnson asked, it looks like the very successful B strategy for 12A multiplexing utilizes independent POL3 promoters and a ribozyme to terminate each CRISPR RNA. What's the advantage of a ribozyme over just a poly A cleavage site?
Yeah, so for the ribozyme, what we found is at least based on all the data we have, is the ribozyme indeed helps us to generate very clean guide RNA, mature CRISPR RNA. And but if we don't have ribozyme, earlier we have shown it didn't really get very efficient editing, And even the other people report without using ribozyme, but we always use ribozyme, and many other people also followed up with ribozyme good editing. So yeah, but that's a lot of different ways of doing that, you know. So yeah.
All right, so I think we're going to end there. There is one more question that just got posted. You can go in and answer that yourself in the chat, but we are going to take a break now. So this will be our last break of the day. Please come back. We have one more great talk coming up from Dr. Dan Boita, so please come back in 10 minutes. That would be 3 o'clock St. Louis time, and we'll see you then. Thanks, everybody.
And then Edward, it's over to you then.
Right. All right, it's my pleasure to introduce our final speaker of our symposium today, Dr. Dan Boydis. Appreciate everyone that has joined us today, and we got the one final presentation here this afternoon. And then a reminder that the talks will be available after the event with information posted. But Dr. Boydis is a professor of in the Department of Genetics and Cell Biology and Development, and also the director for the Center for Precision Plant Genomics at the University of Minnesota. Prior to joining the University of Minnesota in 2008, Dr. Boydis was a professor at Iowa State University, and his research focuses on optimizing methods for efficiently making targeted genome modifications in a variety of plant species, which helps advance basic biology and develop new crop varieties. Many of us know that in addition to his position at the University of Minnesota, Dr. Boydis co-founded Calyx, which is an ag biotech company that uses gene editing for crop improvement. And I believe, maybe we'll hear from you, Dan, has also had a successful commercial launch of some edited soybeans. Dr. Boydis is also a fellow of the American Association for the Advancement of Science, and in 2019, he was elected to the National Academy of Sciences. So without further ado, I'll turn it over to you, Dan.
Thank you very much, Edward. It's certainly a pleasure to be here today. It's been a great symposium. I think you guys really picked a nice group of topics which, you know, very nicely overlap a little bit and really tell collectively a great story. I'm a little nervous to follow my former postdoc, Yu Ping. He just did an outstanding job. He was a great postdoc, and it's really nice to see all the beautiful work he's done as an independent investigator. So Yu Ping, I have data envy going into this talk. Today, and I'm going to talk about work we've been doing for the past 3 to 5 years on what I refer to as overcoming the bottlenecks in editing plant genomes. I mean, it's more than a decade that the powerful tools such as TALENs and CRISPR-Cas systems have come to the fore. And of course, they allowed us to go to the sites in the genome we want to modify, typically introduce a chromosome break and create mutations and targeted sequence modifications. But I still think that the opportunity provided by this technology has not been realized. We have challenges to overcome in using these reagents at scale and, you know, really much more efficiently so that we can create the types of modifications that we'd like to in plant genomes with much less effort and trouble. So the next slide shows sort of this diagram was published in a paper by Freddie Altpeter, where he spearheaded the effort. It really came out of a symposium that was organized by the National Science Foundation. And, you know, NSF realized, you know, on the left-hand side that they and others, both public and private enterprises, have made a lot of investment in plant science, mostly focused on what I would call genome reading, determining genome sequences, transcriptomes, metabolomes, and all of that knowledge has been mined and hypotheses generated. But in order to test those hypotheses and to use that information for crop improvement, we have to go through these bottlenecks. Typically, you know, if we want to create a transgenic plant or a gene-edited plant, we have to deliver the reagents efficiently to the cells, and then we have to coax those transgenic or edited cells back into whole plants. And so clearly, an outcome of this meeting was that we still have bottlenecks to overcome, and I truly believe this is the case in order to fully realize the technology and sort of match it with the investment we've made in the systems biology, our collective systems biology's efforts. So, you know, I came back from that meeting and I told the students in my lab that we wanted to come up with ways to edit plant genomes that are different. So if you go to the next slide, you know, we've really been delivering reagents to plant cells, and much at using agrobacterium or particle bombardment. We heard some nice examples this morning of novel delivery paths with nanoparticles and with other bacterial strains, and those seem very promising. But for the most part, we still use agrobacterium and particle guns. We get our reagents into the cell to create a transgenic cell, typically, or transiently express our gene editing reagents that then create the targeted modification. But we have to coax that cell back into a clump of dedifferentiated tissue and then add hormones over months and weeks to tease back a shoot and a root and ultimately a plant. And, we've been doing this really by and large the same way for, 30 plus years. And so it's a little, frustrating that we haven't made sort of breakthrough advances in the way that we genetically modify plants. And so I challenge my students to just say, let's minimize tissue culture as much as possible, think about novel delivery methods, and see if we can come up with different ways to ultimately edit plant genomes. So the next slide is the first story I'm going to tell today, which is a study that we published the framework for in Nature Biotech last year. And it builds upon some of the themes we've heard in the story, particularly Mike Scanlon's talk on the meristem. We use developmental regulators to induce meristems and accelerate plant gene editing. So the next slide just is a simple depiction of dicot meristem. We heard a lot about the monocot meristems with maize as the model, but similarly, it's the stem cell niche that gives rise to all the above ground organs, leaves, and flowers. And so over the years, we've come to appreciate in dicots that, and we've heard again about many of the regulators of monocot meristem maintenance and induction. But the next slide shows just a few examples of research that has been carried out wherein specific regulators of meristem patterning and identity have been described. So again, you've heard about Wuschel, which is one of the key regulators of meristem identity and fate. It works together with other regulators, transcription factors like clavata3, and these in turn are of course influenced by hormones like plant cytokinins. And others have shown that if you ectopically express some of these transcription factors, or IPT, which is isopentenyl transferase, it's a gene that synthesizes cytokinin. So if you ectopically express in differentiated somatic cells these developmental regulators or genes that produce hormones, you can create these meristematic like tissues. And so my two graduate students sort of outlined an approach, which is described in the next slide, in which they wanted to accelerate plant gene editing and by avoiding tissue culture. And they reasoned if they could come up with a combination of developmental regulators that would induce a meristem and co-deliver gene editing reagents, they should be able to induce an edited meristem. And of course, as I said, the meristem is going to give rise to leaves and flowers. And ideally, then you could just harvest seed that has your gene edit of interest. That was the ambition. So one of my students, Ryan, realized that we really need to figure out which combination of regulators we need to deliver and how to express them. So the next slide shows a few of the types of constructs he tried. The top construct includes a luciferase reporter, so we could see which cells received reagents. And then he would try 2 regulators at a time in various combinations and under either weak, like NOS promoters, or strong, like 35S promoters. And he wanted a quick system in which to test different combinations and expression strategies to see which are most likely to induce meristems. So he decided to deliver his reagents to seedlings. Actually, if you could skip 2 slides, it shows the overall strategy that he used. So we would take 6-well plates and germinate seedlings and then treat them with agrobacterium that carried the constructs I just described. And then he would co-cultivate the seedlings with the agrobacterium. And if you click once more, you can see that some of the seedlings in the Petri dish, of course, would take up the reagents and express the luciferase reporter. And so we could get an idea of the extent of transgene delivery and expression that he was able to achieve. So these initial experiments were all performed with Nicotiana benthamiana, one of our favorite models. And if you look at the next slide, over time, he could follow these sites to which his constructs were delivered, again, by monitoring luciferase expression. And then, as you see in the bottom slides, a few 3 weeks after reagent delivery, he could start to see these growths, these dark green masses occur, and what looks to be like leaf primordium. Then if you go to the next slide, if you wait a little bit longer, some of these cells that have received the reagents actually form small shoots. So the white, what you see or looking at here, again, these are seedlings in a liquid culture, so the cotyledons are a little bit distorted by that growth environment, but the arrows point to shoots that have emerged from the cotyledons. And these shoots could be excised and rooted. And the next slide shows you a couple examples of plants that have been recovered from this approach. You can see the top plant, if you're familiar with Nicotiana benthamiana, it's a little bit developmentally distorted. That is, it doesn't have the normal phenotype. And actually, if you harvest leaves from this tissue and look for luciferase expression, you can see that you've actually created a transgenic plant. So the regulators went in, the luciferase construct along with them, and that transformed cell was induced to form a meristem in a transgenic plant. At the bottom, the plant looks more phenotypically normal, and you can see 4-5 does not express luciferase. And so here what we believe happened is reagents went into cells, and since many of them are can move from cell to cell, they were expressed in some cells, moved into non-transformed cells, and those non-transformed cells were then induced to form a meristem. So you could create transgenic and non-transgenic shoots by this approach. So if you take plant number 2, for example, a transgenic plant, and go to the next slide, you grow it up. Actually, these plants would express or would form flowers that were fertile, and you could harvest seed from these plants. There's a little 6-well Petri dish with glowing seedlings in it. So the transgene was transmitted to the next generation. And in the graph just shows a few examples of transgenic plants that were created and the frequency with which the loop positive, in which the loop construct was transmitted to the next generation. So clearly you can make heritable transgenics by this approach. So the system allowed Ryan to really kind of figure out what are the best combination of regulators to add to these seedlings. So the next slide shows really kind of a summary of some of those results. You can see in black that he started really with not that many seedlings, 30 to 35 or so seedlings in Petri dishes. And then he was monitored growths, shoot-like growths, and full plants that were formed. It's illustrated in the dark blue. And you can see the assay was simple and robust enough for him to quickly identify some combinations of regulators that worked pretty well, shoot, bushel, and IPT, for example. And you can see that 30 to 35 seedlings were treated, and we recovered about 10 or so fully newly formed plants. So it was really quite robust. So with this framework, the next slide shows the bottom construct shows what he then used in subsequent experiments. So now he'd identified his favorite combinations of developmental regulators and promoters to express them with, and then he co-delivered instead of luciferase, Cas9 and a guide RNA. This guide is targeting phytoene desaturase, which is one of our favorite targets. It's a gene involved in carotenoid biosynthesis, and if you inactivate all copies of phytoene desaturase, you fail to make photoprotective carotenoids, and so the tissue tends to photobleach. And so we can if we have efficient gene editing, we can see a nice phenotype of these white sectors. So in the next slide, of course, he was anxious to see whether or not he could obtain gene-edited shoots. So very early on, as I mentioned, you can see in the red circle, these growths formed, and you would harvest these growths, couldn't quite wait for shoots to form, and assess whether or not gene editing was occurring in those cells. And the gel-based assay shown at the right is quite straightforward. It turns out that Cas9 is cutting at a restriction enzyme site. And so if you see the upper fragment, after treatment with the restriction enzyme, it's undigested. And that suggests that you've mutated that restriction enzyme site. And then at the bottom, you can actually, of course, just sequence these amplicons that amplify up the target site and see that, a significant fraction, in this one example, 42.2% of the reads have mutations in the target site. So the growths that were forming were indeed transgenic. The next slide shows some plants that recovered. And here we're just harvesting leaf tissue, amplifying the target site and subjecting it to next generation sequencing. And you can see, 4.9, 18.3, 11.3. The purple denotes the number of, or the fraction of sequence reads that have mutations at the target site. So these plants are indeed chimeric for, they're mutagenized, but they're chimeric. And so the question was, can we transmit mutations to the next generation? So this is illustrated on the next slide. If you look at the right picture, these are some progeny from those plants, those chimeric plants. So some are on the right, fully wild type in green. Some, or as the white seedlings indicate, completely lack the phytoene desaturase activity. And so Nicotiana benthamiana actually has 2 phytoene desaturase genes, and in order to get this phenotype, you have to inactivate both copies of both genes. So biallelic mutations in 2 loci. And indeed, analysis of the target sites indicated that phenotype and genotype were indeed matched. In the middle, we have some heterozygous plants that are still expressing the targeting reagents, and you can see we're creating chimeric plants again, because in this next generation, mutagenesis is ongoing. So I showed you a few slides ago that some of the shoots that emerged, they were green shoots, some were developmentally distorted, but the slide on the left, or the picture on the left, actually shows a shoot that emerged that was completely albino. So even before organogenesis, both alleles of both copies of phytoene desaturase had been inactivated. And of course, this shoot could not develop further because it was photosynthetically incompetent. So, of course, we wanted to move outside of the Benthi model. The next slide shows next few slides show some of our efforts in tomato. We could do the same approach, grow tomato seedlings in culture, add different combinations of our regulators, our developmental regulators, and determine which combinations are best for inducing shoots. Panels C and D show meristem-like structures that are emerging in C, and if you look at luciferase activity, you can see that they're luciferase positive in D, so we could create transgenic shoots through this approach. And the next slide shows that we could also create gene-edited shoots. So here we're targeting a different gene involved in carotenoid biosynthesis, Cy1. This gene makes carotenoids in the fruit, and if it's inactivated, you end up with yellow fruits instead of red. And so he created shoots with biallelic modifications in Cy1, in which he had, a -1 and a -71 base pair deletions in each of the 2 alleles. And you could show then in the progeny that these were inherited in approximately a Mendelian fashion. So the approach is working well now in tomato. We've done a number of other gene edits in tomato, and we hope to expand it out to other species. So my other graduate student, Michael, was working with Ryan, but came up with a slightly different approach, which is kind of illustrated in the next slides. So he wanted to completely avoid tissue culture, because you could argue, that was one of our missions, and a lot of what I showed you still involved growing seeds and seedlings aseptically, inducing shoots and rooting them aseptically before moving them to soil. So it still has a fair bit of tissue culture flavor to the approach. But Michael wanted to completely get rid of tissue culture. And so his approach is actually probably best illustrated on the next slide. His approach was just to take soil-grown plants and then remove the shoots and then deliver to the cut site the reagents. And the reagents, again, are the developmental regulators expressed under various promoters and gene editing reagents, CRISPR-Cas9, targeting phytoene desaturase. So the idea here is that you induce a de novo meristem at the cut site, and if you edited those cells from which the meristem derived, you would create an edited shoot and floral meristems and flowers, and then seeds, and then the gene edit would be transmitted to the next generation. That was the ambition. So he used similar constructs to Ryan. The next slide is just a repeat from the slide I showed previously. Some of the early work was done with the luciferase reporter at the top, along with the developmental regulators. And the next slide shows some of what he observed. Of course, in a plant like Nicotiana benthamiana, if you remove the apex, the plant is induced to form meristems and new shoots anyway. And what he found is in the first 3 weeks or so, most of the shoots that formed were wild type, were not transgenic, did not contain the luciferase reporter. But after about 3 weeks, the shoots that emerged, you could see luciferase positive tissues. And you can see from the picture of the leaf-like structures that are emerging, many of them are still a little bit developmentally distorted and not what you predict for a wild type shoot, suggesting that they are indeed transgenic. So as these shoots grew, the next slide shows what was typically observed. So on the left, well, in both images of the plants, the boxes represent the site where the reagents were injected, and then the circles represent the site where the tissue was harvested. The plant on the left is clearly developmentally distorted, but it's also luciferase positive, so it's probably ectopically expressing those developmental regulators, which are perturbing growth and morphology. The plant on the right is much more developmentally normal. However, it's still gene edited. So if you look at the gel for 115-2, this is an analysis of the target site in phytoene desaturase. Again, we're amplifying that target site up, subjecting it to restriction digestion. And as you can see in the green box in lane 7, the upper band represents DNA fragments that were not digested, suggesting that there were mutations there. And indeed, sequence analysis of the amplicon verifies that, in fact, mutations are present, limiting the restriction digestion. So some of the shoots that emerged had phenotypes. So the next slide shows an example of a few of these. So the leftmost shoot is a chimeric, again, of green white tissue. Again, in order to get the white tissue, you have to have biallelic modifications of both gene targets. The rightmost picture is a developmentally distorted albino tissue. And then we see what looks to be a normal albino tissue in the middle, a normal albino shoot in the middle that's emerged from the site where the regulators were injected. So if you harvest this white tissue and analyze it, as illustrated in the next slide, indeed the target sites, again, genotype and phenotype are well aligned. I think you can just focus on the sequencing reads from the all white tissue, and you can see 90+% of the reads have mutations in the target site, consistent with the albino phenotype of the shoots. So some of the shoots were chimeric, as illustrated in the next slide, and allowed us to recover seeds and look for transmission of the gene edits, which is, of course, what you really want in the end. So if you look at the left, you can see sort of in the background, the leftmost circle is identifying albino tissue. And then if you look at the stem, you can see this albino tissue, chimeric stem with albino tissue that gave rise to an all-white seed pod. And actually, this white seed pod gave rise to seedlings that were doubly homozygous mutant for the phytoene desaturase loci, and that's illustrated on the next slide. So we did sample some of that background parental tissue, and I think it's interesting to note that at both PDS1 and PDS2, you see quite a variety of mutations. But if you look in the seedlings, only a handful of these have been fixed and transmitted to the next generation. And again, all of the white seedlings had biallelic mutations, inactivating frameshift mutations in both PDS1 and PDS2. If you go and look at that, then into the next slide and consider the green portion of that shoot, we harvested parental green tissue as well as a green seed pod and analyzed it. And that data is on the next slide. So here we have what we found is, we had a mixture of green and white progeny. And it turns out that PDS1 had biallelic -1 frameshift mutations. And so the segregation we're seeing was really at PDS2. Whenever it was white, we had like seedlings 28, 29, and 30, we had homozygous mutations, frameshift mutations in PDS2. And the green seedlings had one copy of a -48 deletion, which is in frame and presumably is allowing phytoene desaturase still to function. So again, genotype and phenotype matched well. So the next slide is probably one of my favorite plants. Here we injected reagents at 2 different sites, and we're seeing albino shoots, 2 different phenotypes, one developmentally distorted at the bottom, and the upper one phenotypically normal. And indeed, we do recover non-transgenic gene-edited shoots. So what we think is gene editing occurs in one cell, but the reagent doesn't integrate into the genome. But adjacent cells receive the reagents that express them, the developmental regulators that express them. They move into the edited cell, and then that edited cell forms a shoot, non-transgenic shoot. And so, that's ideal. It would be best if we didn't have to go through transgenesis, and we're working now on strategies to create edited non-transgenic shoots at higher efficiency. So this approach works in other plants. We collaborated with Matthew Clark in the next slide in grape. Of course, grapes readily form shoots, and we could add developmental regulators and form transgenic shoots, as shown by luciferase expression. In potato, it works super well, as shown in the next slide. So it's very easy to avoid tissue culture completely and just create gene-edited and transgenic shoots in potato. Of course, there's still a lot of work to do to optimize this, and I fear that every species is going to need to have some optimization to determine the right levels of regulators to deliver. But the ability to sidestep tissue culture and to avoid some of those genotype dependencies on which tissue culture relies would be, I hope, a great step forward. So if you go to the next slide, the final story I want to talk about is not avoiding tissue culture and plant generation, but thinking outside the box in terms of reagent delivery. And again, we heard great talks on the use of nanoparticles and alternative bacterial species to deliver reagents. But what we focused on is actually viruses. So if you go to the next slide, and if you work in mammalian systems, almost all of the most standard vectors are viruses. And so we really wanted to see if we could develop plant RNA viruses as vectors for delivering gene editing reagents. And so the story is in part about work done by Evan Ellison in terms of viruses and gene editing reagents, and my former postdoc Arjun Kakar, who was interested in developing viral vectors really for synthetic biology applications. So of course, many of you are familiar with RNA viral vectors. Foxtail mosaic virus is illustrated in the next slide. This was a vector developed by Steve Whittam at Iowa State University, but I think it's sort of representative of how many of these viral vectors work. The genomes are compact and they express from often subgenomic promoters. And so what he's done here at the bottom of the slide is he's duplicated a subgenomic promoter that expresses OR5, and you can insert downstream of the duplicated promoter the sequence of interest you're interested in expressing. And so the next slide shows a story that we actually worked on a long time ago with Dinesh Kumar and Magdi Mahfouz's lab at KAUST. And it was really Magdi who spearheaded this project and had really the idea for this particular approach initially. And so what Magdi reasoned is that, well, these RNA viral vectors are limited in cargo. So if I make a transgenic plant that expresses Cas9 and use the virus to just deliver the guide RNAs, then as the virus infects the plant and moves throughout the plant, the process of infection and expressing those guides should create mutagenized somatic cells. And the hope was that, at some frequency, mutagenized cells would form, or the guides would get into the meristem and transmit mutations to the next generation. A little bit frustrating is sort of the bottom line in the next slide of that initial study, and that is the frequency was really very low. Less than 0.2% of the seedlings that we recovered in Benthy had mutations. And so my grad student, Evan Allison, was thinking about this approach, and next slide illustrates how he approached the challenge. And he reasoned that, well, viruses are often restricted from the meristem. So why not just take those guide RNAs and add sequences that will promote cell-to-cell mobility? We just need the guide to get in the meristem. We don't need the virus to get in the meristem. And so we go one more slide. I think it better illustrates the approach. So what you see at the bottom is typical Cas9 with the guide RNA. Yiping talked about RNA sequences that can be added, in his case to recruit proteins. We added RNA motifs that promote cell-to-cell mobility. These include motifs, hairpin motifs from flowering time, an RNA that's made in the leaves and moves into the meristem to promote floral meristem induction. There are also a number of tRNA-like sequences that have been shown to promote cell-to-cell mobility. So we made these augmented modified guides. The next slide shows some of the results. These are so we deliver them to a lower leaf of the plant and then harvest the 7th or 8th leaf above after a period of time and look at editing frequency by next generation sequencing. And so here we're targeting, again, phytoene desaturases, but also an A-gamma locus. And you can see that the editing is really efficient for no augmentation in dark blue, FT, which is the complete flowering time, FT that has mutation in the start codon, so it doesn't make the protein, and 102 base motif of FT that just expresses a hairpin. So all of the guides give good editing for PDS. It looks like the modified guides seem to work a little bit better for A-gammas. What Evan noticed as the plants grew, if you go to the next slide, is that over time, you could start to see these white sectors emerge. And these weren't due to, these aren't the consequence of viral infection. These are actually biallelic mutations in phytoene desaturase that are occurring over time through infection. And the next slide shows his observation that sometimes you could get in the completely white inflorescences, which are shown on the rightmost plant in the picture on the left. And then he wanted to see, of course, whether or not he could recover mutations in his target gene. And it was really pretty exciting for FT or MFT. We're looking at almost 60 to almost 90% of the seeds he harvested had mutations in the target loci. So very effective heritable mutagenesis through infection. The next slide sort of illustrates this point in slightly a different way. It shows the extent of mutagenesis. So we have no augmentation, FT augmentation, and MFT, the mutant form of the RNA that doesn't express a protein. And what we're looking at here is progeny from given plants that have been infected and scoring them for biallelic in dark green, heterozygous for the target in light green, or no mutation in yellow. And you can see where we've added the mobility sequence, FT or MFT, you get a greater extent of mutagenesis, much more green in those two graphs compared to the leftmost no augmentation graph. Now, I said at the beginning that in introducing this story that when we published this study with Magdi and Dinesh, we really didn't see much heritable editing for just the normal guide, but you can see here we're getting, you know, reasonably up to maybe almost 25% in some plants are transmitting mutations. What we found out is that we had to learn, I guess, how to recover the heritable events. And so the next slide is from our collaborator, Dinesh Kumar. And we found that the age of the flowers and the seed pods that had a big impact on the extent of mutagenesis. So here we're looking at seedlings in which we're targeting phytoene desaturase with different tRNA mobility motifs. If you look at the very bottom row, this is the seeds harvested from the base of the plant. Most of them are green. If you look at the middle row, from the middle of the inflorescence, you can see an increasing number of white seedlings. If you look at from the top of the plant, in some cases, all of the seedlings have biallelic mutations in both targets. So this is really quantified in the next slide, where we're just sort of, the x-axis is, you know, the base of the seed pod 1 at the base of the inflorescence versus seed pod 10, and we're just quantifying the number of biallelic, doubly biallelic progeny that is completely photobleached. You can see in purple and blue, as you go up the plant, you know, you get close to 100% of the progeny have biallelic mutations. And the wild type does indeed get better, a little bit better as the plant ages as well. And so you can get, you can recover at reasonable frequency mutations even without the mobility sequences. So in all of this studies I've described here so far, we're delivering a single guide. And so in the next slide, Evan wanted to see if he could deliver multiple guides simultaneously, specifically 3 guides. So he tried a couple of different architectures. So again, we're expressing these from a subgenomic viral promoter. And if you look at the bottom architecture, we're expressing 3 guides basically in tandem. They're separated by, each guide is separated by 4 base pairs. So 1 RNA with multiple guides. The spacer in the middle construct has about a 27 base pair spacer between the guides. So they're still in tandem, no mechanism to release or remove and separate the guides from each other. But the top, however, has some of those constructs that Yiping was talking about, where a full tRNA is present that gets processed by cellular enzymes and will release individual guides. So Evan infected plants with these various constructs. If you look at the next slide, so he's targeting, if you look at the very left of the top graph, he's targeting a site in phytoene desaturase, and then 2 sites in A-gammas, guide RNA1 and guide RNA2. And these guides are 4 kb apart. So this first, he's now scoring 10 randomly selected seeds from this infected plant. And you can, if you just look at progeny 1, you can see you have a biallelic mutations in PDS3. In 1 allele of A-gammas, you have a 4 kb deletion. And and then a plus T insertion. And so the 4 kb deletion arises when those 2 guides cut and you just lose the intervening sequence. And the gel is just a PCR survey to provide molecular evidence, where you use primers outside of the 2 cut sites. And in the wild type, you get a very large fragment, but in the 2 mutant forms, you can recover this smaller 4 kb deletion and then sequence to confirm that it is indeed due to loss of the intervening sequence. So as you can see, though, out of these 10, 9 of the 10 have mutations in at least 1 allele. He then quantified it in the next slide. And it's a little bit of a busy graph, but I'll walk you through it from left to right. So the leftmost graph shows the frequency of mutagenesis when you use a virus that's expressing 1 guide, targeting A gammas the first site, PDS3, or A gammas the second site. And like the data I showed you previously, you get 50 to 80% mutagenesis in the inherited seed. If you use those viruses expressing single guides and infect them together, you can see that you actually, one of them predominates. You don't seem to get a mixed infection in a given cell. So there seems to be some mechanism for virus exclusion. But then if you look at the 3 different architectures, the one with the tRNA, the spacer, or the direct repeat, they will just focus on the spacer SMPV. So again, these are the 3 guides from 1 transcript each separated by 27 base pairs. You get pretty good mutagenesis at individual targets, the light blue, the orange, and the dark blue, 25 to 50+%. But then you see you get really a pretty good robust editing at 2 loci in bright pink or all 3 loci in lighter pink. So indeed, you can do multiplexed editing. Now, this plant was expressing Cas9. I would love to have a plant that's expressing Yiping's Cas12a and try the same approach, because he saw just such remarkable efficiencies for multiplexed editing. So I think, there are certainly other strategies and other opportunities to improve upon this approach. But in the next slide, it's just that sort of summary slide again. We would love to ramp this up so we can make one person can make thousands of mutations per year. So you can think about littering promoters with sequences, or knocking out pathways, metabolic pathways in 1 round of infection. And also you can take those mutant seedlings, that are produced in the first round and re-affect them again. So in 2 or 3 generations of the plant, you can hit multiple genes and create quite a bit of genetic variation. So we and others are of course implementing this in other plants, and I think very soon there will be reports of this working certainly in other dicots and hopefully soon in other monocots. So the final little bit I want to talk about is sort of additional improvements that we're trying to make in these RNA vectors. So if you go to the next slide, it really just illustrates one of the handicaps of the plus strand RNA viral vectors. So most of them can only really handle about 1 kb of carbo. If you go above 1 kb or try to encode multiple genes simultaneously, you often get lost through recombination and your vectors become really ineffective. There's a really interesting paper that just came out on negative strand RNA viruses suggesting that they can in fact carry larger cargo, including an entire Cas9 and guide RNAs. So, that might be another approach to avoid having to make the transgenic plant altogether. So Arjun Katkar in the lab, a former postdoc in the lab, was interested in coming up with strategies to deliver more cargo with these RNA viral vectors. So the next slide just sort of simply illustrates his approach. Rather than, as in the left, embedding the cargo within the virus, his idea was to make sort of a synthetic satellite virus. So these satellite viruses exist naturally and are kind of move along with the virus through infection. So he wanted to make satellites that would move along with the main virus, but also carry cargo of interest. So the next slide shows sort of the first experiment he did with just luciferase. So if you can see the different configurations at the bottom. So he's delivering luciferase just on a tDNA, embedded in a wild type virus, or as a satellite virus. So in the leftmost part of the graph in light gray. And you can see that luciferase expression as a satellite really kind of beat the other two delivery strategies. And it even persisted up to 41 days and continued to be expressed at really quite robust levels. So actually, if you go 2 slides, you'll kind of see where he's really trying to head with this approach. Ideally, he'd like to have a whole suite of synthetic satellites that are simultaneously mobilized by the main helper virus. And you could imagine then each satellite could carry a different gene, perhaps different genes in a biosynthetic pathway. And so you could really then, through infection, perturb metabolism. So he tested this out using, as shown in the next slide, genes that are involved in this fungal bioluminescence pathway. So he was pretty excited when he read about this discovery, this eukaryotic fungal bioluminescence pathway, in which basically you synthesize the luciferin, and you don't have to add ectopically a reagent like luciferin in order to get life. So if you look at the upper leftmost, caffeic acid, which is a derivative of phenylpropanoid biosynthesis, is really present in all plants, can be converted by 2 of these fungal enzymes to histidine and then into a luciferin. And then there's a fungal encoded luciferase that creates an unstable intermediate that emits light and then generates a product that can be converted back to caffeic acid. And so Arjun had shown that this indeed works in plants. If you go to the next slide, you can see he's delivering now all 5 of these enzymes, either in infiltration to Nicotiana leaves in the upper picture, or here into the petals of vinca plant. You can see this nice, beautiful light being emitted from transformed cells that are expressing all 5 of these enzymes. And so what he wanted to do in the next slide is sort of test whether or not he could create this suite of satellites. So he made 5 satellites with the luciferin biosynthesis enzymes, those 3, the recycling pathway, which creates more or converts the end product back to caffeic acid, and then of course the luciferase itself. And if you go to the next slide, he could deliver these reagents and monitor over time for persistence. So 3 weeks post-delivery, he was still getting really good luciferase expression, and you could measure that indeed he was getting products from all 5 of the satellites. So again, the ultimate idea would be, from a gene editing point of view, would be to deliver your Cas9 and the like, and guide RNAs simultaneously, so that you wouldn't have to generate a transgenic intermediate, and which I illustrated we used in those studies, and delivered the guides to infection. It'd be great to add all of the gene editing components simultaneously through infection. So I think I'll just wrap it up there with sort of my introductory slide, which is the next one. I told you really 2 stories of how we're trying to overcome some of the bottlenecks that face us in the gene editing field. The need to go through tissue culture. So it would be great if we could introduce reagents that would create edited organs that would produce seed. And we've been thinking hard about, as I described, novel vectors such as RNA viruses to deliver gene editing reagents. And so the final slide is the folks who did all the work, my lab at the University of Minnesota, and some of my collaborators, Matt Clark in GRAPE, Danish Kumar on the RNA virus work, and Takato on some of the work with the luciferase. So wrap it up there, and be happy to take any questions if there are any. Thank you.
Thank you, Bill. Thank you very much, Dan, for that presentation. A lot of great details, and we have multiple questions in the chat for you. So going back to the first one that came in was asking the reasoning behind choosing Wuxul in combination with STM.
Yeah, so my students really just surveyed the literature for regulators that are important in meristem formation and maintenance. And then since they had a quick and easy assay, they could just try them in all sorts of various combinations. So it was more like, let's use the assay is easy enough. Let's just try everything that under the sun and see if we can find some combination that will induce a meristem. So it was more of just throw everything into the pot as opposed to a hypothesis-driven approach, I would say.
Great, thank you. Next question was asking about the percentage of chimeric plants, and if you have any insights into why it's a lower rate maybe than expected.
Fewer chimeric plants than expected. I mean, I actually think, like when we induce the meristems, I think most of the shoots that we recover are chimeric to some degree. And you don't really sort that out until you pass through the next generation. And I think the same actually goes through the virus infection. I think we're creating all sorts of mutations, and then, you know, that small fraction of cells in the meristem that receive the guides, you probably get a mixture in that cell population also of chimerism. It'd be great if we can control it more and have more predictable outcomes, but at present it's a bit of a sorting exercise in the next generation to find what you want.
Right. There's multiple more questions, so I'll keep going if that's okay with you. And then for those listening in, if you have further questions, please add them to the chat, and we'll get to as many as we can before we wrap up here. Next one for you, Dan, is how often do you see evidence of new mutations in progeny, and is there a difference when using Cas9, CPF1, or talons?
Well, we actually have really almost done everything with Cas9 and haven't compared. The talons, for the approaches I described, you know, they're pretty big proteins and they take up a lot of DNA. So probably would never get one into an RNA virus vector. Plus, they're also highly repetitive, so they would probably recombine. So I have to say, I would really love to try some. Particularly, I've been motivated by Yiping's talk to try some other Cas families of reagents. You know, in addition to native enzymes, some things like base editors and I think could really open up opportunities. So you just don't recover indel mutations, but specific base changes in your target of interest. So I can't really, I mean, in summary, I can't really answer it because we really haven't done the comparison, but it'd be great to do so.
Mm-hmm. Yeah, agreed. Another question is asking about the RNA size limit when fusing it to a movement sequence, if there is an RNA size limit.
Yeah, so I think the entire cargo, we try to keep our entire cargo less than 1 kb. I mean, so your guides are pretty small. We have no problem with expressing 3 at a time. And some of those mobility sequences are, you know, tRNAs and tRNA derivatives. So they're only less than a few hundred base pairs as well. So we can pack quite a few guides and effectively into a viral vector. I think we still have, like I showed you, really good activity with 3. When we go up to 5, like the first guide in the array and the last guide in the array seem to work, but the middle ones never seem to hit their targets. So I think there's some things we can learn about the best expression strategies. But I think you should be able to deliver, once you come up with the strategy, you should be able to deliver 5+ guides without too much problems if you use small mobility sequences, like the tRNA stem loops.
That's related a bit to the next question, asking if you've tried different movement sequences with the same guide RNA. And if so, is there a difference in the editing rate that depends on the movement sequence?
Oh, it's a great question. You know, we're now like stepping back and trying to learn all the best practices now that it works. And yeah, don't have enough data now, unfortunately, to answer that question, but we would love to do those kinds of experiments. Come up with the best strategy to get the most effective editing.
Right. And does the viral delivery of guide work in plants that are heterozygous for the nuclease? Or do you know?
We haven't actually tried it, but I think it certainly should. As long as you're expressing the nuclease, you know, that's the key to have it in the target cells. And we, you know, in that regard, we haven't tried many different promoters. We've just blasted them with the ubiquitin or, 35S or the like. There might be promoters that would express in the tissues that we really want to mutagenize. That would be, better to use, and that's another area of research.
For sure. We've got 3 more questions that have come in. I think we have a couple more minutes to get to them, and then we'll wrap up. So, specific one on the reasoning for using a 27 nucleotide spacer. And did you try, which is related to the other questions, but if you try different lengths?
Yeah, it was fairly arbitrary. My grad student put them together in tandem with 4 base, restriction enzyme site between them. And I think he just had a construct conveniently that worked, and it happened to have those 27 bases. So again, it's a common theme of the question period is that we just tried our darndest to get it to work, and are now trying to learn the lessons of how to make it work the best.
Right. Some of that's the most exciting research too when you get on those.
Yeah. I don't know if you noticed, but the sun finally came out in Minnesota. It's been like 3 days of darkness, and
yeah, it's nice to have it there for you. Dr. Lee had a question on the Cas9 transgenic lines, if they have any phenotypes, and what type of promoter was used.
Yeah, so we've used ubiquitin and 35S, and we don't see any phenotypes. And we have Arabidopsis, Nicotiana benthamiana, wheat, Soteria, rice. We have quite a collection, and don't see any phenotype by ectopically expressing the Cas. Ubiquitously expressing the Cas protein.
And how do you ensure that only true to type meristem cells are transformed? Is this a reason behind the level of chimerism? Because if true to type meristem cells are transformed, they will give rise to true to type non-chimeric shoots.
Yeah, I actually don't know what's going on at that level, at the cellular level in the meristem. And, I think there's, that warrants a lot of thought, actually. And, Mike, I'd love to talk to you about, monocot meristems, because we can infect Soteria and get somatic editing at greater than 50%, but we can never get a heritable edit. How do you get it? How do you get it into a meristem? The biology there is inherently different. And it's another area I think we can improve on the technology, is to really understand meristem targeting a little bit better. Right now, it's just a cell-to-cell mobility sequence, but I think it's all cells. Like, we see better editing in somatic leaf cells, and we just think at some fraction of the time they get into the meristem.
Great. One final question came in, so this will be the last one, and then we'll wrap up for the day. Does the number of synthetic satellites impact the efficiency?
I think that's definitely the case. And I we don't know if we can get them to move. We were analyzing the site of infiltration and the persistence of the satellite within the site of infiltration. But can we get that swarm of satellites to move up the plant and into, non-infiltrated tissues? That's going to be a big challenge. And we think about strategies of like putting essential genes onto some of the satellites so that there's an essential for the virus, like move the co-protein onto one satellite and a movement protein onto another satellite, sort of break up the satellite so that they have to work in concert and they have to move together collectively in order to get the infection. But alas, those experiments remain to be done as well.
Maybe there'll be opportunity for a future Selfie Symposium to have the group back together and get some updates together. So with that, I'll turn it over to Annie to close us out for today.
Yeah, thanks, Edward. And I really want to thank all the speakers today. Clap is the only way to show visual clapping. But again, we really appreciate the time today. Thank you so much. It was a great seminar series. Yes, we hope to do these in the future. I just want to share a brief thanks again just to my fellow organizers as well, just because, again, it was a great opportunity to get everyone together today and really hear from, Dan around wrapping together some of the tissue culture and transformation approaches with editing. We heard from Yuping Chi about advances in some of the different types of CRISPR systems. We heard from Mike around shoot development and some advances there with Mark in the pollen biology space. Was really fascinating. And then to start out the day, of course, we heard from Michael Strano and Ling Yi around some of the opportunities around different types of delivery systems, both with nanoparticles and then agrobacterium and non-agrobacterium. So again, just really want to thank the speakers today. I also put on the slide here that there's been a number of questions around the event recording. There will be an event recording. It's going to be made available to all of the registered participants today. If you have future questions for the speakers, please don't hesitate to contact them either through email or through LinkedIn. We hoped a lot of connections were made today and will spark up new collaborations and opportunities for that. So again, stay tuned through LinkedIn, and we hope to be speaking to you again soon. So again, one last time, thank you to all the speakers, and I hope you have a great rest of your day.
Thank you.
Take care, everyone.
Thank you. Take care.
Thank you.